आप छोड़ रहे थे मोटी वीडियो होता तेरे युक्त ज्ञान तेरे सुप्त ज्ञान ये विद्ये ಹೊನ್ನ ಕಿರಣ ದಿನ ಪಾಠ ಪಠಣ ಗುರು ಬುದ್ಧಿ ವಚನ ಇದು ವಿದ್ಯಾದಾನ ಕಾರಣ ಕಿರಣ ದಿನಪಾಠ ಪಠಣ ಗುರು ಬುದ್ಧಿ ವಚನ ಇದು ವಿದ್ಯಾದಾನ
Leave.
ಜ್ಞಾನ ತಿರುಸುತ್ತ ಕಣ್ಣ ವಿದ್ಯೆ ಹೊನ್ನ ಕಿರಣ ದಿನಪಾಠ ಪಠಣ ಗುರು ಬುದ್ಧಿ ವಚನ ಇದು ವಿದ್ಯಾದಾನ ಕರಣ
Those who are registered for oral and poster presentation, please drop your oral and presentation files at the register counter. And who are registered for as a delegate, please join the session. Inform others also. Please drop your files as earlier.
ಸ್ಟೆಪ್ಸ್ ಹತ್ತಕ್ಕೆ ಆಗದೆ ಇರೋರ್ಗೆ ಒಂದು ದೊಡ್ಡ ಗಿಫ್ಟ್ ನಿಬಾವ್ ಹೋಮ್ ಲಿವ್ಸ್ ಇನ್ಸ್ಟಂಟ್ ಇನ್ಸ್ಟಲಿ
ತೆರೆಯುತ್ತ ಜ್ಞಾನ ತೆರೆಸುತ್ತ ಕಣ್ಣ ಈ ವಿದ್ಯೆ ಹೊನ್ನ ಕಿರಣ ದಿನ ಪಾಠ ಪಠಣ ಗುರು ಬುದ್ಧಿ ವಚನ ಇದು ವಿದ್ಯಾದಾನ ಕಾಣ
I request all the participants to gather in the auditorium. A very good morning to one and all gathered here. The second day's technical sessions will be continued today, and we will be having about 5 lead talks today. So the speakers will be Dr. Prakash Halami, sir, who will be speaking about the health benefits of probiotics preclinical studies. Then the second speaker will be Ashok V. Bankar, sir, who will be speaking about the significance of microorganisms in nanotechnology and interdisciplinary approach. The third talk is by Dr. Anirban Chakraborty, sir, who will be talking about microbiomics, new insights into cancer diagnosis, therapy, and prevention. And the next talk will be by Professor A.P. Lipton, sir, who will talk on novel discoveries and future prospects of marine biotechnology. And the next talk will be by Professor H. Haridas, sir, who will be talking on COVID-19 variant, trial with an Ayurvedic formulation. The first lead talk of the day will be delivered by Dr. Ashok V. Bankar, sir. I request Dr. Ashok V. Bankar, sir, to please be seated on the dais. I welcome Dr. Prakash M. Halami, sir, to chair the session, and Dr. B.S. Srinath, sir, to be the rapporteur. I call upon Ms. Deepika KP to introduce our lead speaker, Dr. Ashok V. Bankar, sir.
Good morning, everyone. I'm Deepika KP of second year MSc Microbiology. It's my privilege to introduce Dr. Bankar Ashok Banerjee for the 2 days national conference at Jnana Kaveri campus, Chikalwara, Kodagu. Dr. Bankar Ashok Banerjee is a native of Pune, Maharashtra, India. He is graduated in biotechnology from Institute of Bioinformatics and Biotechnology, Pune. Done his master's degree in microbiology from Savitribai Phule Pune University and bachelor's degree in microbiology from Mahatma Phule Mahavidyalaya, Pune. Dr. Bankar Ashok Banerjee received CSIR NET GRF Award in 2007-8 and 2008-11. Foreign Travel Grant Award for 5th International Conference on Environmental Sciences and Technology in 2010 from CSIR, Department of Sciences and Technology and Department of Biotechnology at USA, Belgium, Germany. In 2012, for the 6th International Conference on Environmental Sciences and Technology from CSIR DST at USA. In 2013, from France European Science Foundation Workshop at Spain, and in 2015 for attending 6th European Bioremediation Conference at Greece. His PhD research topic was Biology and Biotechnological Application of Yarovia lipodlitica Microbe-metal Interactions under the guidance of Professor Smita S. Zinger. His research experiences are bioremediation of hazardous and toxic metal ions, biosynthesis of nanoparticles and their potential applications. Teaching experience as assistant professor in Department of Microbiology, Pune from 2003-6, 2011-12 to 2012-16 at Savitribai Phule, University of Pune, and at MES Abasaheb Garwar College, Pune from 2016 up to date. He had attended 18 international conferences, 15 national conferences, and 3 workshops. He had published 47 research papers in international journals, reviewer of 17 international journals. During the period of 2019-22, a major research project awarded and huge amount sanctioned for 3 years. Member of editorial board and conference organizing committee. He worked as NAC steering committee member, worked as observer for SET examination. He has been invited as judge, as research person, and as guest lecturer. Selected for expert scholarship visiting researcher to Göttingen University of Germany in 2011. Guided several projects for MSc, MTech, PhD students along with effective teaching and examination work carried out. Thank you for being here. I really appreciate that you took time to be here. We hope your promise may make a change in the young minds. Thank you all.
Thank you, Deepika. I request Dr. Ashok V. Bankar, sir, to deliver a talk on the topic Significance of Microorganisms in Nanotechnology and Interdisciplinary Approach.
So good morning, everyone. I really thankful to Professor Jaya Shankar sir and team and whole Mangalore University to invite me as a resource person uh, to talk on the topic significance of microbes in nanotechnology, and it is an interdisciplinary approach. So student, mostly I prepared this presentation uh, uh, by keeping the view of students towards the research. That means this presentation is much more useful for the PhD students as well as for the postgraduate students, as well as the UG students also. So this presentation is student-oriented presentation. So if you have any doubt, any questions, you can ask me at the end of the presentation. So in between that, you can write your queries and questions. So I will start. Okay. So first thing is that what is the nanotechnology? So nanotechnology, all of you know that now it is an emerging field. And in nanotechnology, we are using the nanomaterials in the nano size. And these nanomaterials or we are using in biology. That's why the two branches are coming together. One is a biology and another one is a material science. So it is a combination of, and that is called as a bio-nanotechnology. So why it is necessary to come together with the interdisciplinary approach? Because this approach has several advantages and several applications that will be useful. So these nanomaterials have some unique properties, and that properties can be exploited or that can be explored for the applications in medical field as well as in another field. So we can synthesize nanoparticles by the various methods: chemical, physical, and biological methods. So out of that, the biological method is much more easy and it is useful because it is simple. We can easily prepare nanoparticles. We can control the size of nanomaterials, as well as it is eco-friendly. That means it doesn't produce the secondary pollutants or toxic material. So to avoid this, so biological material is very important. Then what kind of material we can use for the nanoparticle synthesis? So simply there are plants, fungi are there, yeast cells are there, then bacteria are there, algae are there. So interdisciplinary, we all can come together, whether botany, zoology, microbiology, biotechnology, chemistry, physics, all branches can come together and work. Because research is not the work of the one person or two person. So it is not competition. So instead of that, research is a cooperation. So if we have good cooperation, we can take a good team. So why nanomaterials are important? So first thing is that they are a very small size, and these nano devices can be useful. They can easily penetrate through the animal cells. So it is beneficial in cell culture and all that things. So they can easily penetrate, they can easily interact with the DNA. Also, they can easily interact with the different organelles in our body also. And that is the reason the nanomaterials are the important, and they can easily enter. So there are various plant materials that you can apply for the nanoparticle synthesis. If the people are from botany background, they can utilize their expertise because they know the different types of the plant materials. So by using the simple methods, we can prepare the nanoparticles by using the different parts of the plants. It may be the roots and shoot or the leaves, because they contain a lots of phytochemicals, and this phytochemical interact with the metal ions and they can convert them into the nanomaterials. So synthesis of nanomaterials, we can also apply the microorganisms. So there are different types of the microorganisms. It may be bacteria, then they can synthesize the nanomaterials. So you can see that on the surface of these nanomaterials, they can produce even inside the cells also. Then these cells are also there. Then you can see the scanning electron micrograph here, clear picture of the yeast cells and how this yeast cell can be beneficial. Then fungi are there. So on the surface here, you can see that how the metals, they are interact with fungal cells and they form nanoparticles. Then there are other materials, that is biomolecules, DNA, proteins and all that. So nanoparticle is not mean that only the metals. Okay, so it may be anything, any biological material that is under the nano size 1 to 100 nanometer. That is called as a nanomaterial. So this is simple protocol for the PG students, so they can understand how it is simple. So it is just interaction with bacteria with the metal ions. You can prepare it in the lab, and then we can find the results, and then we can characterize them. So there are two methods. I mean, microism can synthesize either extracellular or intracellular. Extracellular, that means outside the cell. They are synthesizing different enzymes, so the enzyme may play important role, or cell wall may play important role, or any other biomolecule, they may play important role. So it is a part of investigation. So students, here it is not well understood that what kinds of the biomolecules they are involved. Some molecules are known, but there is a much more scope that you can think on that. So I never underestimate the power of students, because students have so much power, think in innovative way, and that is the reason. So I always take the ideas from the students. Maybe it is MSc student or it may be the PhD student. So we should not underestimate their knowledge. So we have to encourage our students. So extracellularly also we can produce the nanoparticles. So simply we can characterize, say this is the example of the word nanoparticles. So we can characterize with the UV spectrophotometer. So here you can see the different peaks according to the color. So color is changed because the properties change of nanoparticles. It is depending upon the size. As the size goes or decrease, increase, so color will be changed. That is called as the plasmon resonance. So that can be, you can easily estimate. So then we can characterize with the transmission electron microscopy, TM. You can do the SEM, scanning electron microscopy, and then you can analyze their component with the EDS, that is, it is a spectrophotometry that it will analyze what content is there, whether your nanoparticles is of gold or silver or any other metal or zinc or calcium. So it will give the composition of your nanomaterials, whether it is a pure or impure, it is mixed or it is composite. Okay, that you have to prove. So it will give the idea about the composition. Then there are various types of the nanoparticles we can prepare. There are different shapes and sizes, and nanoids are there, triangle and hexagonal, circular. So here you can see that the circular nanoparticles. This is the transmission electron microscopy. You can avail the facility by collaborating with the other institute where this facility is available. And you can characterize your nanoparticles. So here you can see that there are triangles and hexagonals. Different types. Here you can see that in bacteria how different shapes and size of the nanomaterials will be. Nanorods are there. So here you can see nanowires you can prepare. Then nanotubes you can prepare. Then nano sheets we can prepare. Then nano flowers also. So we can design the nanoparticles depending upon our requirement. This is the nanodendrites that we can synthesize. So there are other methods also, like XRD. We can find out whether your nanoparticles are very crystalline in nature, and what is the arrangement of the atoms that we can easily understand by this XRD. I will not go in detail because it is again theoretical part. So we have the International Center diffraction data. By using that data, you can find out that what is the status of your nanoparticles, whether it is a crystalline in nature or not, that we can easily, most of the nano, they are crystalline in the nature. So this is a diffraction pattern that we can match with that files. And if it is a match, then you can say that it is a crystalline, you can understand the arrangement of the nano. So this is a dynamic light scattering. This is a DLS. So DLS is the instrument where you can analyze the nano size. That means average size we can find out whatever the nanoparticles you have synthesized, whether it is a 1 to 10 nanometer, or it is a 10 to 20 nanometer, or it is a 20 to 30. What is the range of your nanoparticles that we can easily find out? So like that you will get the graph, and average size we can find out by using the statistical analysis. So then FTIR also you can use. The main function of this FTIR is that when you are using the biological materials, so by characterizing before and after interaction with the metal ions, so which functional groups are involved for the synthesis of nanoparticles, that we can analyze whether it is an amino group or carboxyl group. So like that, this nanotechnology has several applications in medical field, in environmental science, and drug delivery and all that. So many applications are there. So because of their application in agriculture field also, again, this is a very important thing. So it is important in biomedicals, healthcare and food, agriculture, industries and environment and electronics. So everywhere we can apply this nanotechnology. And that's why it is important to understand the basic things of the nanotechnology. So apply your theory in practical. Okay, so you are studying your theory. But unless and until, because theory is evolved from the practical. Okay, so first practical is happen and then theory. So now you are studying the theory, but this theory, if it is in your mind, no use. it should be in practical. You know so many things, but it should be in the practical. So work of creativity and innovation will be there. Okay, so if you think innovative way, in different way, so when you are publishing in your research paper, what they ask reviewer, what is the comment? First comment is that there is no innovation in your research paper, and that's why it is rejected. So this is the 99%, 90 to 99%. This is the reason for the rejection of the research paper. If your paper is good, there is a innovation, they will accept you. And that is the reason people are finding the shortcuts. They are publishing in the predatory journal. They are giving money 5,000, 10,000, 3,000, and they are publishing within night or within 1 month. So that should be avoided. So when it will be avoided, so we have to train our student in such a way that we should not go in that part in predatory journal. We don't have to pay. Okay. So without paying, then we need to publish it. Then how I turn towards the nanotechnology? The first, because how I got the idea why should I should work in nanotechnology? Because of simply this review. So I written this review, environmental and industrial application of Yarovia lipolytica. So Yarovia lipolytica, it is a dimorphic yeast. Okay. So when I written this review, then I understood the different applications of the Yarovia lipolytica. Then I understood that there is no any application of Yarovia lipolytica has been utilized for the synthesis of the nanoparticles. So this review then published in the Applied Microbiology and Biotechnology. I purposely written the journal's name so that it is an exposure to the MSc Part 1 students. So they will understand what is the impact factor, what is the journal, what is the brand name stranger. So you should understand this from your basics. I teach my students from MSc 1, from BSc also. Okay, so they understand what you mean by impact factor, what you mean by citation, what you mean by stranger publication, what you mean by elsewhere, what you mean by ACS. So if they know these brand names, then they will dream for that paper only. They will not dream for the predatory journal. That is the main purpose. Okay, so then we studied some. I took some MSc students and just we studied this Yarovia lipolytica on the different metals. So I found that there is a morphological changes in colony. By considering by simple observation with the students, then what we did, we published this research paper in the Canadian Journal of Microbiology. Again, the impact factor is 3.2. So student gets benefit from the publications because they get admission in abroad, in Germany, in Max Planck, and everywhere if they have the good publication. Okay, even they will get the opportunity for the postdoctoral student, as a postdoctoral fellow. So then we observed so many things. Then we did the SEM, scanning electron microscopy, and we observed so many changes. You can see that this one is control cell. Here, this one is control. And after the treatment of the metals, so we found so many changes in the cells. So response is vary from metal to metal. Okay. So as we change the metal, the response was different. Here you can see that the morphology has been changed. Then we notice here the diameter has been changed. Then we did the morphometric analysis. Then we found something here, precipitation is there. Then we understood that something is going on with Yarovia lipolytica. So then body pattern was also changed. So then we did the morphometric analysis. So in morphometric analysis, we found out that surface to volume ratio was reduced as we are increasing the metal ion concentration. So this was again the first time observation in Yarovia lipolytica, and this is because of the with the help of student. Then we did a simple light microscopy. Here you can see that these are the control cells. After the trace, the pseudomycelia were formed, elongation was formed, and that was a monitor. And with the help of the software, and we find out how much length is increased, and then we took the average size and then we plotted. So it is found that as we are increasing the a little bit concentration, elongation factor is also increased. So in this way, that paper is published in Extremophiles because these two strains were from the marine source. And what was the logic in the marine environment? The metals, these cells are exposed to the metal science, different metal science. Oil spills are there. So in petrol, diesel, the leaded petrol. So some metals are there, and in sediments also there are some metals. So these organisms coming in contact with the different metals. So doing the simple thing, we just check the tolerance, and with the tolerance, then this organism was able to form a biofilm. So we studied the different patterns of biofilm in presence of the heavy metal, and different patterns were observed, and the quantitative study has been done, and later on we did the transmission electron microscopy. So then after the transmission electron microscopy, it was observed that so this TM images we got from the Colorado University, USA, in collaboration. The people like our work, so they said that send your samples, and they took the sections, and then they send all these results. So I observed this. These are the particles. That means metal particles are there, and in cell wall also there are some particles. So it may be the nanoparticles because it is in nano range. Okay, so you can see the these nano range particles were there observed. So then I observed that cell wall here plays very important role to detoxify the heavy metals, and when the metals are goes inside go inside, at that time some protein may be playing important role to detoxify the heavy metals. So that means some protein may also play the important role for the nanoparticle synthesis. Here you can see that. So maximum for the chromium 6. So maximum here cell wall plays very important role. Cell wall is binding these metal ions and not allowing to go inside. The reason is that chromium 6 is highly toxic. So then we found that why with other metal like zinc and all, the metals are going inside. But why for the chromium 6, metal is stuck on the cell wall. Then I found that this chromium 6 is highly toxic, and this organism have ability to convert chromium 6 to 3. That is a less toxic material. Okay, and that is happening on the cell surfaces. So this is the major observation, and then we found that so this organism also having ability to convert chromium 6 to 3. That is called as a chromium 6 reduction. So then later on we found that the chromium nanoparticles were formed, and this is the start of the new journey, that we understood that this is happening, color is changed from chromium 6, then scanning electron microscopy done, but it was very blurred. So it was not up to the mark, and such types of the diagram will not take in the good journals. So we did further this transmission electron microscopy, and then we understand then XRD pattern, then scanning electron microscopy, so how the cell wall is interacting with the metals. And then we isolated the proteins which is involved in the metal binding. So I designed the column that is called as the IMEC column. It is available in the market, but we charge that column with the metals of our interest. My interest was with the chromium 6. So that column was charged with the metals, and then we found that what kind of the metals are binding. So we did the 2D gel electrophoresis, then marked it off, and then we found that there are the different proteins. They are in Eurovia lipolytica, they are able to bind the metal ions, and these proteins may be play important role in the detoxification of heavy metals. Then we did the 2D gel electrophoresis in presence of metal. In absence of metal and in presence of metal, what kind of the proteins will be expressed? So some proteins were upregulated, some proteins may be downregulated. So upregulated protein may have important role in the detoxification. Then what kind of the applications? So then I attend one refresher course in Mysore University, and Gita madam from that department from biotechnology, she gave me opportunity because she was also working on that. So we written some book chapter that is a bisorption of heavy metals, and it was a published. So then from that we got the idea, and I did the practically that with the Eurovia lipolytica, and then adsorption of heavy metals on the surface of the cells. We studied that, also did the so many mathematical modeling, Fredlich Langmuir models and all that things. And this paper is published in the Journal of Hazardous Materials with the impact factor is 14.2. Okay, so because no report was there, this was the first report in the Eurovia lipolytica, and that is from the marine source. Then I always gather the students from MTech and MSc, and we all together work together, and that's why whatever the names are there with me, all they are students, because I never I always give credit to the student. Even the one part is done by the student. I put their name because we should encourage the student. Even they are helping to prepare the plates and culturing and isolation. Even we put the name of students in the research paper because it is an encouragement for them. It is a benefit for them. So then what we did, we removed the hexavalent. As I said, the hexavalent chromium is highly toxic. And that is the reason here, Yerovia lipolytica, we made a composite with the some nanomaterials, that is the Fe nanoparticles, and we reduced the chromium 6. And this paper was published in Journal of Contaminant Hydrology. You can see the impact factor is a 4 for this. So then another work that we did with this Yerovia lipolytica, we removed the nickels. Again, nickel is another problem to the environment, and it is published in the Journal of Environmental Management with the impact factor 8.9. So whatever the student, they work with the high impact factors. Now they are in abroad, they are doing their PhD, some are doing postdoc. So this is a benefit for them. Okay, so their journey is very high. So sometimes they also help me to do the analysis, even they are in abroad. So this is a benefit. This is a benefit of our student. And then we prepared the biofilm, and biofilm was of Yerovia lipolytica was also able to remove the chromium 6 because the biofilm is a form because of the exopolysaccharides, and this exopolysaccharide plays very important role in detoxification of the heavy metal ions. Then then later one of my students who was working in the biochemistry, and he said that he was working on the fly ash. So what I said, send me the fly ash. So he gave me the fly ash. We did the analysis. Then it is found that fly is the product of the mostly the thermal power plant. It is continuously generated, and they contain the heavy metals. When this fly ash it is deposited in the soil, at that time it gets contaminated, metal get polluted. I mean metal get added into the soils, and again it is a problem because metal cannot be degraded. It is accumulated. Okay, we can transform the metal, but metal cannot be degraded, so it will remain as it is. So then it can be transferred to the food chain. Okay, from soil to plant, and plant to human being, and then human being to another. So it's the food chain. So what we did, we analyzed the fly ash and we chelate or we leach out the heavy metals from the thermals by forming the biofilm, because this organism, I found that this organism is able to form a biofilm. So we took the particles of the fly ash, and on that we grown the Yerovia lipolytica, and then we found out that leaching out these heavy metals. So thing is that one thing is what found that this organism, I written the review, and I understood that in review, this Yerovia lipolytica was able to synthesize the citric acid. And then literature survey tells that citric acid is a leaching agent for the heavy metals. Okay, so that logic we found. Then we took the citric acid and we analyzed the citric acid, whether in presence of metal and in absence of metal. So it is found that under the trace of the fly ash, the citric acid was enhanced. So these indicate that the citric acid has an important role as a leaching agent. So then what is the application in the nanotechnology? So actually we work on the then we move towards the nanotechnology, synthesis of the silver and gold nanoparticles by using the some melanin derived from the Yerovia lipolytica. And again, this is published in Journal of Nanobiotechnology. Impact factor is a 9.4. Okay, so here why we could publish, because there is some novelty. Nobody has did this the way we design the protocol. So I always encourage the student, think in innovative way, don't go in traditional way. So what you are doing, you are taking paper, you are reading it, and you can making the protocol. Instead of that, use your brain, change the protocol, break the tradition, and design the innovation. Okay, so if you do that, then and then your research will go ahead. Okay, so that is very important. So then again, we use another strain, that is NCYCM, and we also synthesize the silver nanoparticles, and we found that it is affecting the pathogenic biofilm. So pathogenic biofilm was inhibited after the treatment of the nanomaterials. So again, this was a melanin derived, but in the protocol was modified, so that was not the repeated protocol. It was a different, and that's why we could publish in the AMB Express. So then another strain that is aeroveliphylatic. Again, we modify the protocol. Okay, so you can modify the protocol, you can design your own innovation, and result was that again some cell-associated gold nanoparticles were formed. Again, we inhibited lots of pathogenic biofilms, and again it is published in Madras. Later, again it is interdisciplinary journal. You can publish your data, which is I mean interdisciplinary biology and material science, physics related or anything. So anti-biofilm agents was designed in this way. Then Ashit Rao is one of the MTech students. Now he is doing postdoctoral. He is a postdoctoral fellow in Germany. So we synthesized the nanowires, silica nanowires, and on the silica nanowires, what we did by using some chemicals on these nanowires, we attached the lipase enzyme. Okay, and it was found that now traditionally how we do doing the immobilization, we are using the sodium alginate gel. So instead of that sodium alginate gel, here the nanoparticles were used and the enzyme was immobilized, and then by giving them support on the gene, then it was a recycle. It was 7 times recycled and used. That means we can use the enzyme without losing its activity. And you know the lipase is a very industrially important enzyme. Then the SYBSc student, that is Bhagyashree Joshi, she worked with me for the KVPY, Kishore Vaigyanik Puraskar Yojana. So all of you know that. For this scholarship. And then we published by using simple banana peel extract and their antimicrobial activity. So we published 3 papers on this banana peel extract mediated nanoparticles. So we prepared gold nanoparticles as well as we prepared the silver nanoparticles. Again, it is published in very good SYBSc students if she is getting the 3 papers in that year. So then Vice Chancellor of Pune University came and visited the department and said that you are eligible for the PhD. He spoke with that student. I told that student, don't find a shortcut. Even you are eligible for a direct PhD, don't find a shortcut, complete your MTech and then go for the PhD. Then she completed her MTech, she completed her PhD in ISAR, and now she is in abroad for the postdoc. So this is the benefit for the students, and this is editorial. Then we written on the palladium, and then again palladium nanoparticles were formed and dendrites by simple. We use the banana peel, but for 3 papers. Why it is possible? Because of the modification in the methodology. Simple change, modification, different way, and different results are there. Okay, so if innovation is there, automatically your paper will be published. And then another student came, that is Sandeep. He is from the MSc Part 2. He said, I want to do dissertation work, but I want to work on banana peel only. So I told him that already there are 2-3 papers on the banana peel. How we are going to publish this again? Because there is no innovation. Then I said, bring some innovation. Then we'll go for that. Then he said that, sir, we can prepare the composite of gold and silver, and we can check their efficiency against the biofilm formation of the pathogenic bacteria. I like that idea. We proceeded, and then it is published in the that is Bulletin of Material Science Journal. So then another, that is this is the another story is this pomegranate leaf extract were used for the synthesis of nanoparticles. So now you say that why there are lots of reports on the plant material. Why this paper is published? The basic thing is that the important part of this work is that the protocol is in this way. Within 5 seconds, nanoparticles were prepared. Okay, so that was the phytochemical power of this plant. Then later on, we found that which is responsible. So it is found that the small peptides. So we found the mechanism. Small peptide is responsible for the nanoparticle synthesis. Then we sequenced that. It is found that it is 9 to 10 amino acids were present in that peptides. So then phytoapplications. We prepared the AP nanomaterials compositions, and we use it for removal of the hexavalent chromium, because my topic is to protect environment also. So hexavalent chromium is highly toxic, so it is necessary to give the treatment to the chromium. So because these are the magnetic nanoparticles, we can easily recover. We have to put magnet and we can easily recover the nanomaterials. Again, you can use for the second cycle. So that is the advantage. So we did some collaboration work with the physics department. So they did some irradiation of nanoparticles. So then whatever the irradiation they have did, we developed biofilm on that, and it is found that once they do the irradiation of nanomaterials, biofilm was not able to form. So inhibition of biofilm was studied, and then it is found that this is a very good method by using the microwave assisted method that is developed by Professor Dole from Pune University in physics department. So then also he gave me the material. That material is mostly used, that is the polycarbonate, that is mostly used for the aeroplane material. So he said that mostly on aeroplane. So there is, you know, that in COVID-19, you know that major chances of infection is in aeroplane only, because the closed environment and whatever the material that can support for the microorganism to develop the biofilm. So lots of infection can be takes place in aeroplane. So he said that what we can do to avoid the biofilm formation on this material. Then he irradiated. Then I said that change the that is fluences irradiation rate. So they vary the irradiation fluences, and it is found that as we are increasing the energies, and biofilm formation was decreasing. And why it was decreasing? Then we investigated the mechanism, and we found that because of the formation of the pores on that materials, so it repel the microorganism, is not able to form a biofilm, because for the formation of the biofilm, solid material or the substrate is required, and that was not allowed by after the irradiation. So similarly, on similar way, we publish another paper on that. And in this way, we did the scanning electron microscopy, how the biofilm was reduced, and as we are increasing the irradiation. And then we found out the mechanism. What could be the mechanism that is playing important role here? So it is found that these nanomaterials are very toxic to these pathogenic organisms. And then also we isolated the Acinetobacter from the food material, that is the fruits, and we found that it has ability to produce the exopolysaccharides. So exopolysaccharide is outside the cells, and then it is found that this exopolysaccharide is responsible to form a nanoparticles in of uniform size. Then we did the scanning electron microscopy, and you can see that how on the surface how these nanoparticles are formed, and this is the APS layer. So in APS layer, exopolysaccharide layer, nanoparticles were formed. That means the pathogenic biofilm was inhibited. So then another my student, he said that I want to work on the bacteriophages. So I said, what is this bacteriophages? I don't have the experience, lab experience on the bacteriophages, but they have practical of the isolation of bacteriophages. Then they isolate some phages and they show me these are plaques are very good. I said, purified it. Lysate, prepare the lysate. So then they prepared the lysate and brought that lysate. And it is found that this was a very rare phage. Okay, that we isolated, and that work has been published because it was rare. Then I used the same Podoviridae family phage because it was rare, and we prepared by using the bacteriophage. So this was the first report, and it is published in the Indian Journal of Microbiology. And this paper at that year got the maximum citation. So we got the award for this paper from the AMI. Okay, so because of the good citations. So at the end, some MSc students, then they are now working on this. They are we have prepared the yeast biomass. We already have the strain. I am working on 2 strain, that is Aeroelia connectica. So what student did? They form a yeast biomass, and then we prepared the palladium nanoparticles by using plant materials, and these nanoparticles, once they are formed, we add some on the yeast surface. Now you can say the change color is a change because of the black color of nanoparticles. Because this, I already know that Aeroelia colitica have ability of adsorption of metal. So nanos were adsorbed on this. So we say that it is a palladized yeast biomass. Now what to do? Now we immobilize this biomass and in the sodium alginate gel, and we prepare the beads, and then it is formed immobilized palladium catalyst. Now what is the next? We don't have the expertise further, so we collaborate with the chemistry department, and I told my colleague that can we perform some reactions? We have ready-made catalyst. So tell me some reactions. So he said that yes, sir, you can use that catalyst in this reaction. So the industrially important amines will be formed. So we form different amines. We characterize the nanomaterials. You can butterfly-like structures are formed, XRD and all that things. And later on, this immobilized cells with his cells, you can see the chromium 6, they were so adsorb. So we also remove the chromium 6. Scanning electron microscopy, here you can see that the this is a uh, sodium alginate bead. Then after taking the section, you can see that the cells are added uniformly and they are removing the chromium 6. This is a control. And in this way, now this paper is communicated to the materials later. So this is my simple message to all the students. So you have lots of power. While doing your research, during your career, something wrong can be happen also. Something negative can be happen. But in every situation, you have to be very positive in your life, during your research, during your career. If you are positive, then and then it is possible. So you have to think in different way, innovative way, positively. I think I want to show one video. I think it is not playing here. Can you try, please? So look at this video carefully. Yes. Just have a look, and then I will talk later. This is competition of Barcelona in 1990. And he is a great runner. He is very confident that he will win. Correct? It's fun now.
When I am down and oh my soul so weary, when troubles come and my heart burdens deep, then I am still right here in the silence till you come and sit a while with me. You raise me up so I can stand on mountains. You raise me up on stormy seas. I am strong and I am on your shoulder. Raise me up to more than I can be. You raise me to walk on stormy seas. Yes, I am strong when I am on your shoulders. Raise me up.
So this should be our approach. You may feel, but here you can see that winner is somewhere. But people are standing ovation. To whom? Why this has happened? Because we never fail. Okay, so if this approach in our mind, in student, in teacher, I don't think that our student will fail. So I'll stop my presentation here. Thank you so much for your kind attention. If any questions or any doubt regarding anything regarding career and anything, you can ask me. So, especially students from MSc 1 to yes. Okay. So, yes, very good question. Thank you for asking these questions because I didn't explain that. So we can control the nano size. It is depending upon all the physicochemical parameters. Like that, you can vary the pH. So whatever the reaction you are carrying, at what pH you are carrying that reaction, you can vary it from pH 2 to 3, 4, 5, 6, like that. And you have to optimize that condition. So number of cells, how much cells you are using. So cell concentration is there to vary the, to control the size. Then salt concentration is also there. So all these parameters you have to optimize. It is not like that you are putting metals and you are doing and finish and you are coming. So it takes time. For anything you have to do trial, and then you'll get the result. Sometimes it is very frustrating. But even there is a frustration, even in my PhD, I never frustrated. What is the reason? Because I always thinking positively. Because Abdul Kalam said that problem comes in your life not to destroy you, but to analyze your strength. So tell difficulties that you are too difficult. Okay, that means I'm not give up very easily. Okay, so if my guide tell me that check this manuscript for 5 times, I was checking 10 times. Okay, so that kind of spirit we have to develop. Don't think that my guide is creating problem to me. Okay, if I am thinking in that way, I will never be in progress. Okay, so give me more trouble. If you are telling me 10 times, I will do 20 times. Okay, so that kind of spirit, if you develop, everything will be changed. Your attitude will be changed, your positivity will be changed, and you will never be frustrated in your PhD. Okay, your guide at the end will say that now get up and go. Okay, now it is a finish. So we should have that approach, and then it is a benefit in both way. You will develop. Okay, and once you have degree, so you know that there are many people are getting PhD degree, but now there is a difference between the quality PhD and the only PhD. Okay, only PhD will get within 2-3 years also, but with the good publications, then you will be recognized. You will get the recognition. Okay, so that is your merit. So try to publish in very good journal and try to develop good attitude towards your research guide or towards your laboratory or your colleagues. You know that your colleagues are very important because when I was writing my manuscript in laboratory at that time, many my colleagues were hiding their manuscript from me because they are thinking that he might be still some ideas and all that. It's not so easy. If you are working for 1 year, 2 year, and how can I still within 5 minutes? Okay, so for protein, metal binding protein purification, I took 2 years. Then how can the Jay Shankar sir can do in 15 days? It's not possible. Even I am showing my data, which is unpublished. It's not possible. Why I am saying this? Because it takes time. Optimization, everything, trial and error. For that you need expertise. So it is not so easy. So my colleagues were hiding because they are thinking that I will take idea. So what is the loss then? If you are hiding your research or manuscript from your colleague or from your guide or from anyone else who are interested, what will happen? You will never be understand your mistakes. So instead of that, what I was doing, I am telling my experience, what I was doing, when I was preparing my manuscript, I was preparing in front of all. Everyone was coming and asking me what you are doing. I was saying, I am writing manuscript. And after the print, once I prepare the manuscript, I know I have 10 colleagues. So I took all the 10 printouts and I gave to everyone. And I never tell them that, please help me for my manuscript. Instead of that, I said that find my mistakes. I use that word. Why purposely? Because people like to find mistakes and they enjoy. Yes, this is wrong. This sentence is not good. Grammar is wrong. This is wrong. I said good. And then I collect all the 10 remarks. And then at the end, I was sit and I was really checking. Very good comments, very good change in the manuscript. And that is really I got the benefit from them. And I said then, thank. So what was the result? They got 1 or 2 research paper during their PhD. I got 21 research paper in my PhD. Why? Because of the collaboration, because of the open. So when I went to Belgium, when I went to Belgium for the workshop, so I listened one thing there. Okay, what I learned from there, so they show one parachute picture in one presentation. And they said that more you are open, more you will fly. Okay, we should open to research. More you will fly because someone will give me much more idea. And Jay Shankar sir can give me, Prakash sir can give another idea. So like that, more you are open, more you will fly. More you will hide, more you will down. Okay, so that's why I never compete with anyone else, with my colleagues and with my friends also. I always say that research is not competition. But unfortunately, most of we are, if there are 2 colleagues, they are thinking that, oh, her paper will come first, my paper will not come like that. Instead of work together, join together, and it is a win-win to all, I think. So that is a benefit. So thank you for your question. So by using the different parameters, we can estimate or we can control the nano size. Anyone?
Yeah. Dr. Ashok has given a very good example, learning through the mistakes and how we can actually build up the research career more particularly. And throughout his presentation, he has started with the nanotechnology in microbes, giving little bit blend with some of the plants also, especially, and then making this nanowires, very another important area, and some of this nanoparticles with a antibacterial and antibiofilm activity. And shown his publication ensuring that the impact factor is also mentioned, which is of course very useful nowadays in all our rating as well as profile and several other applications. So from that angle, the speaker has really exhaustively covered several research activities. In fact, I know Dr. Ashok's PhD guide, Dr. Smita, from long time, and then he told me that nowadays she is quite busy, and then she is much more in higher positions. So this is what actually I think the strength of science makes the person in much more higher level. We hope to see even Dr. Ashok, I think in next 10 years, that he goes in a much better position. Yes. Even we exactly, we take this opportunity to even wish our organizing chairman also to go for a higher position. If participants can just clap for organizing chairman, I would be happy. In this lecture and other lecture, I saw actually clear difference. The participants were calm and quiet. So they were really actually measuring the size of nanoparticle, the functionality of nanoparticle, and what the other wonders nanoparticle does, I think. I'm sure that they will get benefited. I think I have to come that side and talk, so I will stop here. If any other people have any questions, I think we can just have. Any questions, please? Yes. No? So once again, thank you very much and congratulations for the nice lecture.
Thank you very much, sir, for your very informative and valuable session. I thank Dr. Prakash Halami, sir, for being the chairman of the session, and Dr. B.S. Srinath, sir, for being the rapporteur of the session. As a token of appreciation to Dr. Ashok V. Bankar, sir, I call upon Professor M. Jaishankar, sir, to present a memento to our lead speaker. The second lead talk will be delivered by Dr. Prakash M. Halami, sir. I request you to kindly be seated on the dais. I welcome Dr. Ashok V. Bankar, sir, to chair the session. And I welcome Dr. B.S. Gunashri, ma'am, to be the rapporteur. I call upon Ms. Hasina Sheikh to give an introduction about our lead speaker, Dr. Prakash M. Halami, sir.
Good morning, everyone present here. I am Hasina Sheikh, Research Scholar, Department of Microbiology. It's my immense pleasure to introduce today's lead speaker, Dr. Prakash Halami, sir, Chief Scientist, Professor ACSIR, and Head Microbiology and Fermentation Technology Department, as well as Head Department of Food Protectants and Infestation Control. Also a nodal officer, CFTRI COVID-19 Testing Center at the CSIR Central Food Technological Research Institute, Mysore. He has nearly 3 decades of R&D experience besides HRD activities. He has obtained his postgraduate degree in molecular biology and biotechnology from National Center for Plant Biotechnology, Pusa Institute, AIRI, New Delhi. Doctorate in biotechnology from the University of Mysore. Also obtained advanced training in biotechnology from Kobe University, Japan. Doctoral degree research at the University of Frankfurt, Germany, and postdoctoral research in USA at the University of Minnesota as a Raman Research Fellow. His laboratory investigates the mechanism of food-grade metabolite production by microorganisms using classical microbiology approaches and by using tools of genomics. His research on antibiotic resistance has been recognized by WHO India office by inducting him in preparation of National Action Plan on AMR, as well as by FAO for the evaluation of ICMR, ICAR projects on AMR. Sir has so far guided 75 postgraduate dissertation students and 11 doctoral students. Sir has 116 publications in peer-reviewed international journals, where 4 Indian patents have been granted.
Stop video.
He has presented 175 papers.
Start video lo போகும்போது நம்ம இது போயிடலாம்.
He has presented 175 papers at national and international conferences, winning 22 Best Paper Awards. He is an editor of journal Food Science and Technology and scientific panel member in FSSAI, New Delhi. Sir has been awarded 2 times CSIR CFTRI Foundation Day Award in the year 2013 and 2019 for scientific contribution, International Travel Grant Awards from 4 different agencies, ICMR New Delhi, Indian National Science Academy, SERB DST New Delhi, and also CSIR New Delhi. Fast Track Young Scientist Project of DST New Delhi, recipient of Lalji Dodo Samarth Nidhi Award of AFST India for the year 2013, CSIR DAAD Exchange of Academics for year 2009 and 2015. He has been elected as a Fellow of Indian Academy of Microbiological Science 2016, Fellow of Indian Association of Applied Microbiologists 2016, Fellow National Academy of Biological Sciences, Fellow Royal Society of Biology, Fellow Biotech Research Society India, Fellow National Environmental Science Academy, and awarded with Best Scientist Award of NABS 2018 and Senior Scientist Award by Microbiologist Society India. He is convener cum member Bureau of Indian Standards. Thank you.
இவங்க பேசுற கேட்காது இல்லையா?
ஆமா அதுல ஒரு ஆமா அப்படி ஒரு பிரச்சனை இருக்கு. அப்ப என்ன பண்ண இது ஒரு கோட் தான் இருக்கு. அப்போ இதுவுமே இதுலயுமே மைக் உண்டு.
ஹலோ
அப்படியா?
அது வைக்கணும்னு இல்லை.
சரி.
அப்போ வச்சோம்னா உங்களுக்கு கேட்கவும் செய்யும், நீங்க பேசவும் செய்யும். தேங்க்யூ.
ஒன்றும் கேட்கலையே.
இல்ல அவங்க ஆஃப்ல வச்சிருப்பாங்க, எல்லாரும் மியூட்ல வச்சிருப்பாங்க. அப்போ ஒண்ணுமே கேட்காதுல்ல. யாராவது ஒரு ஆள் இப்போ யாராவது ஒரு ஆளு நீங்க ரெஸ்பாண்ட் பண்ண சொல்லுங்க, எனக்கு நான் பேசுறது கேக்குதான்னு சொல்லி அங்க பேசுறேன். ஏதோ சாட்ல ஏதோ மெசேஜ் வந்துச்சு.
Your session will start by 12:15. அவங்ககிட்ட கேட்டு பார்க்கலாமா?
ஆமா, ஆனா ஆடிபிள்
Anyway, I have some 20 slides to
கேக்குது.
From yesterday, we are seeing the different aspects of microbiology more interestingly.
இது வேணுமா? இது வேண்டாம் இல்ல?
அவங்க இதை present பண்றாங்க.
This is one of the area which luckily known as actually the opportunity to express few of our research activities in this area. This is not the only topic which we work in our lab. Nevertheless, this is actually the important with reference to the institute mandate where the CFTRI asset is looking how the lifestyle diseases can be prevented, how the food additives can be improved with reference to the microbial process. And from that angle, if you see the CFTRI major research activities will be on only two. One is the prevention of post-harvest losses, and another one is the value addition in agricultural commodities. Now, as far as the microbiology and biotechnology is concerned, it actually touches both. With reference to the probiotics, we are working from last 10 to 12 years. And this was actually started with the ICMR project where we were looking for developing a certain sort of a detection tool where the probiotic bacteria can be identified from the environment or from the nature immediately brought into the practice, and commercialized. More importantly, though, we have several such probiotics, so-called probiotics, but then their health benefit has to be ascertained. And that's why actually most of the probiotics are the researchers which struggle to provide the sufficient health benefits of the probiotic in the human subject. Nevertheless, whole world is depend only on the two major probiotics, the Lactobacillus rhamnosus culture and Lactobacillus KSP serota in a Lactobacillus group. And you will see in the Bifidobacteria, the Bifidobacterium longum, animalis culture, now BB12 culture. Now, why other probiotics are unable to come for the commercialization is really a big question, not only for the researcher, for the industrialists, for the consumers, for the regulatory agencies, and several other people. So, once again, compliments to the organizer of this 2 days national conference on integrated microbial technology for sustainable future. In fact, this is the first time actually in this city center. And I guess a view of this dissertation students who are there from this department, in CFLI especially or in our microbiology department also. More particularly appreciating the efforts of microbiology society, because I am associated with microbiology society, and this year they are actually going to organize the international conference in Bali, Indonesia. So most of the actually microbiology people, they are now preparing the abstract, the tour plan, and then several other briefings. Dr. A.M. Deshmukh is the president, and this guy actually I got in touch to come for this conference. So once again, thanks for the activity. Just to tell in brief about our activities, the antibacterial peptides, mostly from the food grade microorganisms, and their role is what? Biopreservation. Okay. Lysine, most of the microbiology student knows how the lysine is being used for the food preservation, especially the canned foods and the dairy products, especially to take care of the controlling the Clostridium, because Clostridium otherwise make the spore difficult to kill or inactivate those spore even for the pasteurization or even for the autoclaving also. So only the lysine-like molecule works. That got. We are actually looking similar molecules, more importantly through the from the bacillus as well as from the lactobacillus. This is another very important microorganism which we study, is the Bifidobacteria. One of the examples I will be telling. So these are the Bifidobacteria. And this is actually another research activity pertaining to the antibiotic resistance. Antibiotic resistance actually is a very major problem, especially in India, even from the 2007 or 9. Everyone must be remembering about emergence of the super NDM1, the carbapenem resistance and enterobacteriaceae. We don't work on the pathogen for AMR, but ours is actually how in a lactic acid bacteria, the antibiotic resistance problem is. And that is actually because lactic acid bacteria known to be a natural reservoir of the antibiotic resistance or the antibiotic resistant genes. The second, whenever you consume the fermented food, you consume them without cooking, right? That means the live organism. So you just consider that you eat such a live organism in a high dosage, which has the antibiotic resistant genes. And naturally, they will be transmitting those genes to the pathogenic bacteria, to the more virulent bacteria. And that's why they can actually be a very good donor. I am only in the first slide. It's okay, now I can. So these days, as I told initially, this is one of the important aspects with reference to the human health. What is that? The gut microbiome. Everyone wants to live longer. wants to be healthy, wants to be free from any communicable diseases, right? And lately we understood, at least from last 10 to 15 years, that these are actually the role of a microorganism which are found in our gastrointestinal tract, which are associated with the diseases, and that's why Hippocrates rightly said all diseases start from the gut. So good gut means what? So good gut means it should have the less gut permeability, less toxemia, septicemia, less pro-inflammatory cytokines in your blood, more insulin sensitivity, because most of the diseases start with the inflammation. And those actually start with the insulin again. Then higher blood or metabolic or cardiovascular disease. These higher are associated with the good health of the human being. Yes. And that's why you will see the gut microbiota is associated with the several the diseases like the IBD and IBS, diseases. They are also working on allergy also, the metabolic syndrome, cardiovascular diseases, obesity and diabetes. So 70 or 80% diseases which human being suffers is actually are associated with gut microbiota influence. And this actually the balance bacteria, is responsible for this disease. And then why fluctuation of the balance actually gets disturbed because of the diet. Suppose you shift the diet from vegetarian to non-vegetarian, or the stress, the age, consumption of antibiotics, as well as the infection also. So whenever such infection happens, whether it is bacterial or viral, there is a possibility in the shift of the gut microbiota, and that can lead to the several diseases. So this is what actually even we have seen during in the COVID pandemic. Now coronavirus is infecting, but the post-COVID complications, the other metabolic syndromes which were actually arising, was the one of the major. So what is the opposite terminology for the euboiasis? So it is the dysbiosis. So what does the dysbiosis means? You will have the gut permeability increasing, okay? Or endotoxemia, the toxins in the bloodstream. There will be several of the actually age-old people will be suffering with this problem. The systemic inflammations, one or other person actually suffers with the inflammation, either it is a low-grade inflammation or the higher-grade inflammations. The insulin resistance associated with the diabetes, and then several other the diseases of the what I can say the lifestyle diseases also. So this you will see the euboiasis mostly we will see basically with reference to the balanced intestinal microbiota, and when this happens, when you have the high microbial diversity in your gastrointestinal tract. So the more or the you have a richer the microbial diversity, you're supposed to be a more healthy. So that is what the rule is. Then high production of short chain fatty acid. Why the short chain fatty acid? Because this is actually the energy source for the several of the epithelial cells in the gastrointestinal tract. Then healthy gut barrier and then healthy immune system. This actually often happens like when you consume either Mediterranean diet or the plant-based diet or the ketogenic diet. People understood this sequence of the mechanism, and then they actually now are changing their eating habits, changing the eating patterns, either consuming the ketogenic diet or the plant-based diet and the Mediterranean diet. And when you consume this diet, what happens? That in your gut, in a gastrointestinal tract, these are the molecules which will be produced. Okay. But when you eat actually, suppose the diet associated with rich in a high fat, so you will have the dysbiotic gut and then several other diseases. And then the reason for these diseases are, you can see from the disrupted intestinal till the pro-inflammatory cytokines. And these pro-inflammatory cytokines are the one actually are responsible for all the lifestyle diseases. What actually we see during the birth, each and every child actually born in a gastrointestinal tract is just survive. Gastrointestinal tract actually of a newborn baby is free from microorganisms. So from where this bacteria comes, Mother's milk is one of the first source, and that's why the children which are basically born by the normal delivery supposed to be the healthy. Why? Because the mother's milk, which has rich source of the very important sugar, N-acetyl glucosamine NAG, is responsible for a more proliferation of the bifidobacteria. And this bifidobacteria colonizes in the early stage, and they help the other useful bacteria. So when you see with the cesarean delivery, it is a situation something different. Okay. So what I see in the early age, the more variability is the gut microchlorides. And as we grow, more the diversity is there. And this stable diversity is maintained till the age of 30 to 40. Those who are very young, they need not to worry what is this gut microbiota and how it has to be maintained. Why? Because they have already a stable gut microbiota, provided they are being a normal delivered baby. If they are the cesarean baby, then possibility of a disturbed gut microbiota, allergy, or autism, or any neurological disorder, they may have to face. So this is what it is. Okay. And then this diversity actually changes or fluctuates after 40 or 50. Just 5 or 10 years difference here and there. But it doesn't mean that you should not have a healthy diet. You should not have a nutritious diet. What this nutritious diet is taking? This diet actually is taking care of keeping this proportion of microorganisms in a balanced condition. So in microbiology or in gut microbiota, number one is the diversity. Second is the proportion of a different microorganism. It so happens actually some of these pathogenic microorganisms are useful. Only their number should be less. So that is what actually the reason is. So people actually understood, from last 100 or 120 years that how this gut microbiota can be modulated or has the importance or significance with reference to the human health. Okay. And this is what actually Eli mentioned that he has actually observed the story of lactic acid bacteria or the fermented food started much before. Why we have to actually look for the fermented food? Because fermented food has mostly the friendly bacteria, bioactive molecules, and you will be consuming the bacteria which are helping or giving directly nourishment, nutrients to your health. Okay. So Eli mentioned that actually the observation with reference to the Bulgarian peasants or the farming community who had the exceptional lifestyle of more than 100 years. He has observed that the people, those who are consuming the fermented food or fermented dairy product, were able to live longer. The reason? Because they were otherwise consuming a lactic acid bacteria. They are the friendly bacteria. These bacteria, the first thing they do is they acidify the gastrointestinal tract. That means they are not allowing the other pathogenic bacteria to grow. Number two, they are suppressing the putrefying bacteria. Means they are not allowing the bacteria which are has the ability to make inflammatory markers, activate the inflammatory markers to change that. Number three, this bacteria has the ability to digest the food, absorb the food, and then several other things. Okay. So this is what actually the indication of the theory of longevity of life starts from here, that consuming a fermented food provides you a very beneficial microorganisms. Those bacteria are associated for the longevity of life because they reduces the inflammation, they reduces the toxigenic bacteria, they help to digest the food, they help to produce a short-chain fatty acid. And I usually tell cities actually they provide 5 to 10 health benefits. So to find out what those health benefits are. And after studying such product, this is actually the first commercialized products, the Yakult, which is available even now also in the Indian supermarket with a lactobacillus caseiculture, and then this is just a refreshment drink. You get this 25 ml bottle, the lactobacillus caseiculture, and very useful for the gastrointestinal health, what they cleanse, and then subsequently the probiotic yogurt, which is Activa, has beside lactic acid bacteria, the bifidobacteria incorporated in that, and why lactic acid bacteria and bifidobacteria has a distinct conformant, different health benefits that I will be telling in a few minutes from. Now, actually what happens whenever we consume a food with high fat, this is actually the major cause of the problem. It doesn't mean that the people fall ill or they will be sick if they eat the food with the high fat, not necessarily, but if they do not have the ability to digest, if they do not have the microorganism to metabolize the food, in such cases, so it should not reflect that don't start eating the high fat food from today onwards. You can. What happens actually when we eat the high fat food? High fat food actually helps the gram negative bacteria to grow more, number one. And once they grow more, they have actually ability to produce or make an enterotoxin. And this enterotoxin actually secretes, makes the leaky gut, makes this LPS, lipopolysaccharide, and is associated with the inflammation. And then this low or high grade of inflammation is responsible for several of the lifestyle diseases. Okay. And that's why we are making, in general, the less gram negative bacteria, more gram positive bacteria, and within the gram positive bacteria, more like a lactic acid bacteria, because as I told that lactic acid bacteria, they have inherent capacity to produce a lactic acid. That means they acidify your gastrointestinal tract, and this gastrointestinal tract, this is what actually the acidic pH is required to suppress the pathogenic bacteria. Okay. So lactic acid bacteria, in other words, are the probiotics, they act with the three different mechanisms. One is the antagonism, which I told either the antimicrobial substances they produces are the immunomodulations. Okay. And then another is the competitive exclusion. And then everyone knows, even during the COVID time, which we were actually looking for the more immunomodulators, the food which will have a immunomodulatory property, the plant-based extracts or even what the kadava also is very common, even spirulina, it was there in the news for a very long time. What it was doing, because it was actually helping to immunomodulate, modulate the immune system of the host to produce more anti-inflammatory cytokines, to suppress the pro-inflammatory cytokines. Probiotics also work in the similar mechanism, and that's why even few of our probiotics also we tested for this purpose. Now, as far as the commercially available probiotics, which I was telling initially, belongs to two lactobacillus, and one is the bifidobacteria, and then you can see these products are available, and I already told what is actually in general the problem associated with the other probiotics, Few years back, exactly right now I don't know, but in Wikipedia it was there. Some of the probiotics were blacklisted by IFSA and MD. Why? Because people commercialize them, just like pirated journals or the pirated manuscript. But they were unable to exhibit the health claims which they made, and that is what the reason was. Coming to the topic which I am going to tell in next 15-20 minutes, if participants can be seated till I finish the lecture, will be interesting for them also. Just like how Ashok sir was also explaining how the presentation can be useful for the student from that angle, I am going to tell how these experiments will perform. Okay. So why actually the preclinical is required? Because we have such several hundred probiotics, but to bring them in a practice or for the consumption, besides their in vitro probiotic characterization, it has to go for the preclinical. Once preclinical is done, health benefit is recorded in animal model, then to the human subject in a healthy individual. After the healthy individual, then it will go in the disease condition. Once it shows the health claim, then only that probiotics can be marketed or commercialized. This is what the FAO WHO guidelines are. This is what actually the ICMR DBT also has endorsed this guideline. So anyone who is working on the probiotics, they have to follow this 10 to 15 steps. Then only you have developed the probiotics. Otherwise, you are only the basic researcher or somewhere in the part of that. And that's why it took almost 10 years for us to come to the preclinical level. And how it happens, that's what I am going to tell you. Because we have to actually ensure the safety whether the consuming this live organism, is it a safe for the consumer or not? And since most of the lactic acid bacteria, being they are a food grade organisms, consumption is not easy. But sometimes the number can, because probiotics means it should be a sufficient number of 10 raised to power 7, 10 raised to power 9, 1 million or 10 millions or 1 billion. So you can make out that when you are consuming such a high biomass or high dose of the live bacteria, and whether they can cause any health hazard or what. And for that purpose, actually, safety is required. And in safety with reference to again, if it is upon the bacillus source, it is the more, okay? That I will come later. Then you can see how much actually we should consume. Yes, in a minimum therapeutic value is 10 million. Okay, but 10 million, is it the same dose for the everyone? Is it for the same for the children? It is the same amount for the adult also. For that purpose, the efficacy is required. So why actually we do the preclinical? A quick snapshot is here. Risk assessment and the prerequisite for the clinical. Because when we apply for the clinical trial with any medical hospital, they first ask it along with the application in a human ethical clearance, where is the result of the preclinical which is published. So that preclinical is a must in approach. Then we can actually get your probiotic tested, in an animal model or in a living model. Then more subjects can be evaluated and cheaper because you never knows. Directly you go to the human subject and if it calls the health results, then what you will do? So for that purpose, you check in the animal model, be sure that it is safe, that they are working properly, and then maintaining control conditions also possible. You can rear these animals wherever you want. And then more important, the reproducibility and repeatability can be conducted. So these are actually the several of the advantages, and that's why we conduct the preclinical. The first reason, as I told, it is a prerequisite for the clinical trial. Besides that, the several other steps. Now, how we actually? So we know that we actually evaluate that organism is in this state in a Indian case it is our FSSI. In a FSSI that microorganism is enlisted, then we consider it the graph status. That means safety evaluation is not required. Then we directly go for the efficacy only. Okay. And then this efficacy will be done in a disease condition to the biomarker. So what you do is once you have done this safety, then we go for we induce the disease, whether you want to do the probiotics for the diabetics or the cardiovascular or the high blood pressure or which are the lifestyle diseases for that purpose. And then most importantly, we have actually ensured that the probiotic works in general 80-90% as a prophylaxis, as a preventive measure. They don't work like a medicine to cure the disease, but then there are few probiotics which are identified even for the curing the diseases also. But most of the probiotics or the example which I will be explaining are associated with the preventing that disease. So you can see the slide, see our result, and then you can ask me, does your probiotic work for the disease or not? Okay. Yes. Then we ensure that once we induce the disease, so the disease subject, the group of animals, they should actually have the disease also, right? Once you induce the disease, and then histopathology and clinical biochemistry analysis, and then statistical analysis and then interpretation. So these are actually the very important steps. So by doing so, we look how our laboratory evaluated probiotics can be given or subjected for the preclinical trials, okay, in a animal model. So this is what actually I am going to tell. We actually work in a three group of different microorganisms, as initially I told. One is the Bifidobacteria, and as I told, that Bifidobacteria actually colonizes in the larger intestine because of their anaerobic growth requirement of alkaline pH, okay, and they will not be able to tolerate the acid and bile stress. So due to that reason, so this is our own organisms, and this are isolated and characterized in our laboratory. How preclinical we are going to do, I will be explaining that. Next one is the lactobacillus. And in lactobacillus, we have the two groups. One is the lactobacillus fermentum and another is the lactobacillus plantarum. Now, why this lactobacillus and then bifidobacteria they differ? Because lactobacillus group are lactic acid bacteria. They colonizes in the smaller intestine. Bifidobacteria, they colonizes in the larger intestine. The role of lactic acid bacteria digest the food, absorb the food and acidify the gastrointestinal tract. Bifidobacteria digest the food which is not digested in a smaller intestine. Dietary fibers, the polysaccharides, and produce the short chain fatty acids through the gut microbial homeostasis, produce several of the minerals and vitamins. Okay. So this is what actually the work is, and that's why we actually work in both the group of microorganisms, the lactobacillus also and the bifidobacteria. With reference to immunomodulation, both are important, but more predominantly it is actually the bifidobacteria. And only on the bifidobacteria, I can talk hours together in other words. Similarly, on this lactobacillus fermentum, even yesterday we had one of this presentation of lactobacillus fermentum, which I will again explain the preclinical trials. Okay. This is another interesting microorganism which we work with, the bacillus. Bacillus licheniformis. Bacillus coagulans, most of this MSc student must be knowing as sporolac powder, which is commercially available, you know. So we don't work on the Bacillus coagulans, we work on the Bacillus plexus and Bacillus licheniformis. And why this microorganism? The uniqueness, because the sporulating nature. One day we have to be actually careful with reference to the safety of this bacteria. Why? Because most of the Bacillus produces the toxins, and this toxins can be detrimental to your health. So I will be going with reference to the 5 preclinical studies in the next 10 minutes or 15 minutes. How much time is there? You people will be drinking tea. If I explain at least one preclinical trial correctly, then remaining 4 you can understand on your own. You people can be eating biscuits and drinking tea. I think this is not a probiotic one. Okay. So we actually isolated such several bacteria, a bacillus group, lactobacillus group, bifidobacterial group, and several others for exclusively probiotic property. And I told, with reference to actually the bifidobacteria, why we have to work? Because they are very much required for the gut microbial homeostasis, for inducing the immune response, for digesting the food. And actually, as I initially told, that they actually were the actual source of a bifidobacteria is the mother's milk. Afterward, there is no source. You eat pickle, you get lactic acid bacteria. You eat a curd, you get lactic acid bacteria. You eat any fermented dairy product, vegetable product, you get only lactic acid bacteria, not the bifidobacteria. If you want bifidobacteria, you have to ask, or you have to come to me only. Okay. So this is what the story is. We did this bifidobacter preclinical trial in a rheumatoid arthritis induced rat model, and as a requirement of the preclinical trial, this is actually the carrageenan induced arthritis. Okay, and then what actually happens when you induce the arthritis, you will be able to see. So this you can see the group with the Brevi and the Lagam and the bifidum, and this is actually the arthritis induced the negative control. And we set one the standard drug, amikoxam, in this group, and then one is the non-arthritic natural group. Okay. More importantly, first 15 days we gave the probiotic feeding to all these first 4 groups. That was actually was required, and except this last 2 groups. Okay. The probiotic feeding. And then after 15 days, we induce the arthritis. Okay. And then we check what is the effect of a probiotic, on a arthritic induced rat. That is what our game is. And compare the result with the arthritic, which is otherwise treated with the standard drug. Yes, piroxicam. Okay, I will just go one slide before. So what we are doing actually, we are inducing the arthritis in first 5 group. Okay. Now, first we are actually feeding the probiotics in a first 3 group, and one we have the negative control, and then one is the positive control. And after 15 days of the probiotic feeding, we induce the arthritis. Okay. And then we actually sacrifice the mice and then do the analysis. What is the analysis we do? We do the biophysiology and then bone, then blood biochemistry, and then cellular tissues for the RNA expression and disease with reference to the marker associated with the arthritic feature. And then more importantly, the fecal microbiota. And I will be actually summarizing the result with reference to only the fecal microbiota, because blood biochemistry or other histopathology will be difficult to cover in a shorter time, because I have to tell 5 such a preclinical trial study in 5 minutes itself. So big challenge is there. Yes. So we first check when we induce the arthritis, what was the bone physiology of the animal? So you can actually clearly see here this bone path physiology. This is the control, and then when you induce the arthritis, you see here the swollen part of this bone there. Okay. In case of the standard drug treated one, you will see very clearly this swelling is reduced. Right? Now what I expect when I give the arthritis induced mice fed with the probiotic, I see the result should be like a standard drug. Okay. So this is what my expectation is. So you can see here the B. brevis B. longum fed one. This swelling was reduced. What does it mean? That probiotic bifidobacteria are also doing the same work like the peroxicam. Is it clear? Very good. Now I will be telling with reference to only the what happens to the gut microbiota. So you can actually very clearly see here. With reference to only the bifidobacterial count, this is just a conventional microbiological plating. You will see on the 4th week, the bifidobacterial count was increased in a subject where arthritis was induced, but those rats were fed with the probiotics or the bifidobacterial. So you can see these first 2, 3 are with the arthritis induced, but fed with the probiotics. Okay, and only with the arthritis induced, you see the lower one. Lower, less bifidobacteria. Then peroxicam treated, low bifidobacteria, and then control also a low bifidobacteria. From there, where they will come. So with reference to lactobacillus also, the number was more. If you see the E. coli, coliform, staphylococcus, they were also reverse. They were actually more in a disease-induced one with a standard drug. So what is the difference between the probiotic treated one and the standard drug? Anyone can tell? Krishna will tell. You can see very clearly the coliform, in a probiotic treated one was more staphylococcus, where when you introduce the drug. So what is the difference? The difference is the pathogenic bacterial count is more in a disease condition plus in a disease condition where standard drug was used. What is the use of probiotics then? Probiotics actually help to allow the lactobacillus friendly bacteria to grow more, plus reduce the pathogenic count. Reduce the pathogenic count. So you will see the pathogenic count in the first 3 column here, was less. Why? Because the bifidobacteria suppress the pathogenic count. So this is what actually is important. So when you take the standard drug, it is curing the disease, but it is not actually helping the gut microbiota, gut microbial homeostasis. It is not helping for the development of a stable, useful gut microbiota. It is only helping to cure the disease timing. That is what actually the message from this work is. Next one is the anti-inflammatory effect of lactobacillus fermentum In a step 13, induced diabetic cell, very similar model. First one was the bifidobacterial, now I'm seeing lactobacillus fermentum. And this is actually the very important or useful terms in our laboratory. Even now, it is, and 5 PhD students are working on this culture. 3 from biochemistry, which also is working on this lactobacillus fermentum. 2760. Anyone comes to our laboratory and wants to do either MSc dissertation work or the PhD work, they have to work on this culture, so they should know what the wonder this culture is doing. You know, and this culture actually was isolated during that ICMR project on the probiotics. And we actually have done the clinical trial. We have given to the people, they consumed and they told they were very happy. Why they are very happy? Probiotic effect. Okay. So several such information. 2 of the students of Dr. Ram Prasad, they are working. Muthu Kumar student is working. He is now in Germany. Meena Kumari Krishna is working on this. Only one culture. 2760. We also bring, and this is again related to the Pune NCCS. Okay. NCC. Now we transfer this culture in the same deposit because we have developed the product. Using the bifidobacterium, we have developed the 2 products. One is the bifidocurd and another is bifidobacterium enriched soya curd. That's why I was telling that if you need a bifidobacterium, you have to come to me. Why? Because these are actually the part of a CFTRI technology. You consume more bifidobacterium, you are more healthy. You look more healthy. You are free from diseases. Why? I told that the background information with relation to the bifidobacterium. So this is about the lactobacillus, and then we actually tested in high fat diet and the diabetics, right? The very simple procedure. Okay. I will not explain much. You can just see the count of pathogenic bacteria like E. coli, Staphylococcus, Campylobacteria. This was actually we estimated, and then what we expect is that when you consume the probiotic bacteria, the count of this pathogenic bacteria should be less. Okay. So you can very clearly see. You see here the diabetic control, the normal control, then diabetic control, and then these are the diabetics too, but with the probiotic. So what probiotic magic they did, you can see the total aero count was decreased. E. coli count was decreased. Why the E. coli count was decreased? Because lactic acid bacteria, this probiotic bacteria, they did not allow the E. coli to proliferate. So you can see very clear results. Why in the diabetic control E. coli count is more? It is a first DS microorganism. It is actually diabetics condition, more sugar. It is actually helping the bacteria to grow more. And that's why actually once we fall in, diabetics go on increasing, or what we can say, goes in the subsequent stages. Difficult to control. And why it is difficult to control? Because such harmful microorganisms, they will be actually go on increasing in your gastrointestinal tract. So this actually happens not only in E. coli, but Staphylococcus also you can see very clearly. Then Campylobacter. All these pathogenic bacteria are more in a diabetic patient, here in a rat. And when you treat these diabetic patients with the standard drug also, you know, it will actually not work with reference to gut microbiota. It will only help to cure the disease, not with reference to gut microbiota. But when you use the probiotics, what happens? You can very clearly see, you know, these 2 bars in the 4th week, the more count of lactic acid bacteria. Okay. We checked this phenomena again in a high fat diet also, for the lactic acid bacteria, and then you can see the similar results for the diabetic control. And we actually checked again whether this bacteria can translocate in some of the important tissues like a mat or liver or the muscle. And then we saw that there was no such translocation happening. We checked for the anti-inflammatory cell culture in the high fat diet, and then you can actually again very clearly see the E. coli count in a high fat diet supplemented with the probiotics, you know, less E. coli. Okay. So, but more lactic acid bacteria. So in other words, whether you consume the high fat diet or you have a diabetic, if you take the probiotic, you can manage the diabetes. So that is what actually I'm trying to present. The third story I'm telling is about the safety of this probiotics. As I told that we will be consuming in a hydrogel, so this is actually important. with reference to the safety of these cultures. And we observed, we did one in the single dose studies of 14 days and another one was the 90 days, the subchronic studies. And then you can see actually our experimental plan here in a high fat diet. And we exclusively look this gut microbiota modulation. In a first two example, the gut microbiota modulation was conventional microbiological plating. Now here we are actually going to use the real-time PCRs. Okay. And we check the, so what we did is, we had those such experimental samples with the animals. We took their excreta, feces, isolated the DNA and did the real-time PCR. A primer specific for the bifidobacteria, lactobacillus and E. coli. So what we see is where the probiotics was fed, more lactobacillus and bifidobacteria. Where probiotic was not fed to the animal which were eating high fat diet, more E. coli, less lactobacillus and bifidobacteria. So you can test now here. So this is how the result is. You will see the lactobacillus count is increasing with the both the probiotic bacteria, similarly with the bifidobacterial count. You can see in the reverse way what happens with the E. coli. You see the E. coli count is increased in a control one, you know, and E. coli count you can see here the very easily with the probiotics. The count is much less compared to the count of the lactobacilli and then bifidobacter. So what does this suggest? That the E. coli, the suppressing the E. coli count is being taken care by both this probiotic bacteria, the lactobacillus and bifidobacteria, and then supplementing the lactobacillus. Not only you see the more growth of the lactobacillus, but even the bifidobacteria also. And that's why one of the health benefit of a probiotic is that they stimulate the growth of other friendly bacteria. I hope now it is quite clear. When we present this work in a little bit high standard audience, they say, did you see the next generation probiotics? Did you see the Prevetola? Did you see the bacteria associated with the specific for the diabetes? That type of questions are being asked, you know. So we check only the limited group of microorganisms more by the conventional tool. Here we use this PCR sequences which are genus specific, and we could actually enumerate that lactobacillus feeding to the animal help to make more lactobacilla along with the bifidobacteria and less E. coli count. Okay. So this is actually the significant finding with reference to that study which we have done. The fourth one is here, and here actually the first. Till now I explained about one is the bifidobacteria, another two was the lactobacillus. Now I talked about the bacillus, and I told that bacillus being a sporulating or spore forming bacteria also has the health benefit, but since some of these bacillus have the toxin producers, their safety has to be evaluated. Here we actually checked with reference to the strain specific quantification. Okay, and again you can see here the toxicity studies, and then for the single dose, and then the subchronic studies, and then two bacillus cultures, the bacillus flexus and bacillus licheniformis. Yesterday also we had this presentation with reference to the bacillus licheniformis MCC 2514. Okay. So this culture actually, besides its probiotic properties, produces very strong antibodies, subtly, the bacillus licheniformis. For this pathogenic for the 13 weeks, and then besides the transportation of bacteria, so you can actually see the bacillus count was increased in the feces of the fed animal. Okay, and more importantly, even the lactic acid bacteria, they were reduced, but some of this anaerobic microorganism we were actually unable to enumerate. The very simple reason is because the ones the feces gets exposed to the oxygen, out. There is a possibility of reducing their viability. So that is the need. So this is actually the result of study where you can most of this probiotic bacteria was increased in the feces, suggesting that probiotic feeding has direct health benefits to the animal. So this is actually, as I said, telling from the beginning, is a preclinical trial. When we do in the clinical trial, we expect the similar results. Now this is the first four. It was actually in the rat model. Now I tell one example in a mice, and the next one I will tell with reference to analyzing the gut microbiota by the next generation sequencing. Just 2 or 3 minutes, I will not make you people to wait much. It is only at 12:10 on this. I have 5 minutes as well the official time given, and then 5 more minutes the extra. Okay. So we actually take again the same concept, LCC 2760 in a mice model, and the mice model has one advantage with reference to the preclinical. Okay. Much more actually suitable for the this anti-hyperfluorescent activity. We take this study in a diet and then provide it given. And then this was actually analyzed by the next generation sequencing. So what happens actually, why next generation sequencing actually is required now, whether microbiology, biochemistry or biotechnology student, this is actually the new and growing field again and requires to know why, because when we take the anaerobic microorganisms, most of them we will not be able to cultivate or they will not be viable. So to analyze or to know exact the fingerprint, how many such microflora is there, next generation sequencing or NGS is the solution. Plus, we are not aware the most of the gut microbiota, the culturable microorganisms. So we do not have the media to grow them. Due to that reason, just to get the fingerprint, how the complexity of a microbial world is or the microbial diversity, next generation sequencing is required. So how it is done is again the stool sample immediately just been subjected for isolated DNA, then next generation sequencing, and then large amount of actually bioinformatics. Lot of actually opportunities nowadays in the field of bioinformatics because large amount of data actually is being generated and people are not, those who are from the computer science, they will not know the biology or the biology students or biology struggle with the computation field, in other words. So we make such grouping and identify those several hundred thousand microorganisms in the pool. And then we analyze. So what did we do is, we check for the total aerobes in a high-fat mice, Staphylococcus, Campylobacter, and total lactic acid bacteria. And then you can actually very clearly see that the Staphylococcus, when you eat a high-fat diet, here it is the mice. When mice ate with a high-fat diet, Staphylococcus count was low. The fastidious organisms, to digest that high-fat, this fastidious organism is required. Similarly, with the Campylobacter. Nevertheless, high-fat fed mice was given a probiotics, lactic acid bacteria. And as I told from the beginning, why we need more lactic acid bacteria? Because they are the one which makes us happy, keeps us healthy, and will make us to give, this kind of information. And then you can see in case of when you feed the probiotics, even Campylobacter count is less, as well as the Staphylococcus, where in the high-fat diet, the mice fed with the high-fat diet. This also clearly suggests the importance of this. Then we actually said with reference to abundance of this phylum, and then you can actually see that we basically see this alpha diversity. What are the major phyla are associated or what is their proportion? So this is actually with the normal diet with high fat, with the probiotics, and then high fat diet supplemented with probiotics. So you can actually see some of this useful microflora, the phylum of this bacteria are in a higher proportion, and then those who are actually are more with the high fat diet are actually reduced when you give the probiotics. And this kind of actually evidence is most of the journals are expecting this in a much more broader sense. So that information is here. After that, we check what are the top 10 genre distribution of at the genus level. And then again, on the similar line, you can see, the high fat diet and then high fat diet with the probiotics, this one, the most of the fastidious microorganisms, which are otherwise in abundance in a high-fat diet, they were actually reduced. So this actually clearly suggests that the probiotics, not only they proliferate on their own, but they are actually minimizing the proliferation of the fastidious organisms as well as the gram-negative bacteria, which are otherwise are associated with several other lifestyle diseases. So this is what actually the information actually comes from this NGS sequencing. What are the different genera of the phyla are getting distributed? What is their proportion and whether useful or not? For a very layman actually to tell the interpretation of the NGS is here. Okay, we have the high-fat diet. And their high-fat diet with the probiotics. In case of the firmicutes, okay, you will see that firmicutes, it is reduced, okay, but in bacterioid it is higher. So this is not enough. Actually, it is a firmicutes and bacterioid ratio, and that ratio is important. You can see here with the high-fat diet, the firmicutes to bacterioid ratio is high, but with the probiotics, you will see that firmicutes to bacterioid ratio is less, and that is actually considered as a desirable, has a health benefit, has a implication, or the utility of the probiotics. What is the utility of a probiotic should be that when you consume the probiotics, firmicutes to bacterioid ratio should be less compared to the without consuming the probiotics. So if you get such a clear result, that means your probiotics are giving really the health benefits, and this is what actually the information is required. I told that these are actually our animal model preclinical trial. One of this culture, we did the clinical trial also. I think I have the next slide for that one. Yes. This lost the animation, but this is actually one of this product which we checked for the clinical trial, and we are actually looking for analyzing this NGS data and see that when we give to a group of people our probiotic lactobacillus fermentum, whether it was really useful or not. We are waiting for that result. And that's why, as you also told, more hardworking students are needed. Yesterday's speaker also told more hardworking students are needed. When the students are there, then only it is possible to make those kind of breakthrough research and give more findings. Now, from the beginning, I explained about several of this conclusion, which is definitely not new, and you can actually means, in other words, it is a self-explanatory, okay, with reference to this native probiotic bacteria, the disease condition, then gut microbial homeostasis, safety and efficacy of this probiotics, okay, and then the clinical trial which is required, which I told otherwise also to bring this culture for the consumption to the consumer. This is very much required. So with this, once again, I thank all the audience for their patience hearing. They were actually chit-chatting little bit, but time was short to interact more perhaps. I hope this lecture will be useful for you people, those who work in this area. So if you have any questions, I will be actually happy to answer. Otherwise, you can always write to me, and when you visit CFTRI or Mysore, please visit our laboratory so that you can see some of this probiotic product directly for example. Thank you very much. So in case of rat model, we have checked this study, and then we could see very clearly that the probiotic increases the insulin sensitivity. Then probiotics, we made with this lactobacillus fermentum culture, very specific difference with the diabetic induced, the streptozotocin induced diabetic patient. More importantly, we give this lactobacillus and fermentum to the patient with the periodontitis who are invariably of high risk of the diabetes. And they actually had a reduced dosage of the insulin. This work actually we did in JSS Medical College. We sent that paper. one of this MDS student who did this project work using this lactobacillus and fermentum, our culture. She made some sort of chewing gums, and that she gave to the patient. One side was the standard drug and another side was for this one. Though it was for the oral health, but the basis was because most of this periodontitis patients will be invariably a high risk of the diabetes also. And then periodontitis problem actually will be influencing more. Why? Because lot of sugar will be there in the saliva. So due to that reason, consuming a probiotics not only actually was taking care of a good oral health, but it was actually minimizing their requirement of the insulin. That kind of studies we have carried out. When we come to the full-fledged studies, actually we are waiting to tell you frankly for the some fundings to take up the clinical trial. And then there are actually several studies from the probiotic area, which culture, which particular strain, how it is effective, what is the dosage. Each probiotic we have to actually take minimum 15 to 20 days. Then only you can see the difference. That's why I was showing you the first week less difference, second week little bit better, third or fourth week it was to some extent better. Our clinical trial study was for the 20 days, and it was, we could actually see with reference to the gut microbial homeostasis, which we are waiting for those results by the NGS, but their body mass index and clinical blood biochemistry, those results are actually there in our hand, and they are actually quite promising results. Any other? You can use that. Yes. Yes. Not pyrethrum. Peroxicam we are using. Peroxicam. Peroxicam, that is the standard drug used to reduce the pro-inflammatory cytokines. And then bifidobacteria also is doing the same work of reducing the pro-inflammatory cytokines. But this is actually making other arthritis related markers to reduce. That the real-time results I didn't show. In addition to that, the gut microbial homeostasis. You have very clearly the Staphylococcus, Campylobacter, E. coli count will not be changing, or will be less on, will be more only in case of the standard drugs. But when you use the probiotics, disease is cured one side, and another side is pathogenic bacterial count also is a decrease. That is what actually the two things there. Exactly. Any other?
Sir.
Okay, then thank you very much.
Sir, excuse me, sir. I have a doubt. Sir. Excuse me, sir.
Thank you, Dr. Prakash Alani, sir.
So I think I'm not audible.
I'll talk on the topic health benefits of probiotics, preclinical trials. Sir also gave the importance of this intestinal tract health, and health is one of the important thing right now we understood from COVID-19, and how our health is important. And so all diseases, as sir told, that all diseases will be start from mostly from our gut. So our gut should be healthy, and for this healthy gut, we need many people, they are changing the diets, they are concentrating on diets. But sir told that diet is important, but with the diet, another important thing is that our gut flora, and that should be we need to concentrate on that. That's why he told the importance of probiotics, and he also told that how high fat diet is important, and how it is beneficial for the bacteria which are healthy bacteria. So many people, they are avoiding the high fat food because they have some misunderstanding about this high-fat food. But if your gut flora is good, sir told that it is beneficial because it helps to grow the gram negative bacteria. So, sir also explain about the risk assessment. They have trial these probiotics on mice, and sir found very good results. Although the results are primary, but these are the outstanding guideline for the further investigation. And I hope that sir will develop so many health products in future also. So it will reduce the risk of diabetes, because all of you know that diabetes is one of the hub in the world, and in 2000, I think 26, software developed that they predicted that so maximum population of India will be diabetic, and more than 50 to 60% population will be diabetic, and that is the reason I think this is a good innovation. This is a good source to fight with the diabetes. So I wish all the best to Prakash sir and his team, and best luck for the further research. And thank you once again for a nice talk.
Prakash sir, I have a doubt. Excuse me, sir.
Thank you, sir, for an informative session. I thank Dr. Ashok V. Bankar, sir, for being the chairman of the session, and I thank Dr. B.S. Gunashri, ma'am, for being the rapporteur of the session. As a token of appreciation for Dr. Prakash M. Halami, sir, I call upon Dr. I.K. Manchula, ma'am, to present a memento to our lead speaker. The third lead talk of the day will be delivered by Dr. Anirban Chakraborty, sir, who will join us online. I call upon Ms. MP Rakshita, ma'am, to introduce our lead speaker, Dr. Anirban Chakraborty, sir.
A pleasant afternoon to one and all present here. It's a moment of great privilege for me to introduce today's lead speaker, Anirudh Chakraborty. Sir is a professor and director of NITI University Center for Science Education and Research, Delakatte, Mangalore. He is also Dean, Faculty of Biological Science, NITI. Sir has obtained his Master in Microbiology in 1999. He pursued his PhD in Agriculture Science in 2006 at Kagoshima University, Japan. He is the founder of Consortium of Rare Genetics and Bone Marrow Disorder and the Cancer Gene Database at NITI. Sir was an associate professor and coordinator from 2014 to 2017, deputy director and professor from 2017 to 2020, and from 2020 to till date, sir is serving as a director, NITI University Center for Science, Education and Research, NITI University, Mangalore. He has received Best Research of NITI in 2018 and Best Researcher of NUC SER in 2018 and 2020. Early Career Research Award by Science and Engineering Board, Department of Science and Technology, Government of India in 2016, and Postdoctoral Fellowship for the Foreign Researchers by the Japanese Society for Promotion of Science from 2006 to 2008. Sir has total 9 external research grants handled, which are ICMR, DST SERB, DBT, DEST, and DST JSPS where 6 are completed and 3 are ongoing. Sir has total 65 publications and 176.57 cumulative impact factor. Sir has received 3 patents until this day. So we hope your talk will enlighten and guide everyone present here. Thank you.
I request Dr. Anirban Chakraborty sir to deliver a talk on the topic Oncomicrobiomics, New Insights into Cancer Diagnosis, Therapy and Prevention.
Hello. Am I audible? Hello. I need someone to make me a host. Otherwise, I can't share my screen.
Good afternoon, sir.
Good afternoon. Are you able to see my screen?
Sir Chakraborty, sir.
Are you able to see my screen?
Yes, sir.
And can someone please confirm if the slides are moving?
Yeah, yes, sir. Yes, sir.
Are the slides moving?
No, no.
Hold on. Backside light on. Yeah. Is the slide moving now?
Yes, sir. Yes, sir.
Okay, thank you, everyone. First of all, thanks to Professor Jayashankar for inviting me to be a part of the conference. I should have been there personally, but due to certain developments here at the university, I could not make it. I look forward to visiting your campus sometime soon. Today, my topic is on oncomicrobiomics. So basically, since we have a large number of student audience, I have divided my talk into 2 parts. The first part will be about introduction to microbiomics and a little bit more on how it is linked to cancer management. And the second part is about 2 projects that we are having right now, and which are linked to oncomicrobiome, and I will explain the results that we have obtained. So I have been given 30 minutes to speak, so I will try to cover within that time. So first part is about oncomicrobiomics, but before we go to oncomicrobiomics, let's look at omics as a study, as a field of study. Of course, we all know there are different kinds of omic-based studies that can be done. We have proteomics, transcriptomics, genomics, metabolomics, regulomics, phosphoproteomics, epigenomics, and so many different types. Now, if we have to define omics, basically it's a relationship and the actions of different molecules. It could be DNA, RNA, protein, which are involved in making up the cells of an organism. And when you combine this information from these different molecules, we bring them together and we call it a systems biology. Now, this system biology is a very recent and a very widely studied area of research. And when you connect all this information, what we get is basically integrated omics. So from the genotype to phenotype in a diseased condition, there is a crosstalk between these genomics, proteomics, and metabolomics. All of these come together, what you call as multi-omics. And there's a lot of involvement of bioinformatics, machine learning, deep learning, and all these new areas where there is no difference between the areas of expertise. You have biologists working with a computational biologist. You have bioinformaticians who are looking at these different information. So what do we do with this information? Basically, there are three things that can be done. One is to predict a disease condition and see whether there is a difference in between a healthy and a diseased situation. And if we do find a difference, then we try to understand whether certain biomarkers can be developed. And if these biomarkers are really good, then the expression of these biomarkers would probably lead to certain phenotypes that we see in this disease condition. So basically, in these various omics that we talk about, the most recent, perhaps the most recent and the most exciting field is microbiomics. So microbiome, as we all know, I think the previous speaker did speak about eubiosis and dysbiosis. He spoke about the probiotics from a diseased context, and he looked at, he also explained the clinical studies. Now, microbiome basically is looking at the community of microorganisms, and in human beings, we look at essentially 4 different types of microorganisms. It could be archaea, bacteria, fungi, and viruses. And they could be present in various situations, various places in the body. It could be in the mouth, in the skin, in the digestive system, and in the urogenital system. Among these 4, I think the gut microbiome or the digestive microbiome is very widely studied and is one of the most exciting fields at the moment in biological or biomedical research. So what this microbiome does is basically, it gives you an idea about the abundance and the fluctuation that happens. And these are due to various exogenous and endogenous factors. Genetic predisposition is something that is all aware of. And we have a lot of studies now where people have looked at the link between microbiome and genetic diseases. Now, in the last decade, over the last 10 to 15 years, I guess there have been groundbreaking advances, and I will try to explain a few of these in my future slides. Microbiome has been shown to play a significant role in the disease development. And these dysbiotic microbial signatures have been identified, and they are now associated with various pathological conditions. Not only these infectious and non-infectious conditions, but also in cancer. Now, the microbiome, these are from some statistics for some of you to know. I'm sure most of the people, those who are working on this are aware of this, but since it is an explanation or a presentation to the student audience, I wanted to show this slide. It's a very nice slide that is available. You can search it. So what the first one, these are various numbers. So these are, the first one says the number of symbiotic microbial cells that are present in human gut. The second is, although we are supposed to have 100 trillion microbes, only less than 10,000 have been identified so far. So there is a great amount that needs to be discovered or identified. This is the statistics that people have identified. If we look at the human cells, we have 10 times more microbial number compared to the human cells in a human being. And if we do that, obviously, if you compare the microbial genes and human genes, there is a ratio of 100:1. So you have, for one human gene, there are probably about 100 microbial genes. And if you look at the estimate that has been made on the number of non-redundant genes that are present in human gut, which are derived from microbes. There is about 3.3 million such genes available, and they are non-redundant. They are not merely copies of certain genes which are already present. Compared to that, we have only 22,000 protein coding genes in human genome, 25,000 probably. But compared to this, you see the number that we have, about 3.3 million. The last one is the difference between two individual humans. If we take two people, they will be different in terms of the microme, maybe 90% different in terms of the microme. But if you look at the genome sequence of the human beings, which is shown on the left-hand side of the slide, we are similar to each other by 99.9% if we consider our host genome. But if we consider the genome or the microbial genes, we are different by 90%. So that means the microbes are really very important, and they play a big role in the way we look at diseases. Now, the common approach to study microbiome is basically there are two ways. One is the 16S rDNA sequencing. 16S rDNA codes for the 16S ribosomal RNA, and which is highly conserved, and any difference in that would indicate that they have different operational taxonomic unit. NGS, or next generation sequencing, is the most commonly used method, but people do look at culturable microbes, because the problem with the microbiome is that only 1 or 2% of the entire microbiome is culturable. Rest of it is non-culturable. But they are present, their gene sequence can be identified. The other method is by shotgun sequencing, metagenomics, where the entire sequence is taken and it is analyzed. And the analysis part is more or less similar. In the first case, it is we go by the 16S rDNA, we identify them, we group them into different OTUs, and then identify based on the sequence information available on the website, and then we identify which organisms are present. And then once we identify, we look at the variations in them and see whether they are having certain signatures. On the other hand, the shotgun approach, we sequence the entire community DNA, whatever is present, including the host DNA. Of course, there are now techniques to get rid of the host DNA. And then we do the same analysis. We identify with certain databases. We look at the relative abundance. We look at the diversity, so on and so forth. Now, these are some of the examples. This is from a science repository database. If you look here, these are some of the diseases where the links have been established between microbiome imbalance and the onset or progression of disease. So the list of the diseases are mentioned here, and the kind of microbiome that people have studied are oral microbiome, gut microbiome, skin microbiome, vaginal microbiome, placenta microbiome. And these are some of the conditions such as infectious diseases, IBD, which is a very well studied disease as far as microbiome and human disease is concerned. Liver diseases, GI malignancies, colorectal cancer is something that has been very extensively studied, and there are now a lot of biomarkers which have been proposed. Metabolic disorders, including diabetes and other conditions, autoimmune conditions, psychiatric diseases, even schizophrenia and so on, allergy, and finally autism. So there are many such diseases that have been identified, and their links are pretty well defined now. Now let's go to oncomicrobiomics. But before we do that, let's see the cancer hallmarks. I don't think I need to speak about cancer, because I'm sure everyone is aware how dreadful a cancer as a disease is, but it's more than 150 years that cancer has been identified, but still we have not been able to conquer the disease. So the cancer is defined by an unregulated cell growth in the body, and there are several factors or pathways that contribute to that unregulated cell growth. So these are identified as hallmarks in cancer. So in the timeline when the cancer hallmarks were defined, the first definition came in the year 2000, although cancer has been the focus of study for several years. But Weinberg and Hanan, they proposed 6 or 8 core hallmarks of cancer, which were identified based on the research that were done over the last 2 centuries. So these are the 8 main hallmarks: proliferative signaling, evasion of growth suppression, getting an immortal status by having an unlimited replicating potential, invasion, metastasis, angiogenesis, loss of senescence, instability, and evading cell death. And then in the next 10 years, people started looking at cancer from different perspectives. And then in 2011, the same group came up with a revised hallmark. And they introduced 3 more hallmarks that they called as emerging hallmarks. And what are those 3? Deregulating cellular metabolism, avoiding immune destruction, and tumor promoting inflammation. So now people identified that inflammation or chronic inflammation is cancer promoting, and also cancers can avoid immune destruction. And this is very well taken care of in immunotherapy. Now, immunotherapeutic drugs actually harness the immune system or reject the immune system to fight the cancer cells. So these are introduced in 2011. And then 2022, this year, very recently, these 2 gentlemen looked at the hallmarks one more time, and they introduced 3 more hallmarks. One is an emerging and 2 enabling. When you say enabling, if these changes happen, then they can enable carcinogenesis. And these are the new hallmarks, that is unlocking phenotypic plasticity, epigenetic reprogramming, and the last one, polymorphic microbiome. So now it is well understood that microbiome does play a significant role in carcinogenesis. And then came this concept of looking at these cancer hallmarks through microbiome, particularly the intestine microbiota or gut microbiota. Now, this slide shows you certain bacteria that have been identified and how they have been identified to play a role in modulating some of these hallmarks that we have just seen. So that means now people are looking at cancer hallmarks through this microbiota, and that led to the concept of microbiomics or microbiome and cancer. Now, if you look at this slide, you see that it is the data that, or the information that is being shown here is very recent, only 2011. So you're looking at something that has developed in the last 10 years. So in fact, microbiomics or oncomicrobiomics is very recent, very, very new and very emerging in terms of the developments. Now, the first incidence of the role of microbes came in 2011, where they found that people or patients with colorectal cancer have a different microbiota, gut microbiota, compared to healthy individuals. And then in 2013, people actually proved that if a gut microbiota is transplanted into a germ-free mice, and if that mice is given a microbiota from a cancer patient, that microbiota can actually develop cancer in the mice. So that means the gut microbiota obtained from a cancer patient can actually drive tumorigenesis. And in 2014, they predicted that if metagenomics or microbial sequence analysis is done from the stool sample, one can predict the presence of specific microorganism in colorectal cancer patients, which is almost as accurate as a blood test that is being done. And in 2016, they looked not only at the gut microbiota, but they also looked at the oral microbiota, and they found that oral bacteria, or the oral microbiota, can increase the risk of pancreatic and esophageal cancer. And in 2017, when the concept of immunotherapy came in, people found that there were immunotherapy, by the way, is the most effective and the most precise anti-cancer therapy known so far. It is extremely expensive because it involves antibodies and it involves the immune system. But people realized that there were certain responders and non-responders. Although the people were having the same cancer type, but then there were some people who were responding to immunotherapy and some were not. And in 2017, they identified that actually it is the gut microbiota of the patient that decided whether they will respond to immunotherapy or not. And that they found in melanoma patients or skin cancer patients. And in 2018, they found that gut microbiome plays a causal link. So that means it is not a coincidental link, it is a causal link that can allow the body to fight the cancer through the immune system. So if we can modulate the gut microbiome of a cancer patient, there is a good chance that the cancer patient can fight the cancer in them. So oncomicrobiome is basically study of the community of microorganisms in the context of cancer. And how it has been done is basically you compare the tumor microenvironment. So basically the microbiome of the tumor is done and the microbiome of the gut is done. So people compare that microbiome, these two microbiome, and see whether there are similarities or there are certain changes which are present. In this slide, there are certain points that are marked in blue. There is something that are marked in green. The ones in blue are the predisposing factors that can alter the microbiota. The ones that are marked in green are the ways by which we can modulate the microbiota if we need to do that. And then the lower part of the slide tells you how these altered microbiota can interact with the immune system. And when you talk about the immune system, we are basically talking about the myeloid cells, the lymphocytes, T cells, and the B cells. And these are the ones that actually are involved in eliminating the cancer cells from the body. And once this interaction is identified, they can be utilized for three different aspects. One is for diagnosis, the second one is for prognosis, and the third one is for therapy. Now, in diagnosis, cancer-specific microbiome or cancer-specific DNA in the peripheral blood has been identified. So that can work as a diagnostic criteria. The gut and the intratumoral microbiota, the comparison of the gut and the intratumoral microbiota can decide whether a patient will respond to a particular drug or not. These are all based on the research that has been done. This data that is shown here is from a paper that got published in Science in 2020. So you can see how recent these informations are. And if some of you are interested, you can look at this review paper. It is always better to look at the review paper, which are in reputed journals. So the prognosis part can also be taken care of if we look at the microbiota. And the third one is by modulating the microbiota, one can actually do a change. One can actually change by the way the body is fighting the cancer. And towards that, the fecal microbiota transplantation, or FMT that we call about, is something that has been done in many patients with colorectal cancer and melanoma cancer. Now, interventions, how we can modulate. One is that just now I mentioned fecal microbiota transplantation. So basically, it is engulfment or transfer of a microbiota from a healthy donor to a recipient. The recipient would be somebody with a cancer symptom. The second one is introducing probiotics. I think this previous speaker spoke about it, the importance of probiotic bacteria. I don't need to explain that again. Then the third thing is prebiotics. Now, prebiotics are certain food ingredients that can stimulate the growth and activity of probiotics. So these are basically, say for example, high fiber diet could be considered as a prebiotic. So these are some of the steps by which one can modulate the microbiota, and these interventions have been proved to be successful in managing cancer in humans. Now, since it's a microbiology conference, I need to mention what are these microorganisms that have been identified to be having anti-cancer property. The slide shows how they can perform their anti-cancer properties. I think we'll not go into the details of that, but you can see that there are two parts here. One I have mentioned at the top is what has been identified in humans, and the lower one is what has been identified in model systems, in experimental models. So in human, you can see Streptococcus pyogenes, Serratia, Marcescens, Mycobacterium, Lactobacillus, Listeria, Salmonella typhimurium. These are supposed to be known pathogens, but these are attenuated forms. We also have a project right now supported by ICMR, Government of India, where we are looking at non-typhoidal Salmonella as an anti-cancer agent. So we are looking at the ability of bacteria to reduce the cancer growth by infecting a model organism, a zebrafish that I work with. And this project has started last year, and we are looking at non-typhoidal Salmonella. These are auxotrophic mutants that we have developed. Similarly, in mice and other models, they have found Bifidobacterium, Enterococcus, Lactobacillus, and Clostridium as bacteria which have anti-cancer properties. Pathogen-associated molecular profiles, which can be utilized or which has been identified to affect the immune system. And the third one is metabolites from bacteria. There are gallic acid is a very commonly studied one, which can affect the host metabolism. Now, the gut microbiome and cancer immunotherapy, as I mentioned in the beginning, this has been something which has been quite popular these days, and a lot of people are working on this, and they're looking at how the cancer immunotherapy can be made better, or how the response to immunotherapy can be made better in patients by altering the gut microbiota. So the crosstalk here between is cancer, the gut microbiota, and the immune system. And the main checkpoint, the main immunotherapeutic drug that is used is the checkpoint inhibitors, which are in the market. So there are, in immunotherapy, there are 5 different types. The first one is the immune checkpoint inhibitors, which are more common, but people also use CAR T-cell. There are monoclonal antibodies which are used, vaccines which are used, and then immune modulators. So basically, these immunotherapeutic drugs, they harness the immune system, which otherwise remains dormant due to the cancer development. So the idea is to shake up the immune system so that the dormancy that came in due to the cancer is removed and the immune system wakes up and fights the cancer in a much better way. So this immune checkpoint inhibitors are most commonly used, and this is how people do. This is again done in experimental setup, and there are encouraging results that have been found in colorectal cancer, in melanoma, and in lung cancer. So what they do is they take these eubiotic gut microbiota and they look at the dysbiotic gut microbiota. And the eubiotic ones are the ones they respond to the treatment, and the non-responders would be the one who have a dysbiosis. Dysbiosis basically reduced diversity, reduced abundance of microbiome. So the responders are identified, the microbiome is studied, it is taken out, and then this is given to the non-responders or to the people who are not responding to the chemotherapy or the immunotherapy. And this is given along with the immunotherapeutic drug. So this gut microbiome or the bacterial consortia or the FMT can be given as a complementary to the immunotherapeutic drug, and the chances are that people who were not responding earlier will now respond better. So the success of this depends on the diversity, abundance, and how they respond to the immune system. So towards that, this is the future of oncomicrobiomics. Those who are looking at PhDs and postdocs outside India or within India, if you are into microbiology and if you want to get into microbiome or oncomicrobiomic studies, this is the way one can do it. Basically, cancer cohorts are identified, the tumor microbiome is studied, the blood microbiome is studied, and the fecal microbiome is studied, and then they are sequenced. They're also done, there is something called culturomics nowadays, where MALDI-TOF is used to identify bacteria by culturing, and then flow cytometry is done, then immune cells are identified, cancer cells are identified, microbiota is compared, the metabolites are identified, and then there are different preclinical studies that can be done in many different ways. One can make organoids, one can make organ on chip, and finally in vivo mouse models can be done. And then the final thing would be one can identify biomarkers which are linked to a particular condition. So this is the future of microbiomics. Now the second part of my talk, I will try to complete quickly, is the in-house projects that I'm working on, which can be linked to oncomicrobiome. So the first project, it is done by Aprati. She is right now pursuing her MSc in Netherlands in the Radboud University. She was here in the institute, and we did a project together on how a wild type and a cancer-prone animal, whether there is any difference in their gut microbiome, and if there is a difference, whether they have a differential response to inflammation when they are infected. So we have two types of zebrafish. That TP53 is basically a tumor suppressor gene, and if P53 is not there in an animal, they are prone to cancer development. So we have these two fish. One is P53 mutant, the other one is the wild type. They are grown in identical conditions, same water, same feed, same environmental ecological conditions. So we took the gut out of this fish. Instead of going by a totally bioinformatic approach, we did a culturable approach. We tried to see how many microorganisms we can culture, and we isolated the selective species, and then injected this into germ-free zebrafish. Germ-free zebrafish means a system where there is no endogenous microorganisms, and we looked at the anti-inflammatory response, and we compared this between the wild type and the mutant. So these are the wild type. If you look here, this is the wild type fish is on the left-hand side. A mutant, which is showing tumor, is on the right-hand side. There you see, we identified 5 fishes from wild type and 5 fishes from a tumor. We isolated 28 bacteria from the wild type and 21 from the tumor fish, and we characterized them by biochemical tests, 16S RNA sequencing, and by species-specific PCR. So this is what we found. We looked at the total count, and then we looked at the biochemical tests, and we also looked at the 16S RNA sequencing. And then finally, we found that these are the species that were culturable. There could be many that were present, but since we did not go by a shotgun or total 16S RNA sequencing, we looked at only those which were culturable. So what we found was Aeromonas species, Aeromonas hydrophila, Bacillus vibrio cholerae, environmental strain of Vibrio, Staphylococcus, and Staphylococcus aureus. And then we looked at the diversity. On the left-hand side is the TP53 wild-type fish, and the right-hand side is the mutant. So what we found is, in the wild-type, they had greater diversity. They had diverse variety of microorganisms, but they had reduced bacterial load. On the other hand, the P53 mutant, it had higher bacterial load, but lesser diversity. And also we found that Vibrio cholerae and Aeromonas hydrophila, which are opportunistic pathogens, they were present only in the mutant gut and not in the wild-type gut or the normal gut. And then we looked at the antibiotic resistance of these isolates. 80% of the isolates were resistant to epinephrine, 90% were resistant to cefotaxime and cotrimoxazole, and almost 100% of them were resistant to tetracycline, nalidixic acid, and ciprofloxacin. So what it shows means that most of these isolates are resistant to antibiotics, which is actually true for commensal microbes. And then we did the infection study, and we looked at the expression of IL-10 and IL-4. IL-10 and IL-4 are anti-inflammatory cytokines. So their job is to reduce the inflammation. So if there is an inflammation, these cytokines are supposed to be expressed more. And we also know that chronic inflammation is tumorigenic. So if these expression is downregulated, that means the animal is not able to mount an anti-inflammatory response. So what we did, we took the wild-type fish and we took the mutant fish. We injected or we transfected or infected them with Staphylococcus and Vibrio, and we looked at the expression. So what you see here is IL-10 expression in wild-type and in P53 mutant. If you look at the right-hand side, over the day 1, day 2, and day 3, there is a consistent downregulation of IL-10 in the P53 mutant. Whereas in case of wild-type, the downregulation is variable. So that means in the wild-type system, the fish is trying to mount an immune response. But in the mutant fish, the fish is not able to mount an immune response. And so there is a consistent downregulation of the anti-inflammatory IL-10. And the same thing was seen in IL-4. Here also, the right-hand side, if you see at different time points, the immune system, there is a consistent downregulation of IL-4 in P53 mutant, whereas in the wild type, it is variable. So what we understood from this study is that the gut microbiota is variable between normal and cancer-prone fish. And normal and cancer-prone fish show variation in the diversity and the abundance, although they are grown in identical ecological conditions. Cancer-prone fish consistently fail to mount anti-inflammatory response when infected by opportunistic pathogens. So the data, what we obtained, suggests the role of genetics in the variation in gut microbiota and the role of microorganisms in anti-inflammatory response. So this paper is right now under review. So hopefully we'll be able to get the publication soon. The other one is basically on oral microbiome project. So the previous one was the gut microbiome. This is on oral microbiome. So what we did, we took oral cancer patients, and generally people do is they compare the microbiome of a cancer patient and a healthy individual. So in this case, what we did not do this. We took the same patient, we took the sample from contralateral site. What it means is we took the sample from a tumor site, and from the same patient, we took the sample from diagonally opposite the non-tumor site. So the idea is to see if the same microbiota, if the same oral cavity, whether there is any specificity of a microorganism which is harboring in the tumor site and which is not present in the non-tumor site. And this is done by Aparna, who is a MSc Biomedical. She is in the second year. This is our unpublished data this is what I'm presenting now. So here we looked at the 16S RDNA sequencing by next generation sequence, and we looked at the microbiome. So we have a preliminary data. I'm going to show only preliminary data. This is a pipeline that we follow. A lot of bioinformatics softwares, so I need not go into the details into that. I'll show you this. So this is the distribution of the phylum that we found between normal and the tumor. The left-hand side is the normal and the right-hand side is the tumor. N1 to N8 are samples taken from the normal side of the mouth, and T1 to T8 are the samples taken from the tumor side of the mouth of the same patient. Ideally, we do not expect any difference because we expect the microbiota to be same. But surprisingly, we do find a difference. If you see here, that is something which I have highlighted. N2 and N3 and T2 and T3 have a different profile as far as the phylum of the bacteria or the microbiome is concerned. Similar thing is obtained when you look at the genus level. Here also we see a difference between the tumor and the normal part of the tissue. And then we look at the LDA score. If you compare the microorganism, these are the species over here, you see some of them have negative LDA score, some of them are positive. In fact, in the normal part of the tissue, there is a positive score for Posticibulus and Brevidian monas, whereas in case of the tumor side, there is a negative score for the other three species mentioned here. These are preliminary data. We need to do a little more study on this, but because this is a microbiology conference, I thought I can show this data to this audience. So, conclusion, cancer microbiome is an exciting field and it has tremendous potential. A lot of people are working in it, and it's a very new field. And microbial-based diagnostics, prognostics, and exogenous microbial therapeutics have, of course, several challenges. We do not deny that, but there are many things that need to be overcome before it becomes actually a standard practice in cancer therapy. And because chemotherapy and radiation therapy has a lot of side effects, these are not preferred cancer therapies, and people are looking at alternative ways. And despite the challenges, if we have a better understanding of the role of microbes, it can really develop as a new tool in cancer management in the years to come. Finally, I thank my team, the Division of Molecular Genetics and Cancer at my institution, all the students and the PhDs and other faculty. And there are three more faculty members here, Dr. Dikshit, Dr. Krishna, they are my own faculty here. They are helping us in this microbiome project. And there is Dr. Vinay Kumar, who is oncosurgeon, who is involved in the oral cancer project. So with this, I thank the organizers, particularly Professor Jayashankar, once again for the opportunity, and I thank everyone for their patience here. Thank you.
Thank you, sir, Dr. Anirban Chakrabarty, for your informative talk on oncomicrobiomics, new insights into cancer diagnosis, therapy, and prevention. On behalf of the conference organization, I welcome some questions to Dr. Anirban Chakrabarty from the audience. Any questions? Open for discussion.
Dr. Chakrabarty, nice presentation. Can you hear me, sir?
Yes, thank you very much. Thank you.
Wonderful. Yeah, with reference to the FMT fecal microbiota transplantation,
right.
Maybe with reference to colorectal cancer, it is okay, or we appreciate that it may work. It should work. But what is your opinion with reference to the other type of lung cancer is there, or the melanoma, other cancer?
Yes, thank you. That's a very nice question. In fact, FMT has been successful, but there are now red flags for FMT also. there are certain cases where people have seen certain adverse effects in patients due to the transfer of pathogenic bacteria as well. So, but nevertheless, I think the success story is much better compared to the adverse effects that you have. Having said that, people have already done in lung cancer, and people have also looked at oral cancer. So these are two cases in addition to colorectal cancer where people have used FMT successfully. Yes.
Other thing was, what was the basis to select zebrafish as a model system? Why not some vertebrates, or which is more closely related to human being? In other words,
zebrafish is a vertebrate.
Okay.
It is not a mammalian system, but zebrafish is having a lot of inherent properties, particularly for cancer research, because there is structural and functional conservation. The genome sequence is almost 80% similar to human beings. And there are certain things you can do in zebrafish which you cannot do in mice or a rat model. The final thing is they are enabled to xenotransplantation or transplanting cancer cells from human to them. And you don't need to have a nude system. In mice, you need to have a nude mice. In zebrafish, you do not need a nude mice, nude zebrafish to avoid immune response. So there are many advantages. And the other most important is they can develop spontaneous tumor. In fact, one of the historical model for neoplasm is fish. They develop spontaneous tumors even without treatment. And the TP53 mutant that I showed, the tumor development is identical to human beings.
The last one is with reference to the cancer, the biomarker which you identify when cancer is pronounced. Does gut microbiota is really suppressing to in such a condition with reference to the biomarker?
Biomarker, basically, biomarker is not going to help in therapeutic. It could probably predict, or it could probably help in diagnosis. So the biomarker is basically nowadays in cancer, if you're following cancer research, people have moved from traditional biopsy to liquid biopsy. Liquid biopsy is basically looking at cell-free DNA, cell-free RNA, cell-free microRNAs, and so on. And these are present in the peripheral blood. So in similar lines, people can expect that if there is a particular species of bacteria colonizing the gut or any other area of the patient, it could be mouth, it could be skin, it could be the urogenital area, they could potentially be in circulation. And if they are identified from peripheral blood, they can be proposed as important biomarkers, which can actually, and the load could actually define whether they are probably carrying a certain kind of cancer in them. In fact, that has been tried for pancreatic cancer. People have looked at certain archaea, which is present in the oral cavity, and they have used it for diagnosis of pancreatic cancer. And pancreatic cancer, if you are aware, it is very difficult to diagnose cancer. By the time it is diagnosed, it is stage 4 or stage 3. So it's a kind of a silent cancer. There is absolutely no symptom in the patient. So I think this is one example where the prediction can be made much earlier.
Thank you. Thank you.
Thank you for clarifying the doubt, sir. Any other questions? So Dr. Anirban has given a beautiful information on links between microbiome imbalance and onset progression of diseases, oncomicrobiomics, hallmarks of cancer, microbial-derived biomarkers, discovery of the role of gut microbiota in tumorigenesis, modulation of gut microbiome to fight against cancer, immunological modulations, and cancer-associated microbiota. And this is indeed a novel research field for young researchers to take up and work on further against anti-cancer activity, mechanism of anti-cancer, etc. On behalf of the National Conference Organizing Committee, I thank you, sir, Dr. Anirban Chakravarti, for the talk. Thank you. Now I request Ms. Sharanya to read the next speaker. Thank you, sir. Thank you very much.
So I would like to log off. Thank you once again, Professor Jayashankar and team, for extending the invitation.
Thank you, sir. Thank you.
முடிச்சிட்டாங்களா?
ஆமா முடிச்சாச்சு.
ஓகே ஓகே. அப்போ
அவங்களே அன்மியூட் பண்ணாங்க.
The next lead talk of the day will be delivered by Professor A.P. Lipton, sir, who will join us online. I call upon Mr. Ningaraju S. to introduce our lead speaker, Professor A.P. Lipton, sir.
Good afternoon, everyone. I am Ningaraju S., Research Scholar, Department of Microbiology. It's my great pleasure to introduce today's lead speaker, Dr. A.P. Lipton. Dr. A.P. Lipton, sir, expertise in marine biotechnology and aquaculture, former principal scientist and head Marine Biotechnology Division, CMFRI. Former UGC Emeritus Fellow and head of Center for Marine Science and Technology, Mananmaniam, Sundarnath University. He has nearly 46 years of experience in research, teaching, research management, research guidance, and research extension experience. Sir has served as Biotechnology National Associate of the DBT, Government of India, and as well as DBT Overseas Associate in 1995. Sir is the visiting faculty of the Department of Microbiology, Oregon State University, USA. Sir was deputed by the Government of India to countries like Taiwan, Islamic Republic of Iran, Philippines, and USA for scientific pursuit. He was a former Senate member of the Mananmaniam Sundarnath University and former chairman of Board of Studies in Marine Biotechnology, Microbiology, Microbial Technology, and Coastal Aquaculture. He is recognized as UGC Emeritus Fellow from 2015 to 2017. Sir was an expert member of District Fisheries Development and Participatory Management of Coastal Resource, Department of Fisheries Development of Tamil Nadu, Kanyakumari District, Tamil Nadu. His current research areas are marine microbes and marine natural products, marine microbiome, marine bioactive metabolites. Sir had guided several PhD scholars, MPhils, and MSc students in their projects and theses. Sir had published more than 310 research papers, technical articles, abstracts, 3 scientific books, 15 research manuals, and 10 scientific DVDs. Thank you. Sir, I welcome to the session.
Professor A.P.
அவங்களே அன்மியூட் பண்ணிட்டாங்க. இனி இதுக்கு வாங்க. இதுக்கு பண்ணு.
Over to you.
Can you hear me now, please?
Yes, sir.
Okay, shall I start?
Yes, sir.
Okay, good afternoon to all of you. I take this opportunity to thank Professor Dr. Jayashankar and all his colleagues for providing me this wonderful opportunity to share some of my expertise as well as experiences with many of you assembled here. My topic is on novel discoveries and the future prospects of marine biotechnology. For the past few hours, you have been listening so much and you are packed with a lot of research information. I have also heard some about probiotic microbes and their importance and the cancer biology related items. In fact, a lot of information has been shared, and now I am taking you all to a different habitat. I am taking you marine habitat. You know, ocean covers about 71% of Earth's surface, which is almost equivalent to 360 million kilometers square. And we have only a landmass of about 29%. Some interesting facts about the oceans is average depth is about 3,720 meters and deepest point is known as the Mariana Trench in the Western Pacific. Another very interesting factor about the ocean is ocean ridges. They form great mountain ranges, almost about 64,000 kilometers long, which weaves its way through all the major oceans. Rather, it is the largest single feature on Earth, and the highest mountain is Mount Mauna Kea. You can see here, it is its height is higher than the Mount Everest. It's about 10,203 meters, whereas Everest is about 8,850 meters. Depending on the light availability, the oceanic divisions are informed, particularly epipelagic, mesopelagic, bathypelagic, abyssal pelagic, as well as hadal pelagic. The hadal pelagic is a very interesting. You can see now fully dark. Even in this darkness, there are fishes which live. Very interesting information is a discovery of a symbiotic bacteria which was available in the deep water angler fish. This is, this anglerfish, which is known as a humpback black devil, and they have an antenna which always have a lot of light emitting bacteria. Particularly the female anglerfishes, they sport a glowing lure on top of their foreheads, basically a pole with a light bulb on its ends, where the bioluminescent bacteria live. The light emitting bacteria, they have two important roles. One is attraction of the prey as well as a potential maze to the fish. In fact, a lot of studies have been done on this work, on this fish. The researchers discovered that the bacteria, they have lost some of the genes that are needed to live freely in the water. Because the fish and bacteria, they developed a tight, mutually beneficial relationship where the bacteria generate light while the fish supplies nutrients to the microbes. So definitely for the youngsters, those who want to do further research, deep sea marine bacteria for light generation will be a very good topic. Another interesting observations recently made was the discovery of a new species, Mariana snailfish, which was available at about 11,033 meters. It's the deepest record fish in the sea, and in November 2017, scientists have reported the discovery of this species. And as you can see here, the skin is white and translucent without any scales. Its appearance is both strange as well as amazing. Not a lot is known about how the fish lives at such depth and pressure, but snailfish do get to live a predator-free life at the top of a rich food chain. This is how they collect this kind of deep water snailfish from the Mariana Trench. Another interesting feature here, it was published in January 2020. These hadal trenches are characterized not only by hydrostatic pressure but also scarcity of nutrients and high diversity of viruses. A novel bacterium was isolated from the guts of these snailfishes. This is named as Candidatus Mycoplasma liparidae. And this draft genome of Candidatus Mycoplasma liparidae was successfully assembled by Chinese researchers with 97.8% similarity by hybrid sequencing. So these findings provide further insights into symbiotic microbial associations in the hadal biosphere. Now I want to emphasize to the people assembled here, the world's oceans teem with something like 700,000 to 1 million species, and up to 2/3 of which biologists have never named or characterized. This is again, requires a lot of research work in the marine biology area. Of course, I want to show some slides, to create an interest in you. The marine specimens, they have, no religion. Rather, it represents almost all the religions. This is a, crab, which has a cross on its backside, so it is known as a Siluvai Nandu in Tamil. This is a Quran angel where some of the markings are, in the Arabic letters. And this is a sacred chunk, which is normally kept in the Shiva temples all over the India. And this is a shells displayed at Kanyakumari, and these are some of the other this one. This particular sea fan, I want to emphasize because there was a nautical gold rush in 1970s, mainly to isolate prostaglandin from the sea fans. And you can see a lot of other this one food and livelihood, and is always related to marine fisheries. Here you can see the fish sale on the seashore, and a lot of tunas landed in the Tuticorin coast. There are a number of ornamental fishes are also, like the surgeon fish, the oval butterfly fish, these marine eels, and another important thing is the seahorses. This is a very, very important fishery because it's a medicinal fishery. And in the traditional Chinese medicine, it is valued very high. And of course, the government of India has banned about the fishing of um, uh, this seahorses from India, because in traditional Chinese medicine is used to cure many diseases like asthma and other breathing problem related diseases and sicknesses and lower abdominal pain related ailments. So in 1997 there was a very huge overfishing of these particular seahorses, and they were exported in dried condition in many of the countries. Now the government of India has banned. And very interesting features are there. Of course, the time is not much to explain all these in details. Normally, the males have the pouches to keep the young ones, so the females' characteristics are taken by the males. And lobsters, it is adding a lot of value for the menu, lobster-based menu. And mussels, that green mussels as well as brown mussels are landed all over the coast. And this is a site very near to Kanyakumari where the mussels are landed in large number, and they are exported to nearby places in truckloads. It is exported to different parts of Tamil Nadu as well as Kerala. This is of course, mussel meat, which is delicious and a delicacy. Apart from this food and other ornamental purposes, the marine ecosystem also offer livelihood for the old age persons, because as you all know, October 1 is known as the International Old Age Day. Very interesting information given by one of our scientists is the fishes talk to each other. The fishes talk to each other because you can see a lot of snappers like fishes here. They produce various sounds including grunts, croaks, clicks and snaps that are used to attract mates as well as to keep away the predators. The big eye cat, a tropical fish, produce sounds using their pharyngeal teeth. The undersea world is not as quiet as we thought, according to the New Zealand researchers who found they can talk to each other. This is a lady, Susan Milliers. She has given a very good, she has brought out a very, very informative article about how fish communicate with each other when they go in a school. Another interesting thing I wish to share with the young researchers is marine biotechnology. What is marine biotechnology? Marine biotechnology is an innovative field of research in science and engineering that encompasses the use of marine bioresources for a multitude of applications, including the production of food, fuel, materials, or specific or often bioactive compounds, and may contribute to green growth and sustainable industries and prosperous communities. So, marine biotechnology is really a very, very important field. So this is another interesting fish. It's known as a box fish, where they produce some sort of a toxin. In fact, when I was working in Trivandrum coast, I have collected this kind of a box fish, and one of my person who accompanied me, he put the box fish into another, bucket of full of live fishes like the butterfly fishes, squirrel fishes, parrot fishes, and other fishes, and all of them they died within a few minutes. Why? The box fish, they produced a toxin known as a tetrodotoxin, and this tetrodotoxin is a highly potent toxin which was secreted by the box fish, which has resulted in the death of other fishes. This we have evaluated the toxicity also. The tetrodotoxin acts as a membrane receptor and blocks the sodium channel in the nervous system. They also interact on the external surface of sodium channels and interrupt the passive inward influx of sodium ions. Thus, The tetrodotoxin is a potential chemical tool for neurophysiology and neuropharmacology studies. And now recently, we have studied only about the toxin. Now the bacterial involvement in tetrodotoxin in the pufferfish was noted in the fish known as Fugu rubripus. Japanese scientists, they found that TTX producing bacterial strains were Bacillus species, 19 strains, and one actinomyces strain was isolated from the pufferfishes. And these bacteria, they have the capacity to produce the tetrodotoxin. Now, tetrodotoxin in new medications is aimed at treating chemotherapy-induced neuropathic pain, an often debilitating condition experienced by more than 40% of patients who undergo chemotherapy treatments. Now, tetrodotoxin is a very important medicine, and the price is about $145 per 1 milligram. So here I want to emphasize the innovation is a process of making an idea or invention into a good of service that creates value for which the customers will pay. Discovery is a process of finding information, a place, an object, especially for the first time. So here, when we talk about the marine biotic compounds or marine biotechnology, we have to go for marine studies of the marine habitat and scuba. Scuba, you must have, if you want to study very detailed about the marine ecosystem, you have to definitely go through the scuba training. Scuba is known as the self-contained underwater breathing apparatus, which is very useful for the studies related to marine biology. Of course, I'll skip this one. Now, as I told, many of these organisms have a lot of bioactive potential. And now, designing new gadgets for safer and efficient collection is another important area of research. Nowadays, you can see a lot of specific targeted or even depth-wise targeted collections can be made using such sort of a unmanned cruise, or even with manned cruises can be possible. These are some of the underwater robotics or marine robotics, where the bottom collections as well as for coral research is also possible. These are what we call remote operated vehicles or ROVs, which are very important for the collections of samples from bottom of the sea. And this is also useful, particularly nowadays, we have been thinking of a lot of plastic pollution in the marine habitat. And a lot of corals have been, destroyed mainly because of this plastic pollution, because these, plastics, debris, they go and attach themselves on the corals. And as a result, we lose a lot of coral reefs. Nowadays, we have been planning, this kind of ROVs to go and clean the corals in such kind of areas. And seawater has a lot of bactericidal effect due to the production of antibiotics by planktonic algae as well as bacteria. Rosenfeld and Zobel, you would have heard about Zobel marine agar, isn't it? The Zobel in 1947 showed that bacteria produce antimicrobial products, and marine organisms encompass a complex and diverse assemblage of microscopic life forms and occur throughout the oceans. In the year 1966, highly brominated pyrrole antibiotic was first documented from bacteria, and the bacteria was reported to be Ultramonas, which was isolated from the seagrass. The compound was highly active against gram-positive, but it was not active against gram-negative bacteria. The minimal inhibitory concentration, that is MIC, was 0.0063 to 0.2 microgram per ml. A lot of antibiotic purple pigmented chromobacterium, which was isolated from North Pacific, and several antibiotic compounds were isolated from this particular bacteria, like pyrrole, tetrabromopyrrole, hexabromo-2,2-bipyrrole phenolics. And these bromopyrrole compounds showed antimicrobial activity against Staphylococcus aureus, E. coli, Pseudomonas aeruginosa, Candida albicans, and it is also autotoxic to the same genera. The mechanism, as per the literature available, is the bioactive metabolite producing bacteria, they evolve the capability or ability to concentrate the bromide ion from seawater. And through oxidative pathways, this is incorporated into inorganic compounds, and this results in the bromination of metabolites, which possess highly enhanced bioactivities of these metabolites. Thus, the oceans, they teem with a lot of microorganisms, and many of these microbes fundamentally influence the ocean's ability to sustain life on Earth. Some microbes living and transported in ocean water, however, threaten human health also. As I told earlier, they are recorded from extreme environments of pressure, salinity, as well as temperature, and they developed unique metabolic and physiological capabilities that not only ensure the survival in extreme habitats, but also produce novel metabolites not generally encountered from the terrestrial microbes. This one particular explosive bacteria, which I want to show to you. This bacteria was discovered from submarine volcano, which erupted off the coast of Canary Islands in 2011, wiping out most of the marine ecosystem. And these areas have been since colonized by new species of Proteobacterium with filaments that cling to each other and form a very massive white mat covering about 2000 square meter at depths of 129 to 130 meters. And this is, as I told, the remote operated vehicle. With that they made observation. This bacteria was characterized Thio lava veneris. And the International Institute of Species Exploration, they have named this as the T. veneris, one of the top 2018 top 10 new species. And another one is about the interesting finding of sponges at more than 4,000 meters in depth. This is a particular sponge which was available about almost 3,960 meters. And this again is another interesting sponge because this has all the potential for anti-cancerous and other activities. I'll skip up this one. This is a very interesting phenomena where color morphing clams could inspire new TV or smartphone or TV screens. These iridescent cells in the exposed flesh of giant clams are known as iridocytes. These cells generate dazzling array of colors, including blues, greens, golds, and more rarely white. This will help scientists to design more efficient solar panels and television and smartphone screens that is easier on the eyes. The word iridocyte comes from Latin ancient Greek, where irido means rainbow and cyte means cell. So the iridocytes they got their names. And this will definitely give a lot of intuition for the younger generation to make use of such color morphing clams for the TV screens. Another interesting thing is about clams. I have seen many of the clams, boring inside some of the mollusca, that means shells, which I showed earlier about the sacred chang and all those things. These have the capability to bore them, and later on they found they have a lot of acid secreting, tissues there, and with this red zones indicate acidity measured by flat pH detector. Even with a pH paper also it is possible to discover. These are the squids. These are the octopus. They are very intelligent animals. And a recent study indicates that squid and octopus, they can edit their RNA, and this enables the cephalopods to build proteins for which DNA lacks a blueprint. In fact, it allows them to make several different proteins from identical strains of RNA. So this is another interesting feature from the marine realm. Okay, another interesting thing, this is a shark. Can you see a lot of other things which are attached to the shark? The sharks are very fast swimmers, but these are the remoras which attach themselves to the shark, and they attach with the, there are about 13 species of remoras in several genera. Almost all of them have similar designs, and this is again another intriguing thing for the technologists. And as a result of this interesting observation, they could come up with a bio-inspired composite robo. A team of researchers, they developed a prototype inspired by the Remora's adhesive disc, and the device can stay stuck when pulled and twisted, similar to how the Remora can withstand the shearing force of water while attached to the moving shark. They are very fast. Definitely, these Remoras can do it with this kind of suction disc, and a similar bio-inspired composite robo can be developed, and they have already tested it. And the researchers envision that a Remora-inspired adhesive device could aid ocean exploration, ship repair, biological research, among other potential uses. This design has potential to greatly extend the range and endurance of ocean exploration and underwater vehicles. Dear friends, we are fascinated by the intelligence of marine organisms. You can see this is a rock very near to Kovalam beach, where you can see a lot of barnacle settlement, mussels, as well as seaweeds. God has given them a gift, really a very good gift, because they produce adhesives with which they are able to adhere to the rocks, or they attach to the rocks and remain stable in spite of the high wind velocities and all. This is of course an underwater photograph where you can see the mussels, attached to rocky substrata, and you can see the mussels secreting the Byssus threads with which they are able to attach. This is a glass surface. Even in the glass, in the laboratory, we brought the mussels, and now they are able to produce the adductor muscles. As a result, they are able to, stick on to even this, very slimy, substrata, even in glass substrata. They produce hair-like Byssus threads in a process similar to injection molded plastics. In these structures, finding some rare amino acid group known as IDOPA. IDOPA was recorded from this kind of, Byssus threads, and this is how the mussel culture activities are also going on. And I'll just now, as I told some time back, even in rough conditions and in water conditions also, the mussels are able to thrive there, mainly because of the mussel adhesive proteins or MAPs. A promising area is operating newborn babies, because like us, newborn babies cannot be sutured. They require a great challenge because the fetal membranes are extremely delicate and difficult to suture, and this could become much easier with mussel-inspired glues that work even under the wet conditions and don't trigger problematic immune responses from the host. So the Acro Biosystems in USA and China, the leading manufacturers of recombinant proteins, is now producing mussel adhesive protein or MAP, and 1 milligram is costing $110. So you can see enormous potential, enormous areas of research you can do, particularly in these kind of areas of mussel adhesive protein. Always it fascinated me. That's why I shared this particular point with you. Likewise, there are many new therapeutic substances, bone substitutes can be used. I don't have much time to share, like the coral pieces and all, they are definitely substitutes for bone, and it can be implanted also in the patients who are suffering with a lot of multiple fractures and all those things. And soft corals is another area where, collagen powder plus vitamin is now coming into the market. Another interesting piece of information I share with you all today is the marine bacterium has been genetically engineered to produce spider silk. The spiders, they produce spider silk, which is very strong and lightweight, which is made from the silk proteins. And these researchers succeeded in producing the spider silk using photosynthetic bacteria. The spider silk can be used to create lenses for imaging human tissues. Spider silk could be also used to make robotic muscles. And this is again another important thing, because the spider no longer can be used, but the marine bacterium can be genetically engineered to produce spider silk. The spider silk was produced by using the purple colored marine bacteria Rhodovalvum sulfidophyllum. Scientists at the Ricken Center for Sustainable Resources Science have created a microbial cell factory that needs only seawater and a source of sunlight, carbon dioxide. Pardon? This process is carbon neutral, making it eco-friendly and sustainable. These bacteria are photosynthetic, which means it uses sunlight to produce its own nutrients in nature. Another interesting area is pearl culture, where we get inspiration by nature and we get achieved by science, because this is a normal pearl culture program. But of course, of late we have started using the technology for in vitro pearl production in the laboratory itself by tissue culture, in vitro tissue culture methodology. Now coming to the marine macroalgae or the seaweeds, there are rich sources of, because most of the rock areas have a lot of seaweeds attached. This is a seaweed growth you can see on the rocky areas from Trivandrum coast. And these seaweeds are also cultured. This is a Cuppaphagus seaweed which is cultured for many of the food products as well as for cosmetic purposes. This is a harvest of seaweed. You can see number of floating structures which are used for culture of seaweeds. And of course, our country has a lot of potentials of seaweeds. Unfortunately, I should say again, we are not using the seaweed to the fullest extent possible. This is a slide I have taken from Taiwan. There are a number of people, there are manufacturers. They are making ready to use seaweed products, including biscuits and other cookies. They are on the display. The seaweeds are also very, very useful for the beauty care products, including the soaps and creams and lotions. This is again another earthly friend running shoes made of algae. Again, it has a lot of potential because you can avoid the use of plastics or synthetic products. I'll quickly pass it on. There are a number of bioactive compounds isolated from macroalgae. For example, at this side I have given the macroalgae, the species. This is Alva fasciata, is a normal species. A lot of activities are noticed: antiviral, antibacterial, anticoagulant, algicidal, antifungal, microalgal growth enhancement, cytotoxicity, immunosuppressive as well as immunostimulant activities are noted. Of course, there is a lot of possibilities to do a lot of work on this kind of marine macroalgae, particularly the seaweeds. I will show you only one slide, which one of my students have completed. Very, very important information is about the immunosuppressant activity of marine macroalgal extracts, where a simple, methodology was evolved by us. We have used only the guppies, that is normal fish available, which is it can be easily reared in captivity and transported also to the lab very quickly without any damage. We have made only a scale transplantation assay whether the immunosuppressant activity is present in macroalgal extracts. We found complete immunosuppressant activity recorded from the marine macroalgal activity. Another important thing is the seaweed incorporated diet, which was incorporated in the diet of shrimps, that is prawns, where the feed was coated with the seaweed extract. We could get a lot of very good harvest of the shrimps, rather conventional than the control compared to the control. The harvested shrimp showed much growth. That is, average body weight was 35 to 40 gram in about 120 to 150 days of culture. So there is a lot of marine macroalgal possibilities of using the marine macroalgal extracts. Another interesting feature of the marine macroalgal extract is bioadsorption, where one of my students have completed a study about heavy metal removal by the seaweed products. This is bioadsorption, through which she could find higher arsenic adsorption was noticed when the seaweed sargassum, vitae, and the Gracilaria corticata were used as dried powder. Of course, another important resource is the seagrass, where you can find a lot of seagrasses, particularly here. This is one area near Kanyakumari where you have got almost a very big meadow of seagrasses present there. And these students, as one of my scholars have identified, the blades of seagrasses, that is seagrass blades, have a number of very important epiphytes as well as endophytes. She has studied in two seagrasses, Halodeul pinifolia as well as Stringodium isotifolum. These are some of her observations. And one important thing is, not only antibacterial activity was reported from the seaweed, seagrass, seagrass epiphytic bacteria, but also mosquito larvicidal activity was reported, and insect repellent activities of seaweed extracts was noted. And this insect repellent activity, this is a storage pest, and which is known as Tribolium castaneum. And she found that this seaweed extract has also has a lot of potential as insect repellent activity. Another area is biopreservation. Nowadays, we use a lot of chemicals for preservation in order to avoid harmful chemicals. One novel approach was there from our laboratory. You have seen that batfishes, their mucus, they secrete a lot of mucus. The fish mucus contains this, particularly the batfish mucus, tuna mucus, perch mucus. They were, they were isolated and they were evaluated for lactobacillus strains. And interestingly, several strains of lactobacillus were isolated from the mucus of the batfishes. And with this bacteria, very important, reef called fish fillers were, treated with this lactobacillus as a biopreservative agent. And after about different days, it was studied, and which gave the importance that the lactobacillus isolated from this batfish mucus has a lot of biopreservation ability, particularly the lactocockin PSY showed more effectiveness in extending the shelf life of stored fish. And this also has exhibited inhibitory activity against Listeria monocytogenes, which is considered as a food pathogen. There are several studies done by the researchers, our group. They have found that Listeria challenge studies also they have done, with which it is noted that it is possible to have biopreservative effect using lactobacillus isolated from the fish mucus. Another interesting observation is symbiotic organisms, particularly the sponge microbial interactions. Of course, there is a lot of slides I want to show. I will skip some of them. The sponge extracts, they have a lot of antibiotic activity. Initially, we studied only about the sponges. We collected the sponges, then extracted with organic as well as the aqueous extracts, and we found that antibiotic activity. Later on, we found the bacteria present inside the sponges have the same abilities as that of the extracts by the sponges themselves. So now we have been concentrating mainly on the bacteria which are living inside the sponge tissues, and we have been some of my students are working on that. And now this is coming to another area is when we collect a large number of sponges, there will be a lot of problems because the biodiversity will be spoiled. The sponge fauna may be threatened. So now one of my scholars, Dr. Rajendran, he has started a new work on sponge culture in the sea itself, open sea itself, to get novel metabolites, and that's why there is no need to remove them from their natural habitat. This is a kind of long line as well as a tray method where he has studied that the, the sponges, when they are in small pieces, they grow to larger numbers, and they also have some sort of antimicrobial assay, cell viability test, anti-inflammatory activity. Now coming to jellyfish, this is a new source of anti-proliferative drugs can be exploited from jellyfish. Jellyfish is, of course, normally we avoid them because they have their own negative effects on human activities as well as human health. But of course, later on, recently, the studies have proved that these jellyfishes can be exploited for anti-proliferative activities. So here, in a nutshell, I am giving the avenues for sustainable bioprospecting include sponge-derived drugs and bioactive compounds are synthesized associated with from the microbes associated, and sponge-associated microbial clone depository is important, and these are sustainable sources for bioprospecting. There is no need to go back to the sea for collection of rare specimens or destroy their habitats or destroy the organism. We can have them in our lab itself, and it will provide a sustainable source for bioprospecting. I'll cut short. One interesting thing is the microplastics is another important area, particularly in the sea. Microplastics are found in human blood for the first time. They cause damage to human cells in the laboratory. This is a photograph given by David Kelly. Microplastic pollution have been detected in human blood for the first time, with scientists finding tiny particles in almost 80% of the people tested. We have to be very cautious about this. And of course, there are a number of reports nowadays available about plastic eating bacteria. They have found out Idonella saccharensis, which has a capability to, degrade or that eat away the plastic, which is identified by the Japanese scientists. And biofuel is another area where exploiting the algae for sequestration of carbon dioxide and use as light energy for biofuel to replace the crude oil usage from industrial sectors for nutrients. And the hydrothermal bacteria for biofuel using macroalgae is another important area where we can go for next generation fuel production techniques. The global market for marine biotechnology is estimated, of course, about 4.1 billion in 2015, and definitely it has a potential to reach about 6.4 billion by 2025. The key drivers for the market are new applications of marine-derived enzymes in the cosmetics industry, and use of marine algae and microalgae in biofuel production. Of course, only thing is you can have these kind of things only with transdisciplinary collaboration. That itself is a very important area where we have to have our collaboration with other subject area experts also. Then only we can advance in this kind of marine biotechnology areas where we can produce important products for mankind to develop. My dear young researchers, train your mind for information processing. Of course, this is out of subject. Here I am giving a slide. The ants are better than Google search engine for processing information, more efficient than even the Google search. They have found that they are very, very efficient in processing information processing. Your ability to generate innovative ideas is not merely a function of the mind, but also a function of key behaviors that optimize your brain for discovery. Associate, question, observe, network, and experiment. These 5 key behaviors will definitely optimize your brain for discovery, and definitely you will be successful. In fact, I thank Professor Dr. Jayashankar. In fact, I was in your campus about 8 years back in February 2014, I think. 2014 with one of my friends, Professor Dr. John Jyoti Prakash. I thank all of you for wonderfully and patiently hearing the talk. Good luck. Thank you one and all. Thank you. With this, I stop my talk.
Thank you, sir, Professor A.P. Lipton, for enlightening with your informative talk on novel discoveries and future prospects of marine biotechnology. Indeed, you have taken us to the vast information of marine biotechnology, including microorganisms and macroorganisms, natural products, bioactive compounds, and their wide application. There may be many doubts and questions from the audience. I think they can post the questions by email to Professor Lipton. I hope so. Okay. Thank you, sir. On behalf of the organizing committee, I extend my thanks to you, sir. Thank you, sir.
Thank you.
Thank you, sir. I request everyone to gather at the quadrangle for lunch and kindly assemble in the auditorium at 2:30 for the upcoming lead talk. Thank you.
ಕರೀರಿ ತಟ್ಟೆ ಹಿಡ್ಕೊಂಡು ಬಂದು ಇಲ್ಲ ಕುತ್ಕೊಂಡು ಊಟ ಮಾಡಕ್ಕೆ.
Good afternoon, everyone. The upcoming lead talk of the day will be delivered by Professor M. Haridas sir. I call upon Ms. Tanushree M.K. to introduce our lead speaker, Professor M. Haridas sir.
Good afternoon to everyone present here. As the MT of First MSc Department of Microbiology, it's my pleasure to introduce our lead speaker, Dr. M. Haridas sir, FAS, Emeritus Professor, Department of Biotechnology and Microbiology, Dr. Janaki Ammal Campus, Kannur University. M. Haridas sir started his academic pursuits from the Sardar Patel University in Gujarat at the postgraduate level and continues his doctoral and postdoctoral studies in structural biology in AIIMS, New Delhi and Massey University, New Zealand. He was the first biologist to get trained in X-ray crystallography of single crystal proteins. This exposure enabled him to establish the first protein crystallography research group in Kerala. Sir has pioneered the developing of experimental models to establish the theoretical basis in Ayurvedic biology and fermenting Ayurvedic medicines. The fermentation of medicines has been addressed theoretically in Ayurvedic classics. Sir has 100 plus international publications, 30 plus chapters in edited volumes, and 200 patents, 1 international patent, 2 more patents under preparation to his credit. He has guided medicine students for their PhD. Sir has developed curricula for postgraduate diploma in drug discovery and designing, masters in biophysics, biotechnology, and microbiology for Mahatma Gandhi University and Kannur University. He has developed the undergraduate program in biophysics for the University of Calicut. He was able to provide insightful understanding of structural biology, which forms an important content in the frontiers of biology and biotechnology. Sir has been head of the 10 years of Department of Biotechnology and Microbiology, School of Life Sciences, Kannur University. He is the founding and serving honorary director, Inter-University Center of Biosciences, Kannur University. Sir has served in various academic bodies in the tiers of all universities in Kerala. He has also served various research advisory councils, committees, boards of government of Kerala, National Research Centers, medical schools, R&D industries, etc. Currently, he is serving member of Biotechnology Commission Kerala, Biodiversity Board Kerala, Research Council Amrit School of Ayurveda Kollam, Research Committee Arya Vaidya Pharmacy Coimbatore, and Research and Ethical Committee of MVR Cancer Center and Research Institute Calicut. Thank you.
Thank you. Are you hearing me?
Yes, sir.
Okay. I'm extremely thankful to
Hello, sir, you're not audible. Sir, you're not audible.
Hello.
Hello, sir, can you just mute and unmute it again?
I'm not doing it, but it's happening. Are you hearing me now? Yes. Okay. I must confess that, since my time is reduced to 30 minutes or even less, I won't be able to do justice to the talk I thought I would give you, but I will give a succinct abstract sort of I thought I. The important thing, right from the beginning, I'm interested, like many, in anti-inflammatory compounds, because inflammation is, you can say that universal in any pathology, any infection, where the normal biology goes wrong. So, anti-inflammation will have impact on every field of inhibiting inflammation or diseases. That includes cancer and microbial infection or any such pathophysiology. This means, it doesn't mean that the inflammation or anti-inflammation caused by a compound will have the same sort of impact in every disease. The pathways will be different. That means the anti-inflammatory compounds you find or the drugs you find will have different ways, different means to control the pathology. I will invite your attention to the history of some drugs like acetyl salicylic acid. In the beginning, acetyl salicylic acid was antipyretic. It was an extract from a plant, and it was giving reduction in the or control of body temperature. Later in 70s, much after the introduction of acetyl salicylic acid as a drug for antibiotic purpose, later it was found that it is anti-inflammatory. So another aspect of the same compound. So this means, you can have a drug with various functions. So now, in the context of COVID, many people were looking for repurposing drugs. Repurposing means you find a drug, you have a drug, you are looking for the application in another disease. So that means, in the case of COVID-19, you have a new disease, I mean, nobody knew about it. There was no way of controlling it. There was no methodology for treating. So it was a novel disease, and there was no method to control it. So people were thinking of repurposing it, repurposing drugs. So we have been working on. Now let me know how to share. Is it okay? Is it okay, the sharing? Hello? Do you find the slide? Do you find the slide? Okay, I think you find the slide. So now the thing that we have been working on a compound from Artemisia, that is transanethole. And the Artemisia is very famous for controlling, or artemisinin is very famous for controlling malaria. And we got a compound which is anti-inflammatory, which inhibits lipopsinase. And the Artemisia plant extract as such gives many viral control mechanisms as I have given in the slide. We don't have time to discuss in detail, but it shows that this Artemisia extract, water extract, or ethanolic extract gives antiviral property. Okay, now, so what we have found, the compound transanethole, which binds to several proteins of the virus implicated disease, that is COVID-19, many proteins of the virus produced as well as found in the human body. So it is a sort of what we call it a congruent binding. Hello? Congruent binding. That means it has a mechanism of binding to any compound, any molecule, either of the virus or the human body. That means, see, this is a sort of molecular interaction. So any such control mechanisms can be reduced or zero down to the level of intermolecular interactions. So when you have a compound implicating several molecules in the system, that will give a similar type of interaction with any molecule it come in contact with, and this becomes a network. So that is what we have found, that this property can we make use of controlling or whether that would be of use in inhibiting the COVID-19 inhibition. So the enzyme kinetic studies we have done the transanethole purified from the Artemisia plant, then its effect on the virus.
Excuse me.
Not
Your slides are not visible.
hello.
Hello, sir. Your slides are not visible. Please share your slides, sir.
I didn't get you.
Your slides are not here.
Slide is not visible. Can you share from your side?
You go to the option share. Before that, you open the slides in your window, sir. Then you share them. Yes, sir. Now it is visible. Thank you, sir. You can.
Is it okay now?
Yes, sir. You
Okay. Okay. So this is what we have on in silico method only. So the transanethole.
Screen, sir.
Is it okay now? Hello.
So you click on the slideshow, sir. In that PPT. That one, sir.
Is it okay?
At the bottom, sir.
Is it coming?
No, it is. Sir, at the bottom, you click on the slideshow. That one, sir. Yes.
Is it okay?
Yes, sir. Okay, thank you.
Okay, then, okay. So the SARS coronavirus in vitro inhibition was shown by Artemisia species. So what is this plant is not the one from which the artemisinin was purified, which is relevant in the case of malaria. I'm not going to the details of this work, but only to say that the conjugate binding of transanethol is important, which is relevant for the SARS coronavirus 2, that's COVID-19 protein, as well as angiotensin converting enzyme of the human body, which is responsible or which is infection. So this is about the lipoxygenase inhibition shown by the transanethol. Anyway, that's not important now. We need not discuss this aspect. So this is another study what it shows how the lipoxygenase enzyme is inhibited by the transanethol, which is relevant in the case of any infection where the inflammation is involved. And inflammation to many is extremely found in the case of COVID-19 infection. So this is yet another thing. When we talk about the drug molecules, if you are familiar with the Ramachandran plot, the phi-psi angle plotted on a picture like this, which says about the integrity of protein structure, whether how good the protein structure, whether it is a natural thing, which is calculated based on certain parameters of the protein. So this is similarly. You find here x-axis and y-axis two parameters of any drug molecule. That is, what this on the x-axis is polar surface area of the compound, and this is about the lipophilicity, y-axis. So this means if this is within this area, but the white area is about the gut absorption of the drug molecule, and the yellow area is the blood-brain barrier. So if it falls within this area, it means that any compound you find out from natural sources will have a high probability that it would be good to be a drug with some minor modification. So that means there is, you might be familiar with the Lipinski's rule of 5 and such other things. So this is quite visual effect. In this graph, you get that any molecule, whether this is about the transanethol molecule, that falls within the yellow is, it will permeable blood-brain barrier, and the white area is the absorption of the gut, human gut. So what in the gray area, if it falls, it will not be good as a drug which can be already administered because it may not be absorbed by the gut. So this is a very simple way, but in this calculation, many thousands of molecules have been used, and it has been found that almost all the druggable molecules occupy in this area. White area means gut absorption. Yellow area means blood-brain barrier permeability. So this is about the LOX binding with the transanethol. Transanethol it blocks LOX activity. So I don't go into such details. So this is about transanethol, another molecule enzyme implicated in the brain or inflammation pathway, that's phospholipase A2. So similarly, it also blocks more than one enzyme. So the increased cytokine, now coming back to the COVID-19, increased cytokine storm results in increased production of cytosolic PLA2, which links with COVID-19. Cytosolic PLA2 has a specific role in COVID-19. pathogenesis. So we have found this compound inhibits PLA2 as well as LOX, which comes later in the anti-inflammation pathway. COVID-19 also affects neural system where the, this is another aspect, loss of taste and smell is experienced, and one main symptom of COVID-19 infection. So this is a fair chance of reducing that impact of losing neuronal sensing mechanisms. The leukotriene is also responsible for inflammatory responses in patients with progressing COVID-19. So there is a chance that, you have a probable candidate here. We look into these aspects with some other candidates in a different context. So this is about the transanethole. And I have to talk a huge amount of other things that are here because this is about the fatty acid, linolenic acid binding site of spike protein of COVID-19. So I think, if I go to that, my time won't be enough. So this is a protein, the spike protein is the most important protein which binds to the ACE2, angiotensin converting enzyme of the mucosal membrane, which attaches the virus particle and invaginates into the cell and the infection carries on. So what happens, the spike protein, if the spike protein is blocked by some means. So it has been found that linolenic acid, this is a serendipitous discovery. Linolenic acid is a fatty acid which binds to the spike protein of this virus. So this virus, if it is having the linolenic acid or anything at the spike protein binding domain to the acetylcholine esterase of the host mucosal membrane, what happens that the virus will not get attached. So if there is enough linolenic acid in the vicinity of the angiotensin converting enzyme 2, the virus won't be able to penetrate the mucosal membrane and enter into the system. So that's a good knowledge that you have a candidate which can be used to control or inhibit or regulate the viral infection. So this is what we have found. This is about, I'm talking about a few things from various places and putting them together for developing an idea. So I'm talking about an Ayurvedic plastic, Yoga Ratnakara, which deals with treating various fevers. In that, that one is Sannibala Jwara, is very high fever. So what happens that this classical text has given a formula with ginger and citrus medica, sidrat. I don't know what you call it in Kannada. In Malayalam, we call it Ganapati Narakam, something citrus fruit with the shape. So anyway, this term may be more scientific. So you can use either the common English sidrat or citrus medica, and gingival apixnale, use of both things, rock salt and some other salt. So if this is used as a nasal cleaning or nasya, nasal cleaning, that is very good for Sannibala Jwara, not for COVID-19, because nobody knows about COVID-19 at that time. It was only Sannibala Jwara which has symptoms quite similar to COVID-19. So we are approaching to treat the symptoms, then this may be good. In that case, this recipe would work or may work in the case of COVID-19 infection also. So what we have done here, we picked the compounds, the chemicals, the phytochemicals present in this Ayurvedic formulation and try to find how it interacts with various proteins implicated and involved in the COVID-19 infection. Also, we have analyzed the same way various mutant proteins of COVID-19, especially COVID-19 spike protein, as well as the human acetylcholinesterase 2 and MPRO protein, and all variants of COVID-19 available till date, including Omicron. And the last one we haven't tried, I mean, I don't know what will come up, but what is so far available in literature, the last variant is Omicron. So we have found all these variants would be inhibited and inhibited by the compounds present in this Ayurvedic formula. So it means that the nasya, the preparation from citrus medica and gingiber officinale with rock salt and other salts as a nasal application or for gargling, or even consumption is also advised in certain instances. So what may happen, so if this is given as a nasal applicant, the compounds present in this would be blocking the acetylcholinesterase 2 and the virus entering the spike protein of the virus entering, that is the virus of the COVID-19 SARS coronavirus 2. So if both these are inhibited, there won't be infection further. But the thing that, I mean, this is not, I mean, you cannot say that this is, there is no immune system involved. So the information or the type of application will not give a lasting impact. So, I mean, it needs to be given every day, or, I mean, similar sort of thing. So only in case of epidemics and other things, such a system may work. But there are clinical data also with us that this is working wonderful. I'm not presenting the clinical data, but I'm presenting the in silico data in the coming slides. This is mechanism of infection. So I think I don't have time to discuss this further. So I will quickly come to the later aspects. So how angiotensin converting enzyme works and what is its role, how the spike protein interacts. I mean, all these things are important, but we don't, due to the lack of time, I'm not going to this aspect in detail. So this is about some differences between COVID-19 in form of Omicron. So how this Omicron is different from which has been found to be very potent compared to other variants of SARS coronavirus 2. So I'm not going to discuss this also. However, this is equally the compounds would be equally powerful or potent to inhibit the Omicron also. So here in this slide I have given what are the different variants, or how the Omicron variant of COVID-19 SARS coronavirus 2 is different from the first and the UK variant, the starting one. And many other variants. So there are many variants of SARS coronavirus 2. From UK variant to the Omicron, all these are equally inhibited or would be equally inhibited by the compounds present in the Ayurvedic formulation, which are shown in the previous slide, extracted from the classic Ayurvedic classic Yoga Ratnagari. So now, I mean, what we have done, this is, I mean, the last sentence you may find it. The Samhita also advises application of nasal drops by a healthy person before going out in one's house, in public places. This is the regular advice from Shristha to everyone that if you clean your nasal tract, some sort of nasal cleaning and some sort of medical application, as in the Yoga Ratnakara recipe. So that gives you or protect you from various infections. So it is not in the present days that such infections might have been there millennia ago. That's why Sri Shuddha Samhita says that one should be applying nasal drops before going out. So anyway, I'm not going into details, but I'm going to show you some important findings. See, all these are compounds, phytocompounds, taken from the recipe, that is cedrat and ginger, and these are the phytochemicals, and how these are interacting with the COVID-19 spike protein. That is what these are the interaction data, and further, how they interact and what are the binding energy and such other things which are relevant to consider whether these are important or not. So now, this is how it looks like. In the case of these are the dot pictures of phytocompounds onto the receptor binding site of the Coxsackie virus to spike glycoprotein. So now this part, which is shown in this oval pink structure, that how it is binding, how it is sitting, and it will inhibit that occupation of any of these compounds at the spike protein, will inhibit the virus to interact with the ACE2, acetylcholinesterase, and it won't be, it will make, or rather it will make, it will render the virus deficient or inefficient to enter into the mucosal cell, and it will not be infecting the host further. So this is how the mechanism, and the earlier thing what I talked about, the transenethol, is the same way it interacts with, I haven't shown here how it interacts with, but later I will come to it, the transenethol. The phytocompounds of the Omicron, anyway, I think it is also interacting with Omicron similarly. This is about acetylcholinesterase 2 ligand. So one side I talked about the spike protein, that is of the virus. On the other side, I'm talking about the host membrane protein, which is responsible for intake of the virus. So the acetylcholinesterase may be having some other function, but it is interacting with the virus, so that the virus is attached to this enzyme. This protein is a membrane-bound protein. It is present on the mucosal membrane surface, and this protein will get attached to the virus. So that is the area of the spike protein area, or the receptor binding domain will get attached to the acetylcholinesterase protein, the enzyme. So this is, in fact, the similarity, complementarity of these two structures. There is no design of the virus to get attached to the acetylcholinesterase prior to anything else. It is the, or we can say that it's an accident of the nature, or the nature. If it is complementary, it will bind. If it is not complementary, or if it doesn't get attached, then there is no infection. So if there is an infection, you have something similar in you which helps infection. So that is what the virus is getting entry into the human body, because it finds something which is quite complementary to it. So the mutual interaction of the two molecules, one from the virus produced virus protein structure, and the mucosal membrane protein, acetylcholinesterase. So these two are held together. So that makes the enzyme to go inside the flip of the cell membrane, and the viral protein or the virus is getting inside the cell. So that's how the inhibition, the infection happens. So anything which blocks this binding, the virus to the acetylcholinesterase 2, that will render effective or render the function of an effective inhibitor of COVID-19 or any virus in a similar mechanism. So this is about the acetylcholine esterase and the compounds interaction. So one from the virus side and the other from the host side. So this is again, binding energy in such other parameters. So this is how it looks like, how it binds to acetylcholinesterase, all these compounds. So this is about the Omicron. The similar thing in the Omicron. So now, see, this is the important. Interaction average data of 8 compounds such as medicated fever, officinale, dots against the spike protein of COVID-19, Omicron, where Omicron variant and the first form of COVID-19. It is quite comparable. That means whatever binds, whatever controls the first form of COVID-19 also would control Omicron, and probably any other variant comes in the future. That's more important because there are many other, there is a trail of, or a train of variants from the first to the Omicron. So all these are, I haven't presented the data, all these are probably inhibited or possibly inhibited by these compounds, quite like the first one to the Omicron. So this shows that in future also, or whatever, if any other variant comes, variant of SARS coronavirus 2 comes, that also would be effectively inhibited by such compounds. I'm not saying that these can straight away go as a drug, but these are probable and possible candidates, which shows that, as I have shown that boiled egg like picture, which I compared with the Ramachandran plot of the protein structure, x-axis with the lipophilicity, the polar surface area, and y-axis with the lipophilicity. All these come inside the that orange will be Lipinski's rule like, or with this boiled egg picture, graph like boiled egg graph like picture will be beautiful drug candidates. So these are the hypertonic, and these are other aspects of the Ayurvedic formulation, which I'm not going to discuss here. So now, this we have published already, but of course, limitations of this study, there is still danger. In silico evidence needs substantiated in vitro. Before human studies, population has to be standardized. As formulation contains, almost these have been done, and we have clinical data. The formulation study contains ingredients like ginger and possible minerals. Delicate nasal epithelia must be kept in mind, optimizing the dosage. The time-tested formulation nasal cleaning could play an important role in mitigating the contagiousness of SARS coronavirus 2. This nasal purge may be more relevant in people with high risk exposure. Not everyone need to undergo this sort of a nasal purge, but maybe in high risk exposure, those who are in hospitals and those who are in direct contact with the patients, such other persons who are coming under high risk or who are having, who are put to high risk exposure. So the conventional modern medical approach would be to find out best of all these things. You may have the one, maybe the best, but that interact with COVID-relevant protein for the drug development. Why not a cocktail? This is what the Ayurveda suggests. That you can have, not necessarily that you can have only one drug. You can have a cocktail, you can have a mixture. That is what the Ayurveda is. So the outcome, the use of an ancient time tested formulation in COVID-19, administration of this, especially relevant people with high risk exposure, it could be helpful in asymmetric patients with mild symptoms, because these are mild things, so they won't be harm. So, as demonstrated in silico, most major chemicals found in citrate, ginger, will be binding to the receptor binding domain of COVID-19. So like this compound, the initial introductory compound also, transaminase, would also with a fatty acid, linolenic acid, which I haven't discussed here, would be also helpful. So the Ayurvedic formulation mentioned in the Yoga Ratnatara could be modified, could be refined using all these information with conventional modern medical methodology. What we may need, even if we are constrained by amount of applied drug, the same for the best single compound or a cocktail. What would be the best? It's a question we may have to, or we would be looking at. And it's a significant question. The present identified 14 compounds. So this is again what in a conclusion. I have said that here comes the analysis network. So if this all these compounds could be analyzed by the network pharmacology method, it can lead to polypharmacology, and how it works in the bio-network. So all these, there are various application softwares in all these areas. So now things have become slightly better, that the better way of drug formulation, apart from the single drug format, can be found out using all these techniques: network pharmacology, polypharmacology, and bionetwork, all using in the biological or using the computer networking system, powerful computing. So this is my message, that we need to look back into our Ayurvedic system vigorously, and we will have better results. Thank you all.
Thank you, Professor Haridas, for a very wonderful and a very, very exhaustive lecture on COVID-19 variant tried with Ayurvedic formulations. Can you hear me, sir?
Yes, yes.
Wonderful. We can have one or two questions. Is it okay for you?
Yes, I would love to. I would love it.
Actually, we are running short of time, so most of the participants are interested to interact with you. One or two questions.
So I would love to answer any question for which I have answered.
The participants are a little bit shy. In addition to that, they are actually waiting for a valedictory also. Just I wanted to know to what stage of the actual clinical trial you are.
Yeah, the clinical trials, I'm not empowered to do it, but a private hospital near Kottayam, as well as one at Kannur, with some variant modifications. They have tried, I have data that shows that it is the infection of the COVID-19 among the high risk group is totally reduced, I mean, completely eradicated, I would say, if they had applied this nasal purge, nasal application. So but I am not, I mean, able to present the data here, but this is the result that, I mean, this is very effectively controlling the infection among the high risk groups.
You check the cytokine storm also, or only the antiviral activity?
No, I mean, I don't know. I have got your question correctly because it said there was a clarity problem in the sound. But these are not, I mean, these preparations are as in the classical text, not in a modern way. It was prepared as in the classical text.
Okay. Then thank you very much. I take this opportunity to thank Professor Haridas on the behalf of.
Already there is a product in the market where we have been given the scientific evidence. So that is there. That's available in Kerala. I mean, it is. I mean, in that way, it's not a new thing, but it is made for a specific nasal application, as described in the classical text Yogaratnagara. So there was no need of any involvement or specific approval from the drug controller in such other things, because it was an Ayurvedic product.
Sir, good afternoon, sir.
Good afternoon.
Question from me. Actually, in the COVID-19, we have seen the symptomatic, mild symptomatic as well as the severe symptomatic cases. As I'm working as a microbiologist, I just want to know that in your trial, how you find out whether it was a asymptomatic case has been identified? To find out in.
But see, what I mean, we have we have to do more analysis. But what we have done, we have tested RT-PCR. Okay, RT-PCR before any application of these sort of nasal punches, nasal applicants, and we have periodically, when the course, when the test will go for 3 months, we have again conducted RT-PCR and ruled out whether they had infection or they didn't have infection. So it is, I mean, the checking whether the infection was there or not, or they got it, it was based on the RT-PCR.
Thank you, Dr. Sashan, for your presentation. Thank you, sir.
Thank you. Thank you. You are welcome.
Now we concluded all the lead lectures of the 2 days national conference, and we are going to start quickly with the oral presentation. Again, as far as the presentation time is concerned, 3 minutes for the presentation and 1 minute. 3 plus 1 is 3 minutes for presentation and 1 minute in other words. Yes. So as soon as we press the bell, you have to stop. Means we should consider that the time is over. I request all the audience to be here only.
I request all the poster presenters to display your posters and start presentation. No. Oral presentation, Mr. Abhishek.
Very good afternoon, all. Here I am presenting essential oil encapsulated zeolite imidazole framework, an effective photocatalytic antimicrobial agent against multi-drug resistant non-typhoidal salmonella species. These are my contents. Here, metal organic framework is a promising material. It's constructed using metal clusters and organic linkers. Its small size and high surface area volume and zeolite imidazole framework are one of the subclass of MOF with zeolite topology. These are metal organic framework and ZAF8 structure. It's highly stable in chemically stable, and MOF has been explored a variety of uses, including drug delivery, biosensor, bioimaging, catalyst, gas storage, separation, and absorption due to its tunable nature. ZAF8 can transfer therapeutic drug into bacterial infection site antimicrobial therapy. Citral is the main component of citrus fruit peel oil. It's especially found in orange peel. Citrus mixture of two compounds. There is geraniol and neral. According to WHO, is a high priority pathogen is non-typhoidal salmonella. This salmonella is a key causative agent of diarrheal disease, is huge medical intervention expenses and loss of productivity. WHO. Non-typhoidal salmonella is in worldwide. Non-typhoidal salmonella is causing in Africa is higher mortality. After that, in Asia, and this is my methodology. Here, synthesis of ZAF8 and characterization and application. These are results. Here, UV DRS spectroscopy. Here is confirmed synthesis of ZAF8. This is a FPR spectra. You can see that top one spectra is of drug-loaded ZAF8, and which one is first one is without drug. So it is masking the peak. So this is XRD pattern, and here it's confirmed it's a sodalite structure with a demeasuring formula. We can understand 36 nanometer is size. And this is structure. It's like everything is uniform size cubic structure. And it's in vitro drug-based kinetics performed in stimulated interstitial fluid. It's a drug releasing study. It's a dialysis membrane. In vitro hemolytic study, and even for 100 microgram is showing like less than 5% of hemolysis. So photocatalytic activity is performed against two dyes, and methylene blue and rhodamine B. And here in methylene blue it shows more degradation. This photocatalytic activities, drug docking studies with the citral and protein dispersin and osmoporin C. This summarizing the drug. Here is PCR validation of Salmonella enteritis and Salmonella typhimurium. These are the primers which used, and these are antibiotic susceptibility tests, and most of the drugs, these cultures are resistant to these drugs. And MIC, most of the 15 MIC, and MBC dose is 32. These are my conclusion. We have successfully synthesized modified ZAF8 nanostructure employing two different metal clusters. Formation of ZAF was confirmed by UV, DRS, FER, XRD, SEM analysis. Acknowledged. Actually, Salmonella 2, is a diarrhea. Yes. Salmonellosis is It's another type of disease. This is non-typhoidal salmonellosis.
Drug resistant and non-drug resistant difference in nanoparticle.
Most of the drug resistant and non-drug resistant shows minimum inhibitory concentration almost equivalent to equal. Sir, actually, I'm working on both E. coli and most drug resistance. So this docking that proteins are similar, even E. coli and Salmonella, that membrane proteins. So it's taken back to.
Thank you, Mr. Abhishek. Next, Basuraj M.
Good afternoon to everyone. It's my pleasure to introduce myself. My name is Basuraj. I'm a research scholar at Mangalore University Host Department. Today, I'm presenting as part of my PhD research, and the topic is phytochemical profile and antimicrobial activity of medicinal plants against selected multi-drug resistant superbugs. Multi-drug resistant superbugs are one of the major global health concerns in the 21st century, posing an ever-increasing threat to humans, animal health, and food security. Nowadays, bacteria becoming emerging as multi-drug, drug resistance to common antibiotics, and sometimes it is impossible to treat the infections. Particularly, WHO listed VID pathogens has shown challenge to the scientific community, particularly the gram-negative pathogens. Doctors do not have a new treatment against gram-negative pathogens in the last 50 years, and not even effective drug not even entered since 2010. Experts estimate that some 10 million people will die every year by 2050 due to antimicrobial resistance and multi-drug resistance. This is the evolution of antibiotic resistance. After serendipitous discovery in the 1928 of Alexander Fleming penicillin G, he has noticed that in his local lecture in the 1945, some colonies were popped up in his research, in vitro research in his lab. After 24 hours of in vitro study. Medicinal plants are endowed with rich antibiotics, medicinal plants are endowed with rich phytoconstituents. According to WHO, 85% of population rely on medicinal plants and some of the traditional medicine. More than 80% of the synthetic drugs derived from them. Moreover, herbal and traditional medicine has gained cumulative popularity in today's medical practice. The increase in the herbal medicine products due to their cultural acceptability, availability, affordability, efficacy, and less taxation since from ancient times. Due to the lack of rigorous investigation, the phytochemical constituents and pharmacological effects of many medicinal remain unidentified to determine the efficacy and safety. The present study aims to investigate phytochemical screening and evaluate the antimicrobial activity of Phyllanthus meluri and Datura Swaminium against multi-drug resistant superbugs. To explore the phytochemical of food extract of Phyllanthus meluri and the Datura by succinate extraction method followed by LCMS and SPLC. I will explore these are all the secondary metabolites along with other major constituents. Ethyl acetate and methanoic extract of Datura and Phyllanthus showed varying degrees of antimicrobial activities against selected superbugs. However, there is a need for further studies to adequately determine the safety and clinical efficacy of these drug-resistant superbugs. Antibiotics are a precious resource. Thank you.
Please telling antibiotics are precious. Use it as they care. What was the last sentence? What? Last sentence.
Antibiotics are precious resource in the golden era of antibiotics. Now, these antibiotics are resistant to multiple bugs. Particularly gram-negative infection, E. coli, Salmonella typhi, methicillin-resistant Staphylococcus aureus, Acinetobacter, and Aspergillus niger and Candida albicans of fungal species. These are highly resistant superbugs. Yes, sir. 15 micrograms. Sorry, sir?
What is the mode of action?
Sorry, sir?
Mode of action of the medicinal plant against the drug resistant bacteria.
I didn't get you, sir.
Mechanism of action that will create the growth of a drug resistant bacteria. Is it the cell wall inhibition or the metabolic activity inhibition?
Cell wall inhibition.
Okay, thank you.
Thank you, sir.
Thank you, Mr. Basiraj. Next, Mr. Ganesh Prasad.
Hello. Good evening. I'm here to present a part of my research work, that is analysis of gut microbial diversity of two closely related honeybee species. As we all know, the study of gut microbiota is rapidly rapidly growing field of research, and the impact of the gut microbiota on human health is predominantly prevalent in biomedicine, and the association of gut microbiota and diseases with the various diseases is already very beautifully explained by Dr. Alani sir in his presentation. We considered the honeybees as a research model, since a recent decline in the honeybee population has put a spotlight on the potential agents that cause that affect their health. Here, as we all know, the honeybee is a highly valued insect throughout the world, not only for their pollination, not only for their honey production, but also a great importance to humans and ecosystem as a pollinator of many economically important crops and wild flora. Recent developments, which include the discovery of extensive strain level variation, protective and nutritive function, has called a comprehensive study across the globe to explore the gut microbiota of various species of honeybees. Then these are the methodology we followed. We started with a sample collection. We collected reared habitat from rear habitat, that is Apis arana indica, and wild habitat species, that is Apis florea, followed by the genomic DNA extraction using QIAGEN tissue kit, then amplicon sequencing of the bacterial 16S RNA genes by targeting the V4 region, and gene applicants was performed by following the protocols of Illumina
followed by the bioinformatics analysis using KIME pipeline and against the flash tool, followed by the functional imputation, that is using the PyCrust tool against the tech database. The obtained results have been submitted to NCBI database, which are available in the following screen. These are the PCR amplified products of 16S RNA genes. Then here, alpha diversity estimation, the Zenon index indicates that the Apis cerana indica has more diversity compared to Apis florea. And they shared 36.3% OTUs. The phylum Proteobacteria has dominated the gut of Apis florea as well as Apis cerana indica, followed by Firmicutes. Coming to general level classification, here Lactobacillus and Bifidobacterium are involved in the fermentation of various carbohydrates, and it has antibacterial potential. Recently, very recent discoveries shown that these bacteria has antibacterial potential against CCD, that is colony collapse disorder of uneven colony, and AFB, American Folbert disease, and Margonella and Rhodococcus prevotella. These are the firstly reported genera. Metabolic functional imputation suggests that they shared metabolism related gene families prevalently. This is just an expansion of each functional modules. And concluding, the gut bacteria of Serrana indica was more diverse than that of the Apis florea. The observed variation in bacterial genomic diversity among these critical pollinators may have been influenced by the apiary management techniques, ecological adaptation, or habitat size. These are the publications we included culturable diversity for the Apis florea, and this very comparative studies under review. Thank you.
People are clapping a lot for your presentation. It's a novelty.
Apis florea is very least explored, this one, honeybee species. Compared to Apis mellifera, it's a western bee.
You tell me with reference to what is the university in that microbiota or it is
Sorry? And human being, yes, sir. Yeah, is there similarities are there, recent findings, but I am not going to do that as a part of my research work, but we are studying the microbial carbohydrate degrading enzymes from the gut.
Mr. Karthik Veliappa.
Try to go directly.
A very good evening, respected judges and my dear colleagues. So, it is a basic topic. I no need to explain about the endophytes. So, all had been explored till now about the medicinal plants and other things. But now I am going to focus on the bamboo. That is also a particularly a bamboo shoot.
is not much exposed about their endophytes of the bamboo shoots. They had exposed the bacteria and fungi from bamboo seeds, which it will take around 12 years. So these bamboo shoots are grown in seasonal. So this is the right time, that is rainy season, in the spring season, where bamboo shoots are grown. So it is an edible vegetable and it should be a processed one. So it is all about the endophytes. So here I had isolated 5 bacteria and 5 fungi, which are more prominent and effective. It shows antimicrobial properties. So this is my sampling area. I had taken Karnataka state as my sampling area. From there, I had selected few districts, that is around 10, Kodagu, Dakshina Kannada, Shivamogga, Hasana, Bengaluru, Mysore, Chamarajanagar. And almost it covers, it belongs to the Western Ghats. So here you can see the green colored one where I had mentioned the sample collection points. So depending upon the climatic condition and the soil condition, it is divided and I had classified it. And there are around 12 species of bamboos which are edible. There are around 125 species in India, and in that I had selected the species available in Karnataka, and here Dendrocalamus aspera, Stockley, Newtons, and Bambusa bamboos. So many species are available for edible. So this is the habitat, how it is grown, the length, whether it is thorny, and what type of soil condition it grows. So this is the table where I had got the species from different places. So these are the isolates which I had got. Around 56 of bacteria isolates and 37 of fungal isolates I had been isolated. So in that, so before isolating, I had given a clear picture of the shoots where I had taken the samples. So you can see the whole bamboo shoot, and it is the comset, that is which I had not taken that part. The middle part of this bamboo shoots had been taken. So this is processing in the laboratory. I had isolated, viewed, and so this is the antimicrobial property of the few bacteria which I had got, and I had got the nice zone of inhibition which has been focused. And this is the SEM images of those bacteria, and I had done the molecular analysis by sequencing. For bacteria 16S RNA, and for fungi it is 18S RNA. And these are the results. Phylogenetic tree has been constructed. And so here I had got the new accession number of the sequence obtained from our bacteria. And the main 5 are the Bacillus lignoformis, Bacillus phalanginesis, and Bacillus subtilis. And Aspergillus niger, Fusarium solani. So these are the things which I had. So bamboos are rich in fibers, and many of the states does not know about the bamboo shoots, and they had not eaten also, I think so. So I had showed the picture of the bamboo shoots, and here it is a very common dish. That's it. So it is not explored for more. So I had been selected as that shoot sample. So let us see. And the always endophytes are associated to the plant, and the shoot which grows within a week, it will be forming a bamboo plant, whole plant. So mainly which I had isolated the bacteria. After review of literature, I came to know that they are producing some of the growth promoting compounds, that is bioactive compounds. So I am later going to expose on those bioactive compounds. That is my research work. This ongoing process. Yes, sir.
Presentation is over.
Dendrocalamus asper. Dendrocalamus asper. Bambusa bambus. Yes, there are many. Bambusa tool also there, nutrients also there, Stoxy is there, Strictus is there.
What is the method you use for determining antibacterial activity?
Agarwal diffusion method.
What was the positive number?
I had taken the retrieved from some injections.
Injection directly.
Let me see. Negative control, I had taken the only the media product.
Very good. Next.
Thank you, everyone.
Karthik LP.
Myself, Yalpi Karthik, research scholar, Department of Microbiology, Mangalore University, under the guidance of Dr. Vaikya Mandula. I'm here with presenting my topic, that is production of biologically important molecules from Streptomyces historicus in the mangrove soil from the Mangalore region. So we all know that the mangroves are less exposed to the Indian region. So with that, the actinomycetes are well known for the production of many of the antibiotics which are commercially available. The bulk share of the antibiotics, more than 70% of our antibiotics presently available are synthesized only from Streptomyces group. And majority of the literature studies is only focused on the production of secondary metabolites and characterization of their secondary metabolites and the production of antibiotic powders. But there are only very, very few literatures are available on the proteins explored from the actinomycetes. So as a result of that, we have been focused on the characterization of peptides from these actinomycetes groups, that is Streptomyces. This is the general life cycle of our Streptomyces group, and here we see that mycelia, once it is mature growth, then it will start producing the different colors of spores, and these spores again the life cycle reverts back. And this is our workflow which the work has been carried out. These are the four samples. The mangroves of Mangalore region, which we have carried out our work. The soil samples were collected from there. And in that, out of 4 samples, we have collected 18 distinct colonies of actinomycetes. And in that, the ANTB YKMU4 has shown that efficient activity. So we are focusing on this culture now. This is the culture view in the media plate and as well as in the inverted phase contrast microscope and the mycelia views under the PESM microscopy. Here are the spores, how it is visualized beautifully. And the presence of amylase component or enzyme in this isolate, by the help of the protein production, we have could able to know that through the biochemical characterization, that is the hydrolyzing enzyme activity, there the presence of amylase enzyme can be seen here. And this culture through 16S rRNA gene sequencing identified as Streptomyces historicus, and we have received the accession number from the GenBank, that is the MW 785875.1, and it is showing that majority of the species, that is Streptomyces historicus only. And we have been used the extraction of protein through the acetone precipitation method, and later we have been done some chromatographic techniques. And here, this is the size exclusion chromatography with the use of G10. And here the antibiotic activity has been shown, antibacterial activity has been shown by the culture by ANTB YKMU4 on different pathogens. And this is the plates which can show that the antibacterial activity. And it is just comparison with our whatever the protein extracted to the available present antibiotics, that is streptomycin efficiency. And these, whatever the protein extract, we have been used for characterization, that is the LC-MS. There, it has shown that the triprostatin, surfactant B, and fuminosin, as well as microcystin. So these are the compounds is used. This is the one used in genetic studies, and the surfactant is used for the targeted cell activity like cytotoxicity. Okay, because it is a surfactant, it is directly can penetrate through the cell membrane. And this is fuminosin. It is the one of the mycotoxin, and it is used in for the immunological techniques in the case of ELISA, where we can replace the synthetic peptide, that is CT452. And this is the one microcystin. It is also a compound which is helpful for the antimitotic activity. Concluding my topic, that is, as we all know that the majority of the antibiotics are produced from the Streptomyces group, and these are the one key role for the leading biological society, and these can be used as the antimicrobial peptides because they are showing antibacterial activity. This is one of the culture score, actually. Thank you.
Very active volunteering.
Thank you, sir. Looks like that, sir, but it is. Sir? Yes, sir. Yes, sir. Like we have been doing the in vitro studies now. We have been just characterizing that. Yes, sir. It has shown only amylase activity. Based on the antimicrobial activity, sir, compared to other isolates, it has shown majority of the like a broad spectrum, like both gram-positive as well as gram-negative bacteria. No, sir. It is quite bigger. It is not visible here. Yeah. This is the S17 culture that has been mentioned here, sir. That is YKMU4 for our convenience. And compared to this is the Proteus vulgaris, and here the salmonella typhi also. It is showing quite efficient activity compared to the other isolates. Sir, actually, it is looks like it is there. Efficient. What is that? The zone of inhibition is there, but it is not clearly visible in this photograph. Actually. I'm not sure. MRSA has you can listen to it. Yeah. Thank you, everyone.
Sushma says.
A very good evening to everyone present here. Myself, Sushma, under the guidance of Professor M. Jaishankar, and my topic is extracellular enzyme potential of endophytic fungal isolates from the medicinal plants of Biligirangana Hills, Karnataka. So the medicinal plants, as we all know, medicinal plants is a very important raw material for the to cure many of the infectious diseases, and many of the drugs are derived from the medicinal plants. So the endophytes here refers to the microorganisms, maybe it's a bacteria or fungi, colonizing the intracellular living tissues of the plant tissues, healthy plant tissues. So whose presence it will be the asymptomatic. And one of the anti-cancerous drug, anti-cancer drug, Taxol, was identified by Physiolophus microspora, an endophytic fungus from the Taxus valicidia. And these are the materials and methods: collection of sample and isolation of endophytic fungi, and antibacterial activity. So these are the materials and methods. Isolation of endophytic fungi from medicinal plants. According to the reference, I have performed the isolation of endophytic fungi from the medicinal plant and identification of fungal sample by staining methods, by the standard manual, and antibacterial activity and the test against human pathogens with Staphylococcus aureus, Bacillus subtilis, Escherichia coli, Pseudomonas aeruginosa, that is collected from the MPCC microbial type culture collection, and antibacterial activity was performed by agar-bell diffusion method, and the extracellular enzyme activity, that was gelatinase test and the DNA lipase, pectidase, chitinase, proteinase, amylase test. These are the extracellular enzyme activity, and molecular identification also was performed, and by PCR amplification by 18S RNA, that was the fungal isolate, and phylogenetic analysis. These are the plants isolated, endophytic fungi was isolated, and this is the leaf bit sample emerging, the fungal isolate, and some of the pure cultures from this plates. Also, antibacterial activity was done. So this was identified as Alternatia brassicae, and A7, this is the code, or Aspergillus flavus, Penicillium citrinum, Stempalium lycophysici. These are the endophytic fungal isolates were tested against the antibacterial activity. And this is the extracellular enzyme activity was performed. Gelatinase test, this is the positive result showing the enzyme activity. And chitinase test, tyrosinase, protease, amylase test. And this is the antibacterial activity showing the zone of inhibition and the according to the enzyme. This is the antibacterial, the molecular screening of the isolate, and one of the sample which is more effective that was taken, and it was screened for the identification, and this was the phylogenetic tree and the sequence based on that. So organism was identified as Stempalium lycophysici, that was submitted to the gene bank and got the accession number as MB375406.
What was the media in?
Saccharose, dextrose, agar. PDA, media.
Endophytic fungi, they differ?
No, sir. Endophytic fungi are isolated from the plant sample. So we are isolated from the plant sample. Antimicrobial. Antibacterial, I will show.
What is the nature?
Antibacterial, which provides, which inhibits the bacterial.
Did you characterize the antibacterial substance?
Yes, sir.
By study the property.
As a crude extract I have done.
Right now you did.
Yes. From crude extract I isolated, and the fungal sample were treated with antibacterial.
What is the biological significance of endophytic fungi?
It promotes the growth, and it stress condition. It inhibits the stress condition.
Thank you.
Gautami GA. Good evening, everyone. Myself Gautami GA, research scholar, under the guidance of Dr. G.S. Gunashri ma'am. And the topic is optimization of carbon and nitrogen sources for production of exopolysaccharide lipids from the Rhodotorula minuta. These are the table of contents. Microorganisms such as bacteria, fungi, microalgae, and yeasts can accumulate large amounts of lipids intracellularly. Microbial lipids produced by oleogenous microorganisms are being utilized as an alternative or a traditional oil sources. Yeast can accumulate lipids up to 20% of their
20% of their biomass. They are grown in controlled conditions are called as oleogenous yeasts. Almost 75% of the dry cell weight of the yeast cell wall is comprised of polysaccharides. Yeast belonging to the genera Candida, Cryptococcus, Rhodotorula, Hansenuella, and Sporobolomyces produces EPS such as glucan, galactomannan, and phosphomannan. The EPS produced by yeast is more easily separated from the culture broth than those produced by bacteria. Hence, it is used as a large-scale production. Methodology: extraction of total lipids and EPS from the Rhodotorula minuta. Rhodotorula minuta was inoculated in a 100 ml culture broth of MYE media broth for 5 to 7 days at room temperature. After that, after incubation, centrifuge at 6000 rpm for 20 minutes at 4 degrees Celsius. Separate the cell pellets and supernatant. The pellets were used for the extraction of lipids, and supernatant was used as extraction of EPS. For lipid production, the cell pellets were extracted by Soxhlet apparatus by using solvent petroleum ether. And for EPS extraction, the supernatant was collected by 2 volume of the cold isopropanol was added and incubated for overnight at 4 degrees Celsius. After that, the precipitated EPS was centrifuged and washed with cold acetone twice and dried and weighed. Result in this graph shows the total lipid content extracted by Rhodotorula minuta in the MYE medium. Figure A shows in this I'm using the different carbon sources. Xylose shows the more accumulate of lipid production, and in figure B shows the potassium nitrate, urea, and ammonium sulfate shows the more production of lipid. Figure 2 shows the exopolysaccharide extraction from MYE medium from Rhodotorula minuta. In this figure A shows the extraction of EPS. Fructose shows the more amount of EPS production in the carbon sources. So in this figure B shows the extraction of exopolysaccharide from different nitrogen sources. In that KNO3 shows the more amount of EPS production. In this result, EPS production, carbon source is fructose is the suitable sugar for the production of exopolysaccharide, and the KNO3 is more suitable for production of lipid and exopolysaccharide from Rhodotorula minuta. These are references. Thank you.
We also is very active volunteer in the country. What is CL ratio? CL ratio.
Now that one has to do changing the carbon sources. For basic media, I'm using glucose. Other than other carbon sources and nitrogen sources, I will change.
But for how much percent carbon you have EPS production? 1, 2, 3, 4, 5, 6, 7.
4%.
4. And for lipid?
Both are same, 4%. Now I am trying same percent, sir. After that, I will do the concentration of carbon and nitrogen. That one I will do after that.
Very good. Very good.
Gagan.
Good evening, everyone. Myself Gagan M, postgraduate student, Department of Biochemistry, Jnana Kaveri PG campus. My topic is screening, isolation, identification, and characterization of xylanase from biological samples. Firstly, let me give an introduction about xylanase. Xylanase is a class of depolymerizing enzyme which plays an important role in breakdown of beta-1,4 linkage present in the xylem. And it is giving the oligoxylosaccharides and xylose as the end product. And most of the microorganisms plays a crucial role in the production of this xylanase enzyme, such as fungi, bacteria, yeast, marine algae, protozoans, etc. There are two types of xylanases that is produced from this microorganism, such as endoxylanose and the exoxylanase enzyme. And when come to the xylan usually a backbone of hemicellulose made up of a repeating unit called xylose, which are linked by beta-1,4 glycosidic linkage, xylosidic linkage. That is the structure of xylan. In the presence of xylanase enzyme, this beta-1,4 xylosidic linkage will be broken down. As a result, xylose will be obtained. And when it comes to the applications of this xylanase, why xylanase we have to concentrate on? There are huge applications in different industries like agrochemical industries, pharmaceutical industries, textile industries, paper and pulp industries. When it comes to the materials and methodologies, first collection of sample, decaying good sample was collected from the Western Ghats in the Chikkalwara, and then the obtained sample was serially diluted and it was spread plated on both nutrient agar medium as well as potato dextrose agar medium, because isolating both the bacteria and the fungal species. The obtained species was pure cultured and it was grown on xylan agar plates containing 1% xylan as the sole carbon source. After which a Congo red dye was applied and it was de-stained. As a result, it only binds to the xylan and it doesn't bind to the xylose what is produced. Here we can see the clear zone what is produced. That yellow color is the clear zone what is produced as a result of xylanase enzyme production. And then secondary screening, the bacteria or the microorganisms which is showing the clear zone I have selected and I have gone for submerged fermentation and the solid state fermentation. For bacteria, I have used the submerged fermentation, and for fungi, I have used solid state fermentation. In submerged fermentation, I have used 1% beechwood xylan as the carbon source and incubated 37 degrees Celsius for 48 hours, and it was filtered. For solid state fermentation, the different source like different carbon source like wheat bran and then corn cob was used, and it was cultured. This is the picture showing before fermentation and after fermentation. Next is the activity was determined. This is the color showing due to xylanase activity. The activity was determined based on DNS method, and the result showing out the total number of bacterial isolates what I have got and fungal isolates I have got, and the maximum activity showing isolate I have selected for further biochemical purification and further studies. This is the one fungal species I have identified, only the genus level, that is Trichoderma. And when it comes to the conclusion, xylanase have a very important role in different industries. Hence, I have selected this xylanase as my topic, and agricultural waste out what we are getting that can be utilized for the production of xylanase, and that xylanase can be applied in different applications. Thank you.
How you know the culture is isolated, they're potent, producing more than the most of the other foods.
Sir, I have referred many papers, sir. From that,
Did you compare with any other culture, or did you make a comparison between the
yes, sir. Sir, I have compared with Aspergillus, Trichoderma, two of the papers I have referred. In that paper, okay. Okay, culture, okay, sir. I got very less amount, sir, from other, what I have isolated. This is the Trichoderma showing maximum. So I have used this for further biochemical purification so that I can get more amount, more activity. Yes, sir. Yes, sir. No, sir. Sir, actually in the papers what I have referred, they mostly purified the samples. Purification step was completed, but I have not completed the purification steps. Till only isolate, qualitatively and quantitatively estimated, that's all. And purification steps, when I go, I may get more results, sir. Better results. In EXO, sir, EXO is very useful. Sir, because it will be released into the outer environment so that oligosaccharide production. Sir, endo is better, sir. Oligoxylin. So for paper production and such kind of things they use it. Yes, sir. Thank you.
Next is Ashwita Shetty. You can present your slides online.
Good afternoon, ma'am. I've submitted my video. Have mailed my video.
Hello, Ashwita Shetty.
Sir, I made my conference.
Hello, Ashwita Shetty. Can you hear me?
Yes, sir.
You need not have to explain your presentation. Just to show your slides and just 2 minutes we will give you. Just to show the slides. The screening of oral probiotics bacteria from the geriatric cases. You are from which place, Ashwita Shetty?
Assistant Professor,
JSS College,
Education and Research.
Yeah,
My topic for today's presentation is entitled with the screening of oral probiotic bacteria from the geriatric cases. Coming to the content, introduction to probiotics, overview of oral probiotic profile, common occurrence in the body, importance of oral probiotics in elderly people, observed effects of the oral probiotics, etiological factors that suppress the probiotics, and conclusion.
Only 2 minutes.
Coming to probiotics. What are probiotics? These probiotics are the living organisms or food products containing live microorganisms. These are beneficial to health of the host. Probiotics are usually obtained in or isolated from the living system. These organisms, people's oral cavity. These include lactobacillus, effect of pathogenic microorganisms. They modulate the immune response of the host at local and systemic. Their lifestyle undergoes different, fighting several oral disorders, include probiotic oral cavity. And long-term clinical trials are much needed. Thank you.
Greetings of today. My name is Divya Gaze, studying in Jesus Academy of Higher Education and Research, Mysuru, Department of Microbiology. It has like a diabetes, antiviral therapy, and etc. The term Jatropha is usually it composed of maybe bioactive components, and it can be called by different names like Staphylococcus aureus, which is one of the skin-associated bacteria. Regions having a scanty to heavy rainfall and more benefits than the which are recognized as the different kinds of medicinal impact. The mainly the stem latex contains the medicinal uses which having a wound healing property and advantages of this plant. This can be used for the erosion control and medicinal uses. These components can be identified as a parts of this different parts of the plant. Thank you. Hello everyone, greetings of the day. I'm Rakshita Ranian, research scholar, Department of Microbiology, availability of the effective therapies. For decades, about 1.5 million people died from TB, which Karnataka approximately of PTB cases, which makes the Karnataka tops among the other states, Mysuru respectively. Going for the microbial pathogenicity. From the selection, is an amplification of the selected virulent genes. These genes were detected in the isolated DNA, which has found to be distributed in the age groups and the mostly seen in men, whereas the transmission rate in these groups were found to be high. Out of 120 samples selected in developing infection management, preventive strategies, as well as strengthening in remedial measures in the areas indicated. Thank you, one and all. Good morning, everyone. Myself, Kalluri Rameshwari KR, a research associate from the Department of Microbiology. I have carried out my research under the guidance of Dr. Sumanaki, Department of Microbiology, JSS Academy of Biology, inside the London. You know that the Mycobacterium tuberculosis cell is made up of mycolic acid, which is a very thick, and in order to defeat that, so here, hence, the limited permeability of mycobacterial cell membrane, particularly with the outer membrane proteins, confers the inherent resistance to most hydrophilic medicines. The hydrophilic compounds instead of MTB. In the figure, we causing the extrapulmonary TB. It differs in the length and modification depends for the further treatment of MTB and the cases which manage the disease at the early agent extracted preparation using the liposomes and planar lipid bilayer showed that two materials had a similar pore forming properties. So OMPA TB is different from either of mycobacterial porins described as so forth. So in this regards, this is the report of porine-like molecule of MTB, that is Mycobacterium tuberculosis. The porine
ಏನ್ ನಿಮ್ದು ಪೋರ್ಷನ್ ಎಲ್ಲಿ ಮಟ್ಟ ಮುಗಿಸೇನಿ 3 ಚಾಪ್ಟರ್ ಮುಗ್ದಾವ ಎಷ್ಟು ಮುಗ್ದಾವ ಫರ್ಮೆಂಟೇಷನ್ ಪ್ರೋಸೆಸ್ ಎಲ್ಲಿ ಮಟ್ಟ ಬಂದಿದೆ
Please share your slides. Amrish sir, please share your slides.
ಅಲ್ಲ ಐಸೋಲೇಷನ್ ಸ್ಕ್ರೀನಿಂಗ್ ಸ್ಟ್ರೇನ್ ಡೆವಲಪ್ಮೆಂಟ್ ಫರ್ಮೆಂಟರ್ಸ್
In about 5 to 10 minutes, the valedictory function will start.
Hello. 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 Hello.
It's like magic, but real. Good evening, all. I, Sujana, from 2nd MSc Microbiology, take this privilege to welcome you all for the valedictory session of the National Conference on Integrated Microbial Technology for Sustainable Future, IMTSF 2022, organized by the Department of Studies in Microbiology, Gnana Kaveri PG Center, Chikkalwara. Let us begin the program by invoking the blessings of Lord Almighty. I invite Mridula, Tanushree, and Prarthana from 1st MSc Microbiology to please lead us in the prayer. Sorry, I request all the dignitaries to occupy the seats in the dais. I request Dr. Ashok V. Bankar, sir, Dr. Prakash M. Halmani, sir, Dr. B.S. Gunashri, ma'am, Professor M. Jayashankar, sir, Dr. I.K. Manjula, ma'am, to please come on the dais and occupy the seats. I request Dr. A.K. Vinu, ma'am, to join the stage. I invite Mridula, Tanushree, and Prarthana from 1st MSc Microbiology to lead us in the prayer.
ಮಾತೆ ಪೂಜಕ ನಾನು ಎನ್ನಯ ಶಿರವನಿಡುವೆನು ಅಡಿಯಲಿ ನಿನ್ನ ಕೀರ್ತಿಯು ಜಗದಿ ಮೆರೆಯಲಿ ಒಂದೇ ಆಸೆಯು ಮನದಲ್ಲಿ ಎಡರು ತೊಡರುಗಳೆಲ್ಲ ತುಳಿಯುತ್ತ ಮುಂದೆ ನುಗ್ಗುವೆ ಬಲದಲ್ಲಿ ನಿನ್ನ ನಾಮ ನಿನಾದವಾಗಲಿ ಶ್ರಮಿಪೆನಾ ಪ್ರತಿ ಕ್ಷಣದಲ್ಲಿ ನಿನ್ನ ಗೌರವಕ್ಕೆ ದುರು ಬರುವ ಬಲವ ಮುರಿವೆನು ಛಲದಲ್ಲಿ ಜಗದ ಜನನಿ ಭಾರತ ಇದ ಕೇಳಿ ನಲಿಯುವೆ ಮನದಲ್ಲಿ ಮಾತೆ ಪೂಜಕ ನಾನು ಎನ್ನಯ ಶಿರವನಿಡುವೆನು ಅಡಿಯಲಿ ನಗುತ್ತ ನಲಿಯುವ ನಿನ್ನ ವದನವ ನೋಡಿ ನಲಿಯುವುದೆನ್ನೆದೆ ನಿನ್ನ ದುಃಖಿತ ವದನ ವೀಕ್ಷಿಸಿ ಸಿಡಿಯುವುದೆನ್ನಯ ಹೃದಯ ನಿನ್ನ ಮುಖದಲ್ಲಿ ಗೆಲುವು ತರಲು ನೀರುಗೈಯುವೆ ರಕ್ತವ ಎನ್ನ ಕಣ ಕಣ ತೇರು ಬಸಿಯುವೆ ಪೂರ್ಣ ಜೀವನ ಶಕ್ತಿ ಮಾತೆ ಪೂಜಕ ನಾನು ಎನ್ನಯ ಶಿರವನಿಡುವೆನು ಅಡಿಯಲಿ ಶಿರವನಿಡುವೆನು ಅಡಿಯಲಿ ಶಿರವನಿಡುವೆನು ಅಡಿಯಲಿ.
I now invite Dr. A.K. Vinu, ma'am, Faculty, Department of Microbiology, to formally welcome the gathering.
A warm good evening to all of you. As we have come to the end of today's national conference, now we have gathered here to conclude the conference formally. So on this behalf, at this moment, I would like to welcome everyone present here. First of all, I would like to welcome the chief guest of this valedictory function, Dr. Prakash Halani, CFTRI Mysore. Welcome you, sir.
And also I welcome another chief guest of this function, Dr. Ashok Banakar sir. Welcome you, sir. I extend my warm welcome to Professor M. Jaishankar sir, organizing secretary of this conference. Welcome you, sir. And I also welcome chairperson of the Department of Microbiology, Dr. Guneshri. Welcome you, ma'am. And Dr. I.K. Manjula, Department of Microbiology. Welcome you, ma'am. And I also welcome Mr. Jitendra, Department of Computer, that is IT, and technical staff from Bangalore University to this session. Welcome you. And I welcome students, teaching staff, non-teaching staff of Department of Microbiology. And also I welcome the delegates who are present here for this valedictory function. And also I extend my warm welcome to all the research scholars of the Department of Microbiology as well as Biochemistry, and all the technical staff and everyone present here in this valedictory function. Thank you one and all.
Thank you, ma'am. I welcome Dr. A.K. Vinu, ma'am. Now, I request Chief Guest of the valedictory function, Dr. Ashok V. Bankar, sir. It's, sorry. I request Pramila to give the feedback. Pramila, CFTRA. Good evening, everyone. First of all, I want to thank all the members of the institute who have organized this conference and making us to know about the various fields involved in microbiology. Thank you, everyone, ma'am. And the organizing secretary also, Jaishankar sir also. Thank you, sir, for making us aware and giving us this opportunity to participate. And your timeliness also, you want to make everything in time, and you are very active. So even in this age, you are very and we have enjoyed both the days. Thank you, everyone. Bye. Now I call upon Abhishek PM, scholar from Wayanad.
I am thankful to this department to organize such a wonderful conference. Actually, I was present in 2014. We conducted a conference under the Professor Jaishankar sir also, and this conference also, like last time feedback told that she said like in this age, sir is running like a young person here and there, and there was long nowadays like is very active, and moreover that content delivered in this conference is very meaningful, and especially today's and tomorrow's even both online and offline contents. So the organizers, once more, thank you. Thank
If anybody is interested to give the feedback, please come on the stage.
Good evening. I am Rana Tamer Al Khafaji from Iraq. I came first time to India 2019. So I hope to speak in a very good way because in my country, as I mentioned, general language is Arabic. So I was very, very weakly compared with other students. I am very happy today to be here with my teachers and my good guidance, MG sir, and all my friends, new friends specifically. And I want to say thank you for everyone support me and help me here in every moment since I reached to India. Actually, I visited many countries, but in India, it's very different. People is very nice and simple and support me. Thank you very much. Thank you.
Now I request the students from the Jnana Kaveri campus to give the feedback. Gagan of Biochemistry.
Good evening, all. First of all, I want to thank everyone from Department of Microbiology for allowing us to attend the conference and giving permission to do my oral presentation. First of all, I didn't register for my oral presentation or for this conference. The faculty supported very much and told us it will be useful in the future. So we have registered. Thank you, one and all.
I request Arun sir to come on the dais.
Hello. First of all, ಈ ಕಾನ್ಫರೆನ್ಸ್ ಆಯೋಜಿಸಿರುವ ಜಯಶಂಕರ್ ಸರ್ಗೆ ನಾನು ಒಂದನ್ನು ಹೇಳ್ತೀನಿ. ಆಕ್ಚುಲಿ 2 days ಆನ್ಲೈನ್ ಮೋಡ್ ಮಾಡೋಕೆ ಜಯಶಂಕರ್ ಸರ್ ಎಷ್ಟು ಕಷ್ಟಪಟ್ಟಿದ್ದಾರೆ ಅಂತ ನನ್ಗೆ ಗೊತ್ತು. ಅಲ್ಲಿ ಪರ್ಮಿಷನ್ ತಗೊಂಡು ಈ ಆಫ್ಲೈನ್ ಮೋಡ್ ಮತ್ತು ಆನ್ಲೈನ್ ಮೋಡ್ ಎಲ್ಲ ಸಕ್ಸಸ್ ಆಗಲಿ ಅಂತ ಅವ್ರು ಬಹಳ ಕಷ್ಟಪಟ್ಟಿದ್ದಾರೆ. ಆದ್ರಿಂದ ಅವ್ರಿಗೆ ನನ್ಗೆ ಒಂದು ಥ್ಯಾಂಕ್ಸ್ ಹೇಳ್ತೀನಿ. ಮತ್ತೆ 2 days ನನಗೆ ಇಲ್ಲಿಗೆ ಬರೋಕೆ ಇದು ಮಾಡಿ ವರ್ಕ್ ಪ್ರೆಷರ್ಸ್ ಕಮ್ಮಿ ಮಾಡಿದ್ದಾರೆ ಅಲ್ಲಿದ್ದೆಲ್ಲ. ಅದರಿಂದ ಒಳ್ಳೆ ಸಕ್ಸಸ್ ಆಯಿತು. ಮತ್ತು ಯೂಟ್ಯೂಬ್ ಲೈವ್ ಎಲ್ಲ ಸಕ್ಸಸ್ ಆಗಿದೆ. ಯೂಟ್ಯೂಬ್ ಲೈವ್ ಈಗ ಪ್ರೆಸೆಂಟ್ 1032 ವ್ಯೂಸ್ ಆಗಿದೆ. ಅದರಿಂದ ಒಳ್ಳೆ ಇದಿದೆ, ಒಳ್ಳೆ ಆಪರ್ಚುನಿಟಿ ಸಿಕ್ತಂಗೆ ಆಯ್ತು. ಮತ್ತು ನಮಗೂ 2 days ಮೈಕ್ರೋಬಯಾಲಜಿ ಸಬ್ಜೆಕ್ಟ್ ಕಲ್ತಂಗೆ ಆಯಿತು. ಮತ್ತು ಆ್ಯಕ್ಚುಲಿ ನಾವು ಬಂದು MTech ಬ್ಯಾಕ್ಗ್ರೌಂಡಿಂದ ಅದು ಒಂದು ಕಾನ್ಫರೆನ್ಸ್ ಇದೆ. ಫಸ್ಟ್ 2 days 24 hours ಕುಂಟಿರೋದು ಇದೇ ಫಸ್ಟ್ ಟೈಮ್. ಅದರಿಂದ ನನಗೂ ಒಂದು ಆಪರ್ಚುನಿಟಿ ಸಿಕ್ತಂಗೆ ಆಗ್ಬೇಕು. ಥ್ಯಾಂಕ್ ಯು ಸರ್.
I request delegates from Dharwad to speak a few words. Good evening, everyone. First of all, I would like to thank our Jaishankar sir for giving us the chance to participate in this special conference. And along with this, I would like to thank Hasina ma'am and Karthik sir who was continuously contact with us and were updating us with the things going on here. As we were far away, they were always in contact with us and they were so supportive for us. So thank you ma'am and thank you sir. Along with that, the coordinators who were here, they were so supportive and they were helping us to helping us to go through whatever going on in the conference. So thank you guys, and it was a great pleasure to be here. Thank you one and all. Now I request Karthik Bilyappa sir to speak few words.
A very good evening, everyone present here and respected guests. So, validatory function, it is coming to an end. So, from last 2 days, we had worked a very lot and you had gained more knowledge, I think. And we are here mainly to thank technical team who had arrived from Mangalore, and Jitendra sir also, and Arun sir and his team with their equipment, and without any disturbance, they had successfully conducted this online conference. And as well as I also want to note that our online presenters also there, and I would like to welcome them also. We had forgotten them and they are watching us online. So I welcome them also, and I like their patience till they are listening to us. Then the whole session is recorded. At the break time also they are online. So they are asking us to whether the link is there, whether the presenting it is time, and we had somewhat because of busy many presenters were there and it was unable to communicate with them because there was a lunch break, snacks break, everything. So I thank the online candidates who are present now, as well as I thank all my team members, research scholars, as well as MSc students, both years, and they had worked day and night for this program to be a grand successful. I thank all the research scholars of Department of Microbiology as well as the MSc students of Department of Microbiology. And coming to this, the initiation, as you all know, there is an initiation. So this conference was initiated by Professor M. Jaishankar sir, our guide, and he is one person who made it a very successful one. And in support, our chairperson, Dr. Gunashri Madam, and she is also a main player here who made this program a very successful. And from yesterday, we had more chief guests, that also a vice chancellor from Gulbarga University and former vice chancellor Muthuchalam from Salem University. So these are main dignitaries who are present. So, and as well as our Honorable Vice Chancellor, Professor Subramanya Edappa sir, was also online and he had given his talk and blessings through online for our department. And I also want to tell about reception committee, that is Dr. I.K. Manjula ma'am, who had neatly and systematically organized everything from the registration to the end. And also I want to thank our faculty members, Dr. A.K. Vinu ma'am, guest faculty, Rakshita madam, and Sharanya, everyone for their support. And at last but not least, we want to thank our Srinath sir, who had come from Mangalore for this program, and he is also a branch of our microbiology department, now currently working as special officer in Mangalore University. And I also thank our university for granting the fund for this conference. And I thank everyone who are present here as well as online for this conference. Thank you.
Thank you all for sharing your feedback. Now, I request Dr. B.S. Gunashri ma'am, Chairperson, Department of Microbiology, to speak few words about the conference. Respected Professor N. Jaishankar sir, Dr. I.K. Manjula ma'am, Chief guest of today's valedictory function, Dr. Prakash Halami sir, and Dr. Ashok Bankar sir, and research scholars, IT staff from Mangalore University and from the campus, PG campus, all the research scholars from PG Center as well as from various other universities and institutes, and PG students as well from other universities and our Gnana Kaveri campus. A very good evening to one and all. And I feel very proud to say that with all the limitations in this PG Center, we could able to successfully complete this 2 days national conference on the theme, Integrated Microbial Technology for Sustainable Future. So the feedback as well as the lead speakers, their opinion about the conference, what we have got is the topic or the theme what we have given for these 2 days national conference is very much apt. And we are really thankful to all the lead speakers who made their way come both online as well as offline, and they have shared their valuable knowledge through their research outcomes and their academic knowledge. And I'm very thankful to all of them. Actually, and I'm very thankful to our university for granting us with the funds because of which it was able to, and we were able to conduct these 2 days national conference. And actually, we had no expectations of this much of delegates. And at least until this valedictory function, we just thought because of the climatic season, we thought some of the delegates might not come today, but I'm very happy that all of you are here and made this 2 days national conference a very successful one. I thank all my faculty members on my own behalf because we have a formal vote of thanks. I don't want to take much of my time, but still it's my plain duty to thank one and all for making this 2 days national conference a very, very successful one. I have my heartfelt thanks to all the faculty members of my department who were very much supportive for me, and all the research scholars who have worked day and night, and even my PG students who have taken a lot of involvement in each and every aspect, and even the students who have entertained all of us yesterday evening, because the whole day was really hectic, listening to a lot of speakers speaking on various aspects, and they have really entertained us for about 1.5 hours. And we could make out there are a lot of talented students, hidden talents available in our campus. So I'm very happy for that. So I'm really very happy for successfully completing these 2 days national conference. I'm very proud to say that, and I'm very thankful to each one of you, and thank you so much for this opportunity. I'm very happy to say that we had both online and offline sessions. I'm extremely thankful to Mr. Arun and Mr. Hari Prasad who made their way, because we had a lot of confusions whether they are coming here or not. Until the last minute, we had a lot of confusions because they were under dilemma whether we are going to Chikkalwara or not. Because there was, I think there was another minister's program which was, I think it is today. So their plan was actually getting cancelled, but we were worried because we have already announced to the delegates that we also have an option for online mode of conference.
but they just took a lead and they have taken a lot of pains coming over here and making this program a very successful one. So I request Mr. Arun and Mr. Hari Prasad to come on to the dais, and we would, on the token of love and affection for you, we need to, I request Professor N. Jaishankar sir to give away the momentum to Mr. Arun and Mr. Hari Prasad. A big applause for them because both of them have taken a lot of pains and they have made this event a successful one. Thank you, Mr. Arun and Hari Prasad once again. And I am also very thankful to Mr. Jitendra sir. He was here all the time with us, and he knows better than any of us about the internet and other broadband capacities and other things. But he also has taken lots and lots of pains, and he has coordinated with those two people who came from Mangalore University, and he is also one of the important contributors for a successful conference. Thank you, sir. Thank you so much. Thank you, Ananda.
Now I request Chief Guest of the Valedictory Function, Dr. Ashok V. Bankar sir, Assistant Professor, MES, Garware, Pune.
First of all, thank you all the students. Because of you, this conference is successful. I want to give the special time to Jai Shankar sir and whole team. So if the leader is good, everything will be good and fine. And you can see that so many students said that he is very young man. So I have that experience because he visited Pune also, and that young man also came to Pune. And at that time, we spent lots of time together, 2-3 days, I think, sir, right? And then during discussion, I found that the person is very enthusiastic and very interested in conference organizing as well as to give the speech and talk. So I want to give special thanks to, because of sir, I am here. Manjua madam is also there. She was with me in a refresher course. So I want to thank Manjua madam and all colleagues. I want to give the special thanks to the students also, and all organizing committee, all PhD scholars and MSc students. And it is very important thing is that why to attend conference. The first thing is that because of this is a scientific session, this is a scientific feast, and this is the interaction. During this interaction, your personality will be changed, your confidence will be built, and you will get so many information through this interaction. So automatically your interest will be enhanced. Okay, automatically MSc student will feel that I should do something because I saw poster. Okay, I didn't comment anything. I was just observing. The students are enthusiastically explaining each and everything. During the explanation, everything will be changed. Your personality will be changed, and you will never understand that. So there is a change in your personality. But automatically, as you explained, you are facing so many people. So you are learning defense. And that's why the conference is important. You should attend not only in this campus, but out of the campus also you can go and you can visit many places. PhD scholars, this is your good training because PhD is not only the degree, but it is actually it is a training. And once your training will be finished, after PhD, actual research will be start. So don't think that this is research. This is only learning process. PhD is only learning process. After PhD, your original research will be start. At that time, you will be trained. Then your mind will be innovative and you will think different from other people. So what is the difference between the normal person, I mean the ordinary person and scientific person? So this is the difference. You are thinking in scientific way. You are raising the questions. Your thinking is innovative and you can see so many things in a small bacteria. Okay. And this is possible because of your vision, your thinking, and good ideas should be developed. And then it will be beneficial. I always tell to students that what is the difference between you and me? I mean, what is the difference between teacher and student? There is only one difference I tell them. You are late born, we are early born. That's why we are here, you are there. Otherwise, there is no difference. We have lots of idea, and we should welcome that ideas also. So you all are the young mind, and that young mind should be inspired, encouraged, and I wish all the best, that never be discouraged. Even during your PhD, you know that in the Belgium there was one program. So how to handle your supervisor, okay, and how to come out from the trace in the lab. So 111 countries students, PhD students, they have participated in that program. And in that program, so they studied that how trace is created during the PhD. 1 year you are working and you are not getting result. So what happened to that student? So that psychology needs to be understood. And then you have to think positively. That is the reason. And that's why the wavelength between supervisor and student is very, very important. So while MSc student, while choosing your supervisor, think twice, thrice that my wavelength is matching or not with my supervisor, because that is also important part. Okay, that I studied in that workshop. So if the wavelength is matching, then what will happen? It's become very easy to interact with your supervisor. And supervisor also get very easy to interact with the student, and then there is a development. So through this conference also, when you attend in outside, so MSc student will come in contact with many scientists, different people from other countries also. So if you want to do PhD at that time, that interaction is very important with that people. So its wavelength is matched. So I will give my example. My wavelength was matched with my supervisor. That's why I could get paper. If she is not cooperating with me, what is the use of my talent? What is the use of my hard work? So equal importance of supervisor is also there. Equal importance of student is also there. So proper interaction, proper communication, and proper, whatever the work you are doing, to communicate to your guide, you have to communicate to your supervisor, and you have to tell your ideas. And then this problem will be solved. If your communication is lost, you will lose the contact with the guide. And the result is that there will be the development of misunderstanding. Guide will say that my student is not doing anything. Student will say that my supervisor is not doing anything for me. And in this way, frustration will be built. And that should not be built. So it should be break through the communication, through understanding, through the discussion. So I hope that this is good guideline for the MSc students. And whenever you are choosing your career at that time, whatever the hints I have given while choosing your guide, so it is very important the wavelength should be matched. And I hope this conference is beneficial to all the students, PhD scholars, and this conference was also not only useful to you, but it was also it was nice experience to me also. I attended several, 36 national and international conferences, but this was this conference was again unique. Why unique? Because I saw lots of students here. That is rarely happening in the other places. Thank you so much.
So sir had well said, and he had inspired us what to do next and how we should go ahead without any problems, and we should concentrate. So before that, before our chief guest talk, I would also welcome our non-teaching staff and our security guards, as well as our Robin sir, who had worked 2 days continuously and taken a photograph, a beautiful photograph of us. So I also will welcome Robin sir. He should be on dais here. So he was continuously taking the photos, and he had covered almost all these 2 days with his camera. As well as I would also want to thank Mr. Gangraj sir, lab assistant sir, and we had forgot him, and I think he had left, so I want to remember him. Okay, I would like to hand over the mic to chief guest.
Now I invite chief guest Prakash M. Halami sir Chief Scientist, CFTRI, to speak a few words about the conference.
Good evening, everyone. Professor Jaishankar, Dr. Ashok, Dr. Guneshri, Dr. Shanta, Dr. Vinu, other faculty members. More importantly, the participants of this today's national workshop on integrated integrated microbial technology for sustainable development. Last 2 years, definitely we didn't had much offline activities because of the COVID pandemic. And then only people realize how the microbial world is, how it is associated with us. All these days, we, rather than killing the microbes and viruses, we did not do much, maybe to some extent. We tried to induce the immune response. Whether we are young or old, everyone was talking. Actually, I am also equally young. In fact, few days back, one of my very senior, he just casually telephoned because his daughter wants to come to Infosys Mysore, and he said, "Sir, are you retired?" And it happened actually a month back only. I appeared for one very interesting interview, and I was about to tell him that my career is going to start now because I am searching a new job. The job which is existing, it is just a by accident or knowingly, unknowingly it came, but the job which I am going to do, that is actually the job. That is what I wanted to tell. Madkeri or Coorg or Kodagu, or you call also Markera also, I guess. We from CFTRI actually explore most of the time as a picnic spot. Though I was knowing this PG center from quite long time, and it so happened most of the vice chancellor also, whether it is a Professor Saveriyappa or Dr. Gopal or previous one, the Dr. Shiva Shankar Murthy, because he was from the Mysore University. Most of the colleagues, and of course, I know the Dr. Jaishankar sir from a quite long time. We are meeting in one of the activities. And from that angle, the way he has organized this 2 days conference where we had at least, I think, 5 to 6 lead lectures, and from the diverse field, and was a very good opportunity for most of the budding scientists to take up the research activities. Here actually is a very good combination of microbiology, biochemistry, and you have again the botany also, yes. So all these are actually the nowadays the interdisciplinary subjects we need to pursue the research, right? And from that angle, we could see that actually it was a very good blend of science with the research activities, the academic activities, the skill of most of the volunteers, especially which I was actually recording and observing. I didn't know that they are not only the volunteers, but they are actually the competitors of the poster oral presentation, the competitors also. And most of this, the participants who were actually had a very good presentation of the even oral presentation also. Though it is raining continuously from yesterday, and when we were actually telling our colleagues we are going to Madkeri, and then first thing they told, no, it is a landslide in there, don't go. Or it is heavy raining. But at least we didn't have till now problem. But of course now since we have to travel back to Mysore, and then I think most of these participants also will be going. But definitely with reference to the organization is concerned, it was much more the concise way. It was because having that many lectures, lead lectures with a question answers, the oral presentation, the way compactness it was maintained, and then the learning experience, which most of the people definitely got, right? Interacting with the people, and then the diverse field, starting with the nanobiotechnology or the polar microbiology, and then today the environmental microbiology. All these fields are actually the emerging field in the microbial biotechnology, where you see that the large number of microbial world which requires to be exploited, not only for the taxonomy purpose or only for the looking for any fine chemicals or the metabolites, but then looking for their diversity, using for the different applications, you know. So just I want to tell with reference to either it is a oral presentation or the poster, or even if I have to also make any proposal or anything, it is applicable to me also that we have to have a first a thorough literature search, right? I'm not only pinpointing or questioning you people, it is applicable for me also. Just yesterday only when I was here, for one project was getting submitted. And then definitely it was required that whether we have seen the sufficient literature or not, national status, international status, who all are people working on that area. And then you ensure that there are no loopholes, that the whatever you read is a clear. So such a rigorous discussion is also required, okay, with your colleague or friends or other things. And then you make actually the presentation plan of the work. Even Dr. Rachakam is telling that how you actually are transparent, how you are actually open to discuss, share with other colleagues, you know. That is required so that you actually make sure that whatever you have planned is authentic and has a relevance. Okay. Otherwise, you will be actually going perhaps in a wrong track, and that's why most of the research which we do becomes very difficult to have an reproducibility, or the uniqueness, or the novelty. You know, when we were several times in abroad, and then first question they ask is, did you see the literature? And actually, in our case, it so happened, whatever we plan, that don't work. Whatever the experiment we actually discuss, it won't work. It will be something like accidental most of the time. But no, in research, actually, it has to be well planned, pre-planned, and done, which requires definitely the positive control, enough negative control. Most of this oral poster presentation also I was actually asking this question, what was the positive control? So somebody is saying positive control was negative, negative was positive, or positive control was not there. But no, that is required. And then you does the experiment in a duplicate or triplicate. That is also is very much required because these are a biological sample. Statistical analysis is a must, you know, with a preferably again using the sufficient statistical tool. Just last week only we had one of our PhD student who was from, he has registered with Mangalore University. And then they told, no, you have to have a sufficient number of a statistical data analysis using the different tools. So this is actually a very important component in science to interpret the results, to make sure that the results which you have are the novel or new, or you are actually can able to justify how your results are useful. And then you communicate definitely to the society or the scientific community. You are definitely a number one scientist, and I think each every one of these delegates, those who are here online as well as offline, they will agree that this last 2 days, this conference was really a very educative, informative, and with a full of knowledge. Those perhaps miss due to one or other reason, either because of thinking that it will be heavy rain, or perhaps the PG center of Bangalore University, they will not be able to host. Perhaps they were wrong. And I would say that they are the losers, in other words. So you people are the lucky one. You people have definitely a bright future. Help us also to make little bit our future also bright. Help us also to become a little bit more young also. I always also consider myself also young because I take a probiotics very regularly, routinely, because not only giving a lecture on probiotics, but consuming and showing that how healthy and they are also exhibiting a health benefit is also important. And once I say probiotic, then I will never stop, but I think it is not true. I know most of you people have to go a long way, means private. So I take this opportunity to wish every one of you a good luck and congratulate you for attending this wonderful event. I congratulate the organizer also for organizing this such a useful event, and I thank you them on behalf of all the delegates to giving us the opportunity. Thank you very much.
Now, I request Professor M. Jaishankar Sir, Organizing Secretary, IM TSF, to give the presidential talk.
I don't want to take much of your time.
When I was studying BSc, I got an opportunity to meet one of the terrible minister of Karnataka. He is no more today. It is very difficult to go to him. It is very difficult to approach. Such a terroristic minister. But he was a very good intellectual minister too. That was the day I met him to institute a gold medal in Mysore University. Sir, we have this much of amount collected. We have to give it to Mysore University to institute gold medals in various disciplines. Of course, his approach was a little bit negative, but finally a success. When I was in MSc, our genetics professor was Professor N.B. Krishnamurthy. I must remember, as an internationally known professor, you may be knowing, I think, Professor N.B. Krishnamurthy. Just because of his signature, grants used to sanction. But his life was pathetic because of his misdeeds of his own son. Okay, leave it. When he was explaining the Lackaporan model, he was telling about the initiator genes, terminator genes, all these things. So these are all the things which have made me to think not only scientifically, even in the angle of social aspects. If anything to be initiated, there must be a reason. We cannot initiate simply. So I thought that the department has completed 25 years of its existence. It has grown to this extent. It has to grow further. And to grow further, there is a need of support. Support is not necessary for me. I have grown, I have done everything, whatever I have to do, I have finished it. And my dear sister from CFTR, I said that I am still becoming younger and younger. I have achieved whatever I have to achieve, I did from a rural background, I got it. I am satisfied. Professionally, I am fully satisfied. But supporting for the development of my subject, for the sustainable future. So that was there in my mind. So I used that particular 20 years of establishment of this department. Also the International Year of Sustainable Future. And we have created this science. So occasion is created. Occasion has been narrated and explained in such a way that it should go to the deep of the mind of the faculties. Then it is discussed in the head of the institution, Vice Chancellor. And it is not easy for a Vice Chancellor to say no to Jayashankar. In one way or the other, they must say. That is how I impressed him. So he has also accepted. Now, because of all your efforts, 2 days national conference has become fully successful, materialized. we have a group of genes will initiate, but the other genes have to activate. Going somewhere else and getting the ribosomes and amino acids together and making the protein. So that is you have done. That is because of you. And that is because of my great friends. 9 talks, 9 lead talks, 1 keynote address. In no conference such a long, heavy talks will be arranged. It is only here. And the time that we have managed, and all the lead talk people, they were, if Jaishankar is organized, means definitely there will be something different. That is, they know and they respect me. I am highly grateful to them. I never forget them in my life. So this is because of UP. But one thing has happened in the middle. Actually, this inauguration, this valedictory would have been different. But I become little bit busy because of my personal work. And nobody knows that, except Professor Dayanand Aksar. If I am standing here today for this valedictory function, is because of Professor Dayanand Aksar. We used to walk 2-3 kilometers in Kerala. Professor Dayanand, I always be the chairman. Professor Dayanand Aksar always be the member. For so many committees and meetings we were going together. I don't know how this has happened. I don't want to narrate this particular issue. I was expecting more delegates from different parts of the country. Because of that personal reason, I could not interact them in appropriate time when they were calling. Otherwise, gathering would have been different. But one thing I am request all of you. If anything, this is told by various great people. If good things come from any part or any direction, please try to catch. If any bad things, bad issues, bad informations come, immediately you made them to reject. Always invite good things. Even if your enemy gives a good idea, please welcome. There was a lot of negative energy in postgraduate campus, Gnana Kaveri. By doing good things, the negative energy has been kicked out. Now I hope that because of this 2 days national conference, entire negative energy has gone out. That is because of UP. Because you come with a good heart, good mind, good intention, learning. You are all interested. If you see your face, if you see your eyes, I definitely say you people are eagerly waiting for the opportunity to serve the nation, serve the country. That type of look I want, that you have given. I am really lucky to have an audience of this type. My dear friends, I will not take much of your time. We will have a different conference, probably in a different place in future. Let us think about things in future. Now, this is the time for me to announce the Best Work Presentation Award goes to Neelam Mishra. the best poster presentation award has gone to Remy Jacob. If anybody is there, these two people are here. So they can collect the award certificate from the chairperson of the department later. My dear friends, I will tell you only another one or two words. Please recollect the keynote address again. Please recollect the lead talks given by various microbiologists. Professor Lipton, you have heard Lipton's talk. He is more than 73 years old, but still he is doing work. Why don't you youngsters do? You please come forward. Every day we eat breakfast, we take lunch, go for coffee, tea, biscuits, all those things, and even dinner. That should be, your body should be in a position to make all those things, satisfy your body, satisfying your mind, satisfying your thinking, everything. Otherwise, all those what you get full day becomes waste. Always do. Our Prime Minister is telling, even former Prime Minister was telling, so many dignitaries, education itself told, we have to work for the development of the country. If you work for the development of the country, along with that, we will also grow, definitely. If you try to grow alone, definitely not. ನೋಡಿ ಸುತ್ತಮುತ್ತ ಎಲ್ಲ ಫಾರೆಸ್ಟ್ ಇದೆ, ಒಂದೊಂದೇ ಮರ ಬೆಳೀತಾ ಇದೆಯಾ? Are the rhizospheres of one plant helping the only one that plant? Is it not helping the other plant? So this is natural phenomena. We must help together. Those who do not help or do not cooperate for the good things, they will definitely, the society will remove them. So with all these things, I am happy all of you have come forward. If any, I asked you for the feedback. Any lapses in the food, any lapses in the accommodation, any lapses in our activities, attitude, all this. Nobody has said that the talks were too much. Nobody said the food was shortage. Nobody said our attitude was wrong. You are all telling everything good. That means the credit should go to you people. So believing you, expecting you, we have asked and requested those lead lecturers to give it up. If anything has given you any benefited from this 2 days conference, I am really happy. I am really proud. Definitely this has, your support has given me more energy to do more work in my future endeavor. I thank you each and every one of you for giving me this opportunity. My colleague said, I don't want to have, when brochure was prepared, I was hesitating. Can I write organizing secretary Jayasek? Actually, I was not interested. Muthutalian said, if your name is not there, nobody will come. So, after analyzing that, then I have worked. Actually, my dear friend, if I talk more, I may cry. I have some personal problems. I wanted to leave today. I have to travel 1,200 kilometers today night. Kindly bear with me. I thank you one and all for giving me this opportunity. Thank you. If any mistake or if any lapses have happened, and I take the responsibility. Thank you, Olana.
Time has come to draw a curtain to our program. Now, I invite Ms. Hasina Sheikh, Research Scholar, Department of Microbiology, to give the vote of thanks.
Good evening, everyone. As we have come to the end of this conference, I would like to take the privilege to thank one and all present here. First and foremost, I would like to thank Dr. Ashok V. Bhanakar sir for sharing his valuable knowledge on nanotechnology. Thank you, sir. I convey my heartfelt gratitude to Dr. Prakash M. Halami sir for sharing his knowledge about probiotics. Thank you, sir. My sincere thanks to Professor M. Jayashankar sir for organizing this conference. Thank you, sir. My deepest thanks to Chairperson of Microbiology Department, Dr. B.S. Gunashri ma'am, for her support. Thank you, ma'am. I also thank Dr. I.K. Manjula ma'am, Assistant Professor, Department of Microbiology, for her support. Thank you, ma'am. I would like to thank Faculty of Microbiology Department, Dr. A.K. Vinu ma'am, for her constant support and guidance. Thank you, ma'am. I also like to thank Dr. B.S. Srinath sir, Assistant Professor and Special Officer, Mangalore University. Thank you, sir. I especially thank faculty members of Microbiology Department, Ms. Rakshita MP and Ms. Sharanya MG. Thank you. I would like to thank non-teaching staffs of Microbiology Department, Mr. Gangaraju sir, Mr. Mallesh sir, and Mr. Prashant for their support. Thank you. My heartfelt thanks to all the lead speakers who gave their talks from yesterday by sharing their valuable knowledge. My sincere thanks to media person, Mr. Zami sir, Faculty Department of MA in Canada. Thank you, sir. I deeply thank Mr. Robin sir, Faculty Department of Social Work, for capturing pictures of today's conference. Thank you, sir. I would like to thank all the technical staffs, Mr. Jitendra sir, Mr. Arun, Mr. Hari Prasad, Mr. Manjunath, Mr. Purnachandra, Mr. Vinay, Mr. Shivkumar, Mr. Mallikarjun for their continuous support in handling those technical sessions. Thank you. I'd like to thank Mr. Nagaraj, head cook, and his team for providing us delicious food and refreshment for these 2 days. Thank you. I'd like to thank housekeeping staff for their help in making necessary arrangements. I'd like to thank security in charge, Mr. Etish, and his team for constant cooperation. I'd like to thank all committee volunteers for their hard work and help for making this conference a successful one. My heartfelt thanks also goes to all the delegates and participants for their active participation. I would like to thank all MSc students, research scholars of Microbiology Department for their continuous support and involvement in making this conference successful. Thank you, Anand.
Thank you, ma'am. I request all of you to raise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंधु गुजरात मराठा द्राविड उत्कल वंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाता जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 जय